His main research contributions have been in the epidemiology of streptococcal infections, in their sequelae in India and Southeast Asia, and epidemiology of plasmid-mediated drug-resistant and virulence factors. He authored more than 150 publications in national and international journals and has written the Hindi translation of microbiology by Cruikshank in those days. He was awarded FRC Path and Fellowship of National Academy of Medical Sciences and IMSA, Dr. Y.S. Narayana Award, B.C. Roy Award, and Dr. S. Agarwal Oration, Dr. H.I. Jhala Oration, and Lifetime Achievement Awards for IMM. He was instrumental for introducing the reorientation of medical education in India and was responsible for finalizing the national medical entrance examination patterns for MBBS and postgraduate. He has also been the president of Indian Association of Medical Microbiology in 1982. So in his memory, your life was a blessing, your memory a treasure, and your love beyond words and missed beyond measure. We will first begin with the first presentation of the Junior Best Paper Award. So can I please request the first person, as indicated by mail to you, the code number, to please come on the stage. बेटा KB1 बच्चे को तो बुलाओ, बच्चा कहाँ है? KB2, KB6. These students were here a moment ago.
A very good morning to all the speakers, chairpersons, and respected delegates. I'm here to present my topic entitled Association of COVID-19 Vaccination with the Breast Milk Anti-SARS-CoV-2 IgA-IgG, a comparative study between single dose and double dose vaccination group. So I'll be dealing initially with the introduction, the methodology, results and discussion, and in last conclusion. So starting with the introduction part, so breast milk is regarded as the most essential nutrient for the new-term babies, especially when we are talking about the preterm ones. It contains different bioactive compounds, including antibodies, and contributes to immune defense of infants. So the recent pandemic, COVID-19 pandemic, has emphasized the importance of vaccination in controlling the disease. However, limited research has evaluated antibody response in the breast milk in the vaccinated individuals. So this study aimed to examine the presence of Ig and IgG antibodies in the breast milk of lactating mothers. It also evaluated the association between COVID-19 vaccination and presence of antibody in the milk, and which contributes to our understanding of passive immunity in the breastfeeding infants. So overall, it's a study aimed at assessing the presence of SARS-CoV-2 antibodies in the milk of lactating mothers. So this study was prospective hospital-based observational study which was conducted for 1 year duration, and the sample size was 151. For this study, it includes 151 healthy lactating mothers who were willing to donate their milk post-counseling. And this study excluded those which were with any pre-existing medical condition that could affect breastfeeding, and also excluded those mothers who were received any vaccination other than COVID-19 vaccination. So the samples were collected from the milk bank after proper counseling of the lactating mothers by lactating counselors, and taking samples for some serological tests like HIV, hepatitis B, C, and syphilis. The breast milk was collected with the breast pump under hygienic conditions. 2 to 4 ml of pre-pasteurized donor milk was collected from the milk bank for antibody detection. So the milk was stored at minus 80 degrees Celsius initially, and at the time of antibody testing, it was centrifuged at 1000 G for 10 minutes to separate the fat layer of the milk, and the rest of the supernatant was transferred to a new tube for antibody detection. So for Ig and IgG detection, we used commercially available ELISA kits. Talking about the IgA antibody, we used anti-SARS-CoV-2 IgA by NovaLISA SARS-CoV-2 ELISA kit from the Novatech, and for IgG, anti-SARS-CoV-2 IgA by QLISA COVID coronavirus ELISA antibody test kit was used. Now talking about the results of the test. So COVID vaccination status of the women who donated their breast milk during the study period. So we have taken total 151 women, out of which 76 were vaccinated at the time of collection, and out of this 76, 44 received one dose of vaccine, and the rest of the 32 received both the doses of vaccine. Now, the association of COVID vaccination status with the Ig and IgG positivity amongst the human milk donors. So out of total 151, Ig antibody was positive in 40 samples and IgG was positive in 30 samples. Out of this 40, 22 samples were received from the vaccinated donors, and talking in terms of IgG, 26 samples were received from the out of this 30, 26 were received from the vaccinated donors. So statistical analysis showed a significant association between vaccination status and the presence of IgG antibody. Now, the association of COVID vaccination status with the IgA antibody among the single dose group and the double dose group. So we have taken total 76 donors, vaccinated donors, and 44 were with the single dose vaccination and 32 were with the 2 doses of vaccination. So we compared these two groups and found that 63.6% of the milk was positive for IgA antibody in comparison to 36.4% of the samples from the donors who received only one dose of vaccine. Similarly, for IgG antibody, we compared these two groups and found that 65.4% of the samples positive for IgG antibody in comparison to 34.6% of the sample from the donors who received only one dose of vaccine. So this data was statistically significant for this finding that the double dose group showed maximum antibody response in comparison to the single dose group. So now coming to conclusion, study provides valuable insight into the presence of anti-SARS-CoV-2 IgA and IgG antibody in the breast milk and the impact of COVID-19 vaccination on their levels. The result demonstrated that completing the recommended double dose vaccination regime leads to higher level of antibody in the breast milk compared to receiving a single dose. It also suggested that the vaccination schedule plays a critical role in enhancing the antibody response and potentially providing greater protection against the SARS-CoV-2 infection in the breastfeeding infants. Findings supported the importance of promoting and ensuring full vaccination coverage among the lactating individuals to maximize passive immunity transfer and safeguard the health of both mother and the child. Now, in the last, to our knowledge, this study was the first study on the SARS-CoV-2 antibodies in the breast milk from India. Donors who received 2 doses of vaccine demonstrated a high level of antibodies in the milk, but still very few literature is present on this topic across the globe. Further research is warranted to explore the long-term effect and durability of antibody transfer through breast milk. Thank you. Yes, sir. Yes, sir. Yes, sir. Sir, we have excluded those.
How can you exclude them?
Sir, recent few months, sir, we have excluded those. In the recent,
You haven't mentioned the time frame on that.
okay, sir.
Yeah, have you correlated with the serum levels of antibodies also?
Sir, unfortunately, we didn't go for serum testing. It would have been give good results.
I thought that would have given you. And you, in your initial one, you didn't say anything about unvaccinated people. But the slides, it comes up.
No, sir. Sir, we have taken total 151 women, and out of those, 76 were vaccinated, and the rest of 75 were not vaccinated.
Okay, right.
Are you aware that there are different types of vaccines which were used against COVID?
Sir, in this particular study, the women were vaccinated with the Covishield and the Covaxin. Out of 76, 70 were vaccinated with the Covishield, and rest of the 6 were vaccinated with the Covaxin.
But your data does not represent which vaccine has been relevant to the study that your results are
yes.
showing.
Yes, sir.
And another thing is, you must have got the recommendations from Government of India that there was a third dose also prescribed and was given as a result of the second wave, third wave, likewise.
Okay, sir.
So, does your sample, it doesn't include third dose at all, isn't it?
Sir, we have just evaluated it for 2 doses.
Would it have been more relevant to include third dose group also?
Yes, sir.
Okay. Okay.
What are the clinical implications of your work?
Sir, we can promote the breastfeeding during COVID infections, that it could provide the antibodies passively to the infants, and it would be beneficial for the infants and in the passive immunity it could provide, sir.
Okay, thank you.
Thank you. Can we have the next contestant, KB4 paper? The IT team, please put up the KB4 slide PPT.
Good morning, respected judges and delegates. I am Senjuti Sengupta, and I am here to present on discrepancy between phenotypic and genotypic linezolid resistance in Mycobacterium tuberculosis isolates. Linezolid, a member of oxazolidenone family, was approved in 2016 by WHO for multi-drug resistant treatment, and it improves the multi-drug resistant TB treatment success by 3-folds. Automated phenotypic culture-based drug susceptibility testing is currently the gold standard for detection of drug resistance, but it has delay in results. The targeted whole genome sequencing is currently the new tool for detection of drug resistance. The aims of my study are to determine the frequency of linezolid resistance in Mycobacterium tuberculosis isolates from cases of drug-resistant tuberculosis patients attending the National Tuberculosis Elimination Program in Uttar Pradesh, and to decipher the agreement between phenotypic and genotypic linezolid resistance in Mycobacterium tuberculosis isolates. The material and methods have been described under the following headings. This is a type of observational study which has been done under National Tuberculosis Elimination Program in an IRL accredited tuberculosis laboratory and it after being approved by the Institutional Ethics Committee. The study was done on 527 isolates after making the following assumptions and after following this particular formula. Sputum samples or TB isolates from the drug-resistant tuberculosis patients with pulmonary tuberculosis who had never received linezolid and consented to participate in our study were included, and those isolates that have a low bacterial load and the sputum culture were contaminated on midget 960 were excluded from our study. The sputum samples or TB isolates from the drug-resistant TB patients were consecutively enrolled from January to June 2021 till the desired sample size was achieved, and were then further subjected to phenotypic drug susceptibility testing with second-line anti-tubercular drugs. The isolates which were sensitive to linezolid were then 20 of them randomly selected and subjected to genotypic nanopore sequencing technique, and the isolates which were resistant to linezolid were subjected to phenotypic recessirin microtiter assay for linezolid and genotypic nanopore sequencing technique. The recessirin microtiter assay works on the principle of enzymatic reduction of the dye by viable cells and is used to assess the proliferation of the cells and the metabolic activity of the bacterial cells. This is the microtiter plate of recessirin microtiter assay, in which the linezolid has been serially diluted from concentration of 4 microgram per milliliter to 0.03 microgram per milliliter. The change in color from blue to pink indicates the growth of Mycobacterium tuberculosis, and the microtiter well in which this change is prevented is considered the minimum inhibitory concentration. In nanopore sequencing, the DNA is extracted of Mycobacterium tuberculosis using the CTAP protocol, and then targeted PCR was performed on RRL and RPLC genes. Then the nucleic acid in our sample was quantified and signature was provided to each of the samples by means of barcoding. These are the forward and the reverse primers of RRL and RPLC genes. The quantification of nucleic acid was done again in the sample, and the sample was loaded in the flow cell in this particular position. The sequences obtained were analyzed using the TB profiler pipeline, which allows the analysis of whole genome sequencing data of Mycobacterium tuberculosis to predict the drug resistance. The results and discussion have been described under the following headings. 527 Mycobacterium tuberculosis isolates were taken, and 1 of them were monoresistant to rifamycin and 90 were monoresistant to isoniazid. All of these were sensitive to linezolid. 154 were multi-drug resistant and 252 were pre-XDR. Out of the multi-drug resistant isolates, 6 were linezolid resistant, and out of the pre-XDR, 14 were linezolid resistance. The frequency of primary linezolid resistance in our study was 3.7%. In a study done in Germany, the frequency of linezolid resistance was 1.9%, and in another study done in Mumbai, it was about 3.1%. There is a considerable geographic variation in linezolid resistance ranging from 22.2% in Spain and 0.2% in United States. The linezolid MIC range is 0.03 to 4 microgram per milliliter. 6 of our isolates showed MIC of 4 microgram per milliliter, 4 showed 2 microgram per milliliter, and 10 of the isolates showed 1 microgram per milliliter. The linezolid epidemiological cutoff in our study was 1 microgram per milliliter. In our study of the linezolid resistant isolates, MIC 50 was 1 microgram per milliliter, 90 was 4 microgram per milliliter, and geometric mean MIC was 2.82 microgram per milliliter. In another study done in India by Singh et al. In 2022, MIC 50 was 0.5, 90 was 1, and in yet another study done in China, MIC 50 was 0.25 and 90 was 1. Lesser number of isolates have been taken in our study compared to other studies, and this mainly limited our study. Of all the 20 isolates, only one isolate had the signature mutation in RPLC gene, that is substitution of cysteine by arginine at the 154th position. None of the randomly selected sensitive isolates had any mutation in the RPLC gene, and the sensitivity of 49.18% for the prediction of genotypic resistance, which was very low. There were no false positive predictions, and specificity was 100%. Positive predictive value was 100%, which was high, and there were no false positives. In a similar study done in Mumbai, there was mutation in RPLC gene, C154R mutation in 50% isolates, and in RRL gene, G2814T, it was in 9.375 isolates. In another study done in China, there was mutation in RPLC, RRL, and RPLD gene in 36.84, 10.5, and 5.26% isolates. Lesser number of isolates have been taken and lesser number of targets, that is only RRL and RPLC targets have been tested for gene mutation, and these two mainly limited our study.
30 seconds for you.
Among the 20 linezolid resistant isolates, 7 expired and 13 were cured. A moderate baseline resistance to linezolid, 3.7% were detected. There is a discrepancy between phenotypic drug susceptibility testing and genotypic nanopost sequencing testing. More number of targets need to be tested. Thank you.
Open for questioning by judges.
Yes, sir.
Undertake the similar work in future, what changes you would like to do in the design of the study?
So first of all, I would like to take more number of isolates in my study. And second thing, I would like to include more number of targets. Like in our study, we had included only 2 targets, but there are many more targets like RPLD, and there are many literature showing more number of targets for linezolid resistance. So we could have included these also in our study.
What do you think are your recommendations for the National Tuberculosis Elimination Program from the scope of your study?
So from my scope of from the scope of my study, sir, until and unless nanopore sequencing technique, there are few limitations, like it has targeted sequencing. So my recommendation would be until and unless it has been it has been confirmed that we can get more like whole genome sequencing can be included in.
Is there any room for combination therapy in the scope of your further studies?
Yes, sir.
What combination you would recommend?
Yes, sir.
What combination you would recommend?
So, it depends upon the LPA, so line probe assay results. So, if we will first of all do the CB-NAT and see whether rifampicin is resistant or sensitive. If rifampicin is sensitive, then we will give our drug sensitive rifampicin sensitive regimen. And if it is resistant, then we'll further do our line probe assays and see whether the first line and the second line drugs are sensitive or resistant. If we see that along with rifampicin, isoniazid is also resistant, that means multi-drug resistant. In that scenario, we will be giving the multi-drug resistant TB regime. If further we see that only isoniazid is resistant, then we will give H-mono or poly regime. If we see that along with the rifampicin and that is along with the MDR, if fluoroquinolone is resistant, then we will give the short oral regime. And along with the fluoroquinolone, if we see any of the class A drugs, that is bedaquiline, linezolid, or fluoroquinolones, any of them are along with that resistant, then we will give the long oral regime.
Thank you.
Thank you, sir.
Hold on. There are two more of us waiting for you. What's the mechanism of resistance in linezolid?
So, in linezolid, the linezolid basically attaches to the 50S and the 30S interface and prevents the formation of 70S complex, and due to which the protein synthesis will be inhibited and it will then kill the bacteria.
Okay. And where does this particular gene that you've done target? Which particular aspect?
So, RRL and genes.
Yeah. Okay, fair enough. Yeah. But if the gene is not predicting the phenotypic characteristic, what are the other mechanisms that are operating?
So, if the gene is not if the gene is not predicted, there can be more mechanisms, like there can be this extrachromosomal efflux pump mechanism in which the drug has been effluxed outside the chromosome, and it can also be that these genes are located in such locations that our nanopore sequencing is not able to detect it. And so there can also be technical defaults in our nanopore sequencing method, due to which it is not being detected.
And when you find linezolid resistance, what are the alternate drug that is used?
So, linezolid resistance.
Yeah.
So, if we find that linezolid is basically included in a long oral regime,
Right.
that is for 18 to 20 months, and it is included in the inclusion phase as well as continuation phase.
What are the other components in that regime?
So, in intensive phase, we have bedaquiline for 6 months. We have linezolid, we have levofloxacin, and we have ethionamide, clofazimine, and No, sorry, sir. I would like to repeat. There is three drugs from the class A and 2 drugs from class B. That is linezolid, levofloxacin, and bedaquiline, and from class B, that is clofazimine and cycloserine in intensive phase. In the continuation phase, we have linezolid, clofazimine, and cycloserine.
Fair enough.
Just 2 points I want to make. One, you should not reveal your name when you are presenting, as far as this is concerned. And you overshot the time that was allotted to you. Thank you.
Thank you, sir.
KB6, please. Do not reject. If the student wants to remove her name, she can remove it.
Good morning, respected chairpersons, delegates, and one present over here. Today, I'm here to present my paper titled In Vitro Assessment of Novel Topical Antifungal Drugs Against Onychomycosis, a focus on efinaconazole, luliconazole, and tavaborole in the Indian setting. I will cover my topic under following headings. Beginning with, what is onychomycosis? Onychomycosis is the infection of nail apparatus by fungi that include dermatophytes, which constitute 60 to 70% of the total, which leading it to the most common cause of onychomycosis. Secondly, by non-dermatophytes, and thirdly, yeast, which are the least to cause onychomycosis. Coming to the prevalence of onychomycosis, in India, the prevalence ranges from 0.5 to 12%, while globally the prevalence is 5%. Onychomycosis is often considered only a cosmetic issue with minor importance, but especially in immunocompromised and in diabetic patients, it can lead to serious health complications, which include paronychia, onycholysis, and tissue necrosis. The treatment, according to the British Association of Dermatologists guidelines for the management of onychomycosis released in 2014, includes systemic therapy, which include griseofulvin, terbinafine, itraconazole, and fluconazole. While these drugs are highly effective, but they come with serious side effects such as hepatotoxicity and drug-to-drug interaction. The second alternative is topical therapy. Topical therapy includes amorolfin, cyclopirox, teoconazole, and 10% efinaconazole. I would like to bring into notice that in the paper released in 2014 by British Association of Dermatologists, 10% efinaconazole was found to be more effective with clinical mycological cure rates approaching up to 50%. While the side effects of topical therapy are very minimal, such as periangle and nail fold erythema, contact dermatitis, and local burning. Moreover, Nail Society of India in 2023 published its recommendation for treatment of onychomycosis, which included newer drugs like luliconazole 5% nail solution, efinaconazole 10% solution, and tavaborole. Here's the clipping of the paper which showed the same. Coming to the brief mechanism of these drugs, luliconazole and efinaconazole belong to the class of drugs of azoles, which act by inhibiting which belong to the class of drugs of azoles, which act by inhibiting 14-alpha demethylase, which is an important enzyme in the synthesis of ergosterol, which forms an important component of cell wall of the fungi, leading to the death of fungi. While tavabarol acts by inhibiting leucyl transferase RNA synthesis, which is an important enzyme in the protein synthesis, thus inhibiting the protein synthesis. Now, the recent approval of these newer topical agents has opened the doors for the clinicians to use these newer drugs regime. But due to the availability of the limited data on them, their effectivity is still unknown. Therefore, the antifungal activity of these drugs needs to be evaluated, bringing to the aim of our study, which was to measure the in vitro activity of the newer topical antifungals, including efinaconazole, tavabarol, and luliconazole against dermatophyte isolates in the patients of onychomycosis. Our study was an observational study done in the mycology department of the department in the period of 1 year, and the sample size when calculated with the prevalence of 5% in the central India came out to be 73. The methodology included collection of nail clippings from clinically diagnosed cases of onychomycosis. The sample was divided into two parts. The first was used for microscopic examination using 20% potassium hydroxide, and the second part was used for culture on seborrheic dextrose agar and dermatophyte test media. Subculture on PDA for identification by MALDI-TOF MS and antifungal susceptibility testing was done. Antifungal susceptibility testing was performed for efinaconazole, tavabarol, and luliconazole, which was sourced from Cayman, USA, using microbroth dilution method as per CLSI M38A3 guidelines for molds. After the MICs were obtained, epidemiological cutoffs were calculated, which were calculated by using the MICs that encompassed more than equal to 95% of all the MIC values. Coming to the results and discussion, 107 patient samples were taken, out of which 58 were culture positive. Out of them, 30 were identified by both MALDI-TOF MS and slide culture and LCB. Out of the 58 culture positive samples, Trichophyton rubrum came out to be the most common isolate, including 53% of the total, followed by Trichophyton mentagrophyte interdigital complex, 29%, followed by Trichophyton tonsurens, 16%, and lastly, Epidermophyton flocosum, 2%. Coming to the MIC distribution of luliconazole for dermatophytes. The MIC range for all dermatophytes was the lowest for luliconazole, that was 0.004 to 0.008 microgram per ml. Majority of isolates of Trichophyton rubrum, Trichophyton mentagrophyte, Trichophyton tonsurens, and Epidermophyton flocosum showed the lowest MIC tested, that is 0.004 microgram per ml, which suggests that luliconazole has a potent in vitro activity even at very lower concentrations. The epidemiological cutoff came out to be less than equal to 0.004 microgram per ml, which shows that, that for luliconazole, relatively lower concentration of drug is required for its in vitro activity against dermatophytic isolates. For the MIC distribution of efinaconazole, the MIC values for all dermatophytes for efinaconazole ranged from 0.008 to 0.5 microgram per ml, with maximum isolates of Trichophyton rubrum, Trichophyton tonsurens, and Epidermophyton flocosum having an MIC of 0.016. The epidemiological cutoff for efinaconazole is 0.5 microgram per ml. The MIC distribution of tavabarol ranged from 1 to 2 microgram per ml, and the epidemiological cutoff came out to be 2 microgram per ml. Among the two recently approved FDA drugs, efinaconazole and tavabarol, Efinaconazole has lower mean MIC as compared to tavabarole. This suggests that efinaconazole tend to be more effective in vitro than tavabarole. However, merely determination of in vitro activity isn't sufficient for determining the clinical outcome. Various other factors such as fungicidal static activity, keratin affinity, the carrier used are also important. One of the important factors also includes nail penetration. Since nail is a greater in thickness, there is a difficulty in penetration. This issue is addressed by tavabarole, which shows greater nail penetration activities and having concentrations greater than MICs. Higher penetration of tavabarole could be contributed to its lower molecular weight. On the other hand, Tachibana et al. In his study showed superior efficacy of efinaconazole in vivo over tavabarole in a guinea pig onychomycosis model. Superiority of efinaconazole over here could be because of its lower MIC in vitro and also fungicidal activity in the presence of keratin. Now we have few advantages of these newer drugs which I have discussed over the treatment used currently, which include these are the promising new therapies approved by the FDA which have demonstrated decreased keratin binding affinity, greater mycological cure rates, and better pharmacokinetic profiles in the treatment of onychomycosis. These drugs can also circumvent hepatic toxicity and drug interaction observed with oral antifungals. Therefore, there is a reduced need for their monitoring. I have done a SWOT analysis of my study. The strengths of my study include that this is one of the first studies to be conducted in the Indian setting, including the prevalent dermatological condition.
Your time is up, please hurry up.
While we should also see a study in the weakness of, with some weakness which includes smaller sample size and institutional bias, which might lack the external validity. Therefore, to conclude, the findings from the current study regarding the in vitro performance of efinaconazole, tavabarole, and luliconazole indicate that these new drugs show great potential as a prospective candidate for advancing the development of a new antifungal treatment of onychomycosis. Thank you.
Open for questions, please. 30 seconds were given to her extra because her PPT got stuck. But still she has exceeded her time.
While you were getting your patients of the condition that you are studying, what specific aspect you have tried to find out from the clinical history taking and for selection of the cases that you are taking under study?
So we included all the patients of onychomycosis, and while taking specific measures that we didn't include conditions such as psoriasis or other nail conditions which mimic the onychomycosis caused by dermatophytes. So we also excluded the bacterial and other causes except for mycological causes which cause onychomycosis, sir.
Any specific relevance to the occupation of the patient who was subjected with this condition?
Yes, sir. Sir, specifically we took the history if the patient belonged to lower socioeconomic strata, or if the patient is belongs from the labor class or is a farmer, because these patients are more susceptible to fungal infections for their nails.
How do you recommend the application of this?
So these newer drugs are available as topical solutions. Efinaconazole is available as...
Quite true. But the point is, how many times a day does a
Yes,
person
sir.
have?
Sir, efinaconazole 10% solutions is recommended daily for daily application. In case of fingernail onychomycosis, it is for 24 weeks, while for toenail onychomycosis, it is for 48 weeks. Tavabarole 5% solution, sir, is recommended for 48 weeks for both toenail and fingernail onychomycosis.
Yeah, they administered simultaneously or in sequence?
Sir, daily applications, so separately.
Okay, fine.
What is a better study design to have the comparison or the superiority of the established drugs?
Sir, I might have to read about that, sir.
No, it was like, what is the better way of designing the study when you are comparing different drugs or having a newer drug comparison? All right.
Sorry, sir.
Thank
Sir.
Thank you so much. The winners will be announced in the valedictory session. We'll start at 10. We would now begin with the presidential oration. I'll request Dr. Pallab Ray, past president for 2021, as Dr. Kamala couldn't be here due to some other commitments. I request sir to please take the stage as chairperson. I will now request our Honorable President, Dr. Aarti Kapil, to please come on the dais. Dr. Aarti Kapil has done. या वहाँ से alternate लगाओ. ये chair center में करो. क्या लगा दिया ये? आप बैठो. Ma'am, please take this chair. Dr. Aarti Kapil has done her MBBS from Indira Gandhi Medical College, Shimla, and her MD in Medical Microbiology from PGIMR, Chandigarh. She is a Fellow of National Academy of Medical Sciences and life member of many scientific bodies. She is former professor from Department of Microbiology in Delhi, from where she superannuated in 2023, and has worked a lot in the field of antimicrobial resistant, healthcare associated infections, and molecular epidemiology of infectious diseases. She was the editor of Anand Narayan and Panikar Textbook of Microbiology, 9th edition, besides contributing book chapters in many textbooks. She was also awarded the Dr. S.C. Agarwal Oration by IIMM in 2015, Dr. Ajita Mehta Oration by Hospital Infection Society of India in 2019, and Dr. D.S. Agarwal Oration by IIMM Delhi chapter in 2022. She was the associate editor of IJMM in 2012 and subsequently editor-in-chief for 3 years from 2013 to 16. She has been an expert group member in multiple steering and standing committees of national bodies, is an NABL assessor and a Shia International Ambassador for India. She has delivered multiple lectures in fields related to AMR and infection control and is currently working as Professor of Microbiology at North DMC Medical College and Hindu Rao Hospital, New Delhi, and has been the President of Delhi chapter of IIMM during 21 to 23. Now this prestigious IIMM Presidential Award 2023 is being conferred to her for her dedicated services and contributions in the field of medical microbiology in India on behalf of IIMM on 23rd of November at the 46th National Conference of IIMM here at KGMU, Lucknow. I request the past President Dr. Pallab Ray to present her with her medal and citation. I would now request our Honorable President Dr. Aarti Kapil to please deliver her oration address. Ma'am, it's been loaded. The PPT is loaded. What's it called?
Aarti Kapil, Presidential Award. Thank you, Dr. Pallab, respected honorable past presidents in the audience, my seniors, my teachers, and dear friends. It's an honor to deliver presidential oration today, and for that I am thankful to IIMM for having given me this opportunity. Today, the topic that I have decided for my presentation is a mix of conventional and contemporary man, machine, and microbes in a race against AMR. And the microbe that I chose is Salmonella typhi, mainly for the reason that since I started my work as a student of microbiology, I worked in Salmonella typhi and its antimicrobial resistance. And somehow, over the last 30 years, have continued to work in that area. And also, one interesting thing is that when I wrote my thesis, the introduction started from typhoid fever continues to remain a public health problem. And even till now, the various studies which use genomics, which use large data sets from sequencing, also start from the fact that typhoid fever remains a public health problem. So somehow this organism has played with us when at times we thought it is over, it's no longer a problem, it comes back. So that was one reason. And now, how do I move the slides? Spacebar. No, it's not moving. The slide is not moving. So interestingly, the presence of this one. So this, the evidence of typhoid being a cause of death has recently come based upon the sequencing data from one of the very ancient burial pit near Athens, where the graves, mass graves, the DNA sample was available from the mandible of these patients, of these skeletons, I should say, and they found the evidence of Salmonella typhi, and others were negative. And probably that was, now people think it was possibly responsible for all that wartime siege by the Spartans. And then, of course, E. typhosa was the name given around 1940s. Interestingly, what we know about the disease today, the pathogenesis and its natural epidemiology, is something which was available to us in 1970 by very interesting experiments by Dr. Hornick, because this is an agent which has no other animal model. It's human restricted, and only human volunteer experiments at that time, though I'm sure ethical considerations were given, but I must tell my juniors, my students to read. These are very interesting articles, 2 articles published in the same volume of NEJM, and that talks of how this disease occurs. However, even now, based upon a recent systemic review and meta-analysis, we find that the incidence of typhoid fever continues to almost remain the same in India, about 400 to 500 cases per 100,000 cases per year. We recently conducted a study in Ballabgarh rural area and also found, over 2 years, also found that this is probably the incidence in our northern area also, and so it was a prospective study of fever cases. A surveillance of enteric fever of India, which was steered by CMC Vellore, has recently been concluded and published widely. And many of us in the hall could be a part of, maybe we were a part of this study. And it is in a way very representative, because it has sites tier 1, 2, 3 from rural and urban areas, the primary, secondary healthcare, and they also, in this also we can see that the incidence is almost same in our country, and different states, you can see the color, the darker colors have a higher incidence, while the lighter we have the lower incidence, but mainly 2 organisms responsible for typhoid fever in India, S. typhi and S. paratyphi A. They still contribute to around, whether it's a rural setting or an urban setting, hospital setting or the community setting, they contribute about 60 to 70% S. typhi and another 25-30% S. paratyphi A. Very rarely B has been isolated. So they remain by and large the cause of typhoid fever in our country. So although S. paratyphi A numbers are less, percentages are less, but we have to be careful while designing any intervention therapies in terms of vaccine design, and have to keep this in our mind. So this disease, which is a community acquired, which causes systemic infections, which causes bloodstream infection, and it's severe if you talk of community acquired infections. So antibiotics was supposed to have been a great armament, a great intervention and great number of antibiotics in our armamentarium. In 1950, chloramphenicol for the first time published in a case series. This is a set of case series in the California Medicine Journal, published in 1950. And here the highlighted area shows the time to defervescence of fever in these cases. So it was like a magic. After 2 to 3 days of chloramphenicol, the fever started coming down. So that was very good, and it was thought to have solved all problems of typhoid fever till in 1972. Plasmid-mediated chloramphenicol resistance was reported by Dr. Panikkar from India, and also at that time from Mexico, carrying the CSSUT plasmid. And this plasmid was known in Japan for Shigella and probably transferred from there. And then subsequently, different parts of India also had outbreaks due to this plasmid-mediated chloramphenicol resistance. In Chandigarh also, we reported and we studied, and at that time, we didn't have any molecular method, so conjugation experiments used to be done in which you incubate the resistant strain with a known sensitive strain, and then next morning you see the sensitive one has become resistant by MIC. So that was very interesting, and then this time the use was really restricted to amoxicillin at that time, and once that was being used around late 80s and early 90s, we had this so-called MDR outbreaks, which now we call MDR, ACSSUT, amoxicillin, chloramphenicol, and cotrimoxazole, the current phenotype that we describe as MDR. So what happened? Ciprofloxacin came into use very widely. A very good drug, very easy to give, very convenient, compliance very good because it's an oral drug, BD dosage. But what is the current status due to this increased ciprofloxacin use? A lot of clinical failures soon after it came into wider use. And at present, the first one, as you see here, is the, I hope this works, anyway, so the first on the extreme right of your side, left from you on your side is the ciprofloxacin, and the MICs. Again, this is the data summarized from these multi-site studies. And ciprofloxacin now is mostly non-susceptible. Coming to the intermediate, though shift to resistance is still not that high. Azithromycin, still sensitive, but MIC is very near the cutoff of the resistant. Ceftriaxone, enzyme, continue to be good, continue to be susceptible, and MICs are still well below the breakpoints of resistance, but there is an evidence of creeping MIC and pockets of, or small clusters on isolated reports of ceftriaxone resistant have started to come from India also. First time from Maharashtra, we got some strains which showed resistant to ceftriaxone, and one isolate from Chandigarh. At that time when they were sequenced, fortunately they did not carry ESBLs. They were all carrying TEM and SHV type of genes for resistance to ceftriaxone, and it did not spread beyond that small cluster. But then recently from Noida, Delhi NCR region, from Noida Super Specialty Hospital, this Dr. Sumit Rai shared this strain with us. He reported a small cluster in a family, and where we had ceftriaxone resistant isolates. These were carrying CTXM15, which is ESBL, but they were technically non-XDR because they were not MDR in the true sense. They were susceptible to chloramphenicol and azithromycin. But that way, they were different from the Karachi outbreak. And I'm sure you all know about this Karachi outbreak, where, again, it has got stuck, I don't know. It's not moving. The Karachi outbreak, where we know that it was a XDR strain. Yeah, thank you. And only susceptible to azithromycin. And that was the cause of worry because it led to a large outbreak. And azithromycin, as we know, is not an ideal drug for severe or serious cases. One would still like to use some kind of beta-lactam antibiotics for very sick patients. And so in these patients who suffered from infections, serious infections, or needed hospitalization for these strains, were given meropenem. I think this was just to give something other than the cephalosporin experience from the gram-negative sepsis. And so we also wanted to characterize these strains from Noida Super Speciality Hospital and look at how they are related to the Karachi outbreak, because now this spread transcontinental or across the regions is very important in understanding the epidemiology. So we also, these CTXM15 positive salmonella isolates were all monophyletic, but different from the Karachi outbreak strains. They were H58 haplotype, which is the commonest right now in Southeast Asian region. And the gene was clustered on INC1 plasmid, as was the Karachi outbreak. However, they were different, as I said, because they were sensitive to chloramphenicol and azithromycin. So I think these are some individual pockets which are, or events occurring in terms of resistance emergence. And therefore, it makes it important to understand how these are spread, how they are getting transmitted, and understand the geographical distribution. And for that, some of the tools of molecular epidemiology are being used. One of the tools, because we wanted, antibiogram is a good tool. It is helpful. It is one of the parameters which give you an initial idea about the relatedness of strains. However, you need something reproducible, something which can be compared across the countries over the years. So one MLST database for all international isolated salmonella was made. And so we conducted initially the MLST based on the sequencing of the 6 house typing genes. And in this, we find that the sequence, sequence typing was done, and also we tried to do the population structure. This is one way of analysis, E-burst analysis. So you basically have to do your job, have your isolates rightly analyzed, proper DNA isolation, and then the machines do the job. So this is one way of its computer analysis. So in this, you can see in the center, the brown bigger dot in the center is the founder strain Salmonella typhimurium. Then below that, with the red line, red circle, are the Salmonella typhi, which all belong to a single clade. On the extreme right side is the green circle of paratyphi A. So these are various analyses which tell you how the organisms are related, and when they spread, how do they, what should be your policy of treatment guidelines. So this is just a pictorial representation of these sequence types across the different geographical area on the map. And we see ST1 still remains the most common sequence type across, and in India that is the commonest. We found ST2 some, and one rare ST3 was also found in this, and another one was earlier reported from other Southeast Asian regions. If you see as the paratyphi A sequence types, it is also most common ST85 across the world, followed by ST129. So this just gave you the idea that yes, there are a similar type of sequence type, more or less conserved organism, but we had a much better potential to do these studies once the complete genome was sequenced. CT18 strain was the first one to be sequenced. It changed the whole way researchers looked at Salmonella typhi. and it was published in 2001 from the MDR isolate from a patient from Vietnam. Soon the second genome followed, DY2, because that time it was a requirement, it's a vaccine strain that we know. And so with this, Salmonella, overall typhoidal and non-typhoidal, became one of the first pathogens which quickly transitioned to a routine sequences by public health laboratories wherever there were resources and capabilities. So this kind of data is available and we can play with it, we can compare our strains with it, we can analyze the various aspects of this organism where we do not have animal models. So probably by study of their genes, by virulence, we can go ahead. So with this data, as I said, with this sequencing, one was able to study more clearly in a better way how the transfers, the intercontinental and international transfers within the geographical areas have taken place in Salmonella typhi, mainly looking at the XDRs. So if you look at the picture on your right, the yellow line, just to make it simpler because the print is so thin, so the yellow line shows the transmission of the XDR phenotype, the Karachi strain, how it has also gone to the United States, which otherwise doesn't have typhoid fever as a problem, and even in many European countries, the Western Europe. And so there, if the patient comes culture positive or the patient is suspected of typhoid fever, they have to keep the travel history in mind. So therefore, this kind of information becomes important in understanding the epidemiology and having your intervention strategies in place. Using this same sequence data very recently, almost 13,000, I think the largest collection of sequences have been analyzed and published as a part of the Global Typhoid Genomics Consortium group. Many of us are a part of it, and Indian strains are fairly well represented. And here, again, although it reiterates the findings that we have, but it gives you an idea that how you can analyze this data individual country-wise across the countries. For example, something which we might be interested in, the green arrow shows the India figure, and then these are the countries, UK, USA, Chile, and like this. With India, you have Bangladesh, Nepal, Pakistan. So you can just have a quick look at what are the different MDR phenotypes which you see in these countries. And so although antibiogram and manual, it is possible to do this, but very laborious. Here you do just sequencing and you can analyze it in different ways. And quickly, if you can have a look, see in India, the graph of India shows a very high number in yellow of ciprofloxacin non-susceptible. But the blue line, ceftriaxone resistance is hardly there. If you see Pakistan, there the ceftriaxone resistance is very high. MDR is increasing, which we don't see in India. Nepal is more or less same to India. And similarly, you can quickly analyze across the countries. For example, if you see Malawi, there is an increase in MDR, which we don't have. So some of these analysis, so this is an extensive analysis, a big paper, and we can have many ways by which we can raise a question and analyze it based upon what we want to know. So that is the potential of the sequence data which you have available with you. But coming back with all this, now we are still dealing with a disease where patients fall sick. Apparently we have antibiotics, but patients do not respond. So we wanted to do a systematic study in our pediatrics department, considering the fact that was a department where very judicious antibiotic practice is being done. So even there, we found that the mean duration of fever of the patients after admission they were admitted already 9 days fever. Admission another 6 days with a careful and everyday follow-up and antibiotic therapy being given, and with all laboratory and culture facilities available. Even then, almost 20% of the inpatients needed 2 antibiotics, 2 anti-typhoidal antibiotics, with rational use also. That was of course, the basis was clinical decision and worsening of the clinical condition. And about 7 to 8% of OPD patients also in the subsequent visits. So no fixed dose anti-typhoidal therapy, but sequential after clinical evaluation. So this is the kind of problem that we face at present. And so another aspect of severity that was looked at by this surveillance study was the ileal perforation in tier 3 cities, institutions where patients are admitted with ileal perforation, non-traumatic. So there were definitions of what would be the enteric fever perforation and what are the other. But these are all non-traumatic ileal perforation. And in that also, combining confirmed, probable, and possible, about 50% of non-traumatic ileal perforations were due to salmonella or typhoid fever complications. So this is a tip of an iceberg. So this is still occurring, although its analysis is not done very regularly because it's difficult to really say it was a typhoid perforation unless it occurs immediately during the acute infections. So overall, due to AMR, due to increased cost of treatment, due to the complication occurring and prolonged stay in the hospital, the cost of illness due to typhoid fever in India is very high in this proportion, although it may not be a very large proportion, many of them getting treated in the community itself. But then, considering a disease of lower socioeconomic status, this cost, which can vary in a ileal perforation or complication, 10,000 to up to 30,000, depending upon the condition. And sadly, about 70% of the expenditure is incurred out of the pocket of these patients, families. So now, how do we move further with all the facilities or technology that we have? How best we can use, at least to ask our research questions, do some future planning to have something which can be an alternative therapeutic modality. So quickly, some of them, you are all familiar, in silico drug designs and structure-based drug designing is something which is used, and we tried to develop the model with the help of our biophysics department. The tertiary structure, here you look at the protein interactions of the molecules and the drug targets, and a drug with the target. And so this is, for example, constructed the DNA gyrase model, and the green is the gyrase A, the yellow there is gyrase B, and then the DNA skeleton you can see behind, helical, and the blue one is where the ciprofloxacin acts, because ciprofloxacin and DNA gyrase still worth working on. And we wanted to also study that how this mutation, which is not leading to MICs on the resistant range, but still causing clinical failure. So you can see the first figure where you have an arrow is the wild type. You see a very good interaction, binding is very good, and it is a best possible fit. And that is why it becomes effective. But even single mutations, which you see in the subsequent B, C, D, and more than one mutation in E, moving away the ciprofloxacin from the binding sites on gyre A. And this is how the structure, computer-generated models and the docking experiment. So what you have to do is you have to give information, and the computers will do modeling for you, but it is you who will decide what you want. The second thing you can do with these models, in silico designs, is look at the possible alternative targets. So if GAIR A is not working, we should also try and look, we tried to look for GAIR B as a possible target for any drug, because these are conserved proteins and they are likely to be lethal to bacteria if something acts on them. So ATP on GAIR B unit also, so a lot of chemicals, molecules were studied, their interactions were studied, some novel quinazoline derivatives were found, they were then chemically synthesized. So a lot of work and with our collaborators was done. The MICs were carried out after synthesis, and there were some promise. They did show MICs in 8 to 32 ranges, but I think a lot of more questions have to be answered in terms of their safety and toxicity and actual use in clinics, but at least a way ahead. Another thing you can do with this structure based also, only on computers, is to have a drug repurposing. So again, in this, one of the groups that we are working with, we found that FTSZ1 cytokine protein, which is involved in cell division, again, if you inhibit that, it will kill the bacteria. So again, by structure-based drug designing, 6 drugs were found, 6 drugs inhibiting this protein, and they were still working on which one now will go further in terms of its better MICs and possibly pharmacokinetic studies. Then comes the pan-genome. Now, this is another, see, after all, whole genome sequences give you the in-depth look on the total gene of the, insight, I should say, on the total gene of the organism. So if you take a population of Salmonella typhi and construct and look at its population, you construct a pan-genome. And just to make it easy for you, because these are a lot of computer-generated analysis, these are functional databases of various genes, COG and KEG available. So here, if you just look at what I've highlighted in the with 2 arrows, the yellow areas, they show the unique genes in Salmonella typhi, which are not there in non-typhoidal Salmonella. So means generally, if I can generalize, these could be a potential target for further studies and in-depth analysis of what role do they play, and we can have a look at them as a heat map, this is called, which gives you an easy visualization. So the upper area includes the, again, this is the same database of KEG, where I had shown you the pan-genome analysis. There's another way of looking at it, because you can understand very easily that Salmonella typhi are the upper isolates and below are all non-typhoidal. And the green, the fluorescent set of genes being there, which we do not find in non-typhoidal Salmonella. So probably involved in invasiveness, while you don't find some of them in typhi which are present in non-typhoidal could be responsible for their human restriction. They do not cause disease in other animals. So these are some of the ways we can use these computer-generated models, and we can use this technology in developing vaccine candidates, the diagnostic assays, the biggest limitation that we have right now, and new antibiotic molecules. And as we say, clinical microbiology also move into Amrit Kal, we should be able to use the technology, we should be able to use the machine learning to the best of our use, because we don't have to worry, I think, because it is our potential, the human brain, which will still have the power and the ability to produce such machines, assess their ability, and ask questions because of their creativity. Thank you. And I must acknowledge all my teachers, PhD students, research team, financial support, the collaborators, and the Department of Microbiology at AIIMS New Delhi, where I have carried all this work and got the platform to carry it. Thank you.
Thank you very much, Dr. Aarti, for an excellent presentation on typhoid fever. I remember Aarti's days 40 years back when we were colleagues and contemporaries, and the two of us, we happened to be the last two thesis students of the legendary late Professor K.C. Agarwal.
Yes.
And we, both of us, we share our interest in enteric fever, and we have been, in the last decade or so, we have been part of a consortium, national consortium on typhoid fever work, and many of the many of the papers you have cited, both of us were associated with it. And your subject was, I must say, a very interesting subject on typhoid fever, which is a unique organism and a unique infection, an organism which is highly adapted to human being, still producing a very severe, often life-threatening infection. And I'll say out of nearly more than 2,000 Salmonella species which target the GI tract as the target organ, this is an organism, being a Salmonella, targets the reticuloendothelial system.
Yes.
And this is one organism which gives us a live demonstration of the fact that increase the load of antibiotics, increase resistance, stop it, you get rid of the resistance. In early 90s, we had 100% resistance to the primary 3 drugs, and today it's much less than 3% because we have completely stopped using those drugs. Thank you once again for an excellent presentation on typhoid fever and your tryst with Salmonella typhi. It's customary not to have any question, answer, or discussion on the topic in a presidential oration. So with that, I thank the organizers and hand over the mic to Dr. Sonal Saxena, the secretary.
Thank you. Thank you for your kind words and patient hearing. Thank you.
Thank you, ma'am. Thank you, sir. I would now request Dr. Aarti, President IMM, to chair the next 2 awardee sessions. Dr. S.C. Agarwal Oration Award has been awarded to Dr. Pragya D. Yadav. She is Scientist F and in charge of Biosafety Level 4 Laboratory at the Indian Council of Medical Research, National Institute of Virology, Pune. She is known for research contributions in the field of biosafety, emerging and re-emerging highly infectious viral pathogens. She has handled multiple research projects, and her exemplary work during COVID-19 pandemic with successful isolation of SARS-CoV-2 from clinical specimens, which was utilized by the Bharat Biotech to generate the first indigenous Covaxin in India. Her area of interests are multiple viruses, molecular biology, public health, research and development, biocontainment science, translational research, and preclinical vaccine studies in animal models. She has published more than 300 papers in national and international journals, including Lancet and Nature. I request Dr. Pragya Yadav to please deliver the oration.
So thank you and good morning, everybody. So it's actually a great privilege to present something in front of so many president of IM today with the current and past, and I know everybody is a stalwart in the field, and I dedicate this oration to Dr. S.C. Agarwal, who was a very famous, renowned scientist and founder member of IM as well. So with this, I present some of the work which we did, but when I present this work, it's not just my work. It's a work of a team, it's a work of an institute, and I'm fortunate enough to be there to guide them and to be part of that particular work. So with this, so as we talk about the pandemic, and after the COVID pandemic, there is a lot of awareness which created among not only the scientific people, but also among the common man, that what is a pandemic, what is diseases, and a lot of curiosity about the vaccine, even about the kits, and there was a lot of media hype which we see during the pandemic, and that had created a lot of understanding and questions not in the students, even the children of 5 to 10 years now know the name of the diseases and viruses. So if we look at the history of pandemic, there were many diseases which caused diseases, and some of them were present for decades, and nobody could make the vaccine or drugs to treat them. And if we, some of you remember the example of that smallpox, which stayed for a very long time. There were many viral diseases even stayed for decades and killed many of the people across the world. Before this pandemic, in 2009, many of the virologists know that there was a disease called H1N1, which is an influenza virus, and it was created havoc across the world and also killed a large number of people. Still, influenza virus is in circulation and continuously a threat for causing many diseases that include avian influenza and other diseases as well. If we talk about smallpox, the only one of the disease which caused a large number, 33% of death in the infected people, and it took decades. So it started in 4th century, and we could eradicate this disease in 1978 after long efforts from many countries, and India could eradicate this disease in 78 after post-freedom and a lot of efforts from WHO and government to become a smallpox-free nation. But still many other pox diseases exist, and recently we saw the emergence of monkeypox, which was present only in the 8th country and called as endemic diseases. It's not only present now, it's circulated in more than 102 nations during the post-pandemic phase, which we call so far. So in this process, what matter for a country like India to have these facilities where we can handle these high-risk group pathogens? So there are, for just quickly summarizing, there are many category of viral pathogen we call as biosafety level 1, 2, 3, 4, but there are certain high-risk pathogen. For them, we do not have any treatment, and we do not have any way to treat, but they are so infectious that if you get infected, everybody will die, and those people who come in the contact, they also have possibility to die. So to do the research or to prepare any kind of research, you need certain facility, and this called high risk is called biocontainment facility. And we had made the BSL-3 first in India in NIV in 2005, and it was a time when there was no regulatory standard in India existing. So how you go validation of a facility, how you do the training was a lot of different challenges we see after 20 years. Then we made the BSL-4 facility, which is become functional in 2013. And Raghunath sir is here. He was chairman of the committee, and he knows how we proceed slowly. When BSL-4 is established, people ask what you will do there, where we have any BSL-4 pathogen. And initially when the facility BSL-3 was made, there was a Casanur forest disease, which considered to be BSL-3-4 pathogen. But I assure you, from the time this BSL-4 is built, and now we have not rested a single day, and we are not working on a single pathogen, because you have seen that how infectious diseases are emerging and challenging humankind every single day. So you are always constantly busy. There another tool we developed during the pandemic, a mobile BSL-3, which was deployed recently in Kerala outbreak. And you can go to a site and you can work in a BSL-3 containment zone, testing or any kind of research. You can grow the virus, you can genomic sequencing on site. So these are the some of containment facility which was created. But when I say the biocontainment, it has a lot of work behind, and making any BSL-3 or 4 require a team which is very dedicated. There is no 9 or 8 hours duty. You have to 4 hours active and respond to all kind of emergency, like you are fighting on a border for the country. If we talk about the recent priority diseases which was defined by WHO, and there are many, and out of these diseases, at least 4 are present in our country. For COVID is one of the high priority disease, CCHF, Nipah, Zika, and Ebola. there were many other diseases which is not concerned to India, but preparedness is always demanded, and we have to keep alert. We have to prepare that. So if we look at the history of last year, which has started with the 2001 when the first coronavirus outbreak happened, killed many people, affected more than 8 countries. So we started learning from that time that India need a preparedness for viral diseases. And in this process, there were many times we had a threat of yellow fever, we have threat of Zika fever, and also Marburg fever. And every time when there was a demand, we prepared the VRDL's laboratory, which initiated by the Department of Health Research in 2009 with the bigger efforts, and it is now strengthened more, and we have more than 150 laboratory across the country. And there was some chosen laboratory, and some of the heads of those laboratory like Dr. Bharati Malhotra, Dr. Amita Jain, Dr. Jayanti Sastry, and many others are sitting here. They have continuously worked to enhance the surveillance in our country, detection in our country, and which help to respond and reduce the panic whenever there was any fear or emergence of new disease. And in this process, there was a demand of creating protocols, training program, not only on the biosafety, but how you work in the so there is one component to work in the laboratory, but there is another component where the clinicians are dealing with the patient. So we initiated a project in 2015 with the CDC, GHSA, where we did many on-site and off-site training, which included a large number of the people training and making them understand the importance of biosafety and biosecurity in context of emerging infectious diseases. If I just summarize some of the recent findings, how we utilize this research as a tool to understand about the viral diseases. There is one disease, forest disease, and we worked for 30 years, and this work was closed because there was no biocontainment facility. It was reinitiated, and the disease was supposed to be believed only present in Karnataka. We can see it is spread or present all over the Western Ghat. But the good thing that we studied many aspects of the disease, and the latest advancement is that we have now 11 KFD centers across the Western Ghat. They can do timely testing and report the cases to government. We also did a study of the complete genome, and the past vaccine which was made long back is not effective anymore, so new vaccine is in a process to make. The another disease which I would like to mention, nowadays you are seeing the news because Zika is reported in many states recently. But Zika was present even before pandemic. When the first case was confirmed in Ahmedabad, we found the strain which was present in Ahmedabad was totally different. The strain which is spread during 2015 to 17 in Brazil and other country, and there was a serological evidence present prior to that in country for the Zika virus. During the pandemic, we could see that there was an outbreak not only in Rajasthan, MP, but other state also. So we did a survey using our VRDL network, and we found that there was at least 11 states where the cases of fever with rash were presented, found not only positive, but they were confirmed by sequencing. But we can see that during the pandemic time, as soon as we started becoming normal, the dengue, chikungunya, these infections has also increased because it is a mosquito-borne disease. So if the mosquito will be there, these diseases will keep spreading. There were many co-infection cases were also reported from many areas. The next disease, which has been discussed recently in the media, Nipah outbreak, because in 2023, there was an outbreak in September, but prior to that, the first outbreak happened in 2001 in Siliguri, West Bengal, and some of the senior members remember that this was a time that nobody even knew how which was the disease, and there was nobody who knew that it is a Nipah virus, and it took the country to 5 years to confirm this was a Nipah virus infection with the help of CDC. And that was a time we didn't have any containment facility, we didn't have any laboratory to understand, no molecular facility. And as soon as these facility came in the picture, the second outbreak, which happened in 2007 in Nadia district, we could quickly identify sequence, but all of the patients died even if before they reached to the confirmation. In 2018, there was a huge number of cases, 23 cases, and 18 of them died, and that created a lot of panic. But that panic has really pushed the understanding of training to the healthcare worker, and that helped Kerala to respond in 2019, 21, and 23. That not only reduced the death, but also reduced a lot of other factors which affect the economy of any state in the outbreak. Those training also helped Kerala during the COVID pandemic for the containment and also a different research study we did. So to understand, initially there was no clue that how this disease is spreading in India. Prior to India, the Bangladesh and Malaysia reported these cases, but in Malaysia, the cycle exists between pig and bats, and there is no human-to-human transmission. So finally, after long studies of 8 years, we could correlate that bats are the responsible reservoirs in India, and the transmission happened. In 2018, and after that, there was many studies and efforts which was done, including developing indigenous assays for Nipah virus, and we don't have to take anybody else's support. And that helped in initial, not only the testing the bat sample, but also testing the human sample whenever there is an outbreak. With the recent data of many years of the bat survey, we found that there are a large number of states who presented bat positivity for Nipah virus, and that include not only IgG antibody, but also the viral RNA positivity. It means there is a risk, and we should able to deal with, and the clinician, when the cases are not only coming with the AS, but also coming with ERDS and LI, they should consider testing for Nipah virus and following those kind of preventive measures to not get infected. The next disease, CCHF virus, which was detected first time in 2011 in a BHF case, and that again a cluster of the cases happened in Ahmedabad, including 80% of death in the cases. But after that, once the cases were confirmed, not only Gujarat, but also Rajasthan presented many outbreak and constant outbreak investigations, serological studies, study in cattle and sheep goat could reveal that there is a lot of risk having in this country for tick-borne disease. And one of the survey which we did in animals showed that a larger number of the state showed IgG positivity in these animals. And we also did a tick survey, which is not a very good tool for such disease, which is a zoonotic disease. And we could see that Rajasthan and also Gujarat have an active circulation of disease in these ticks. We could develop different diagnostic assay for all these diseases and have the capacity to do any research in the current scenario. So before the pandemic, these efforts were continuously made in the country, but I don't have to tell about the COVID because everybody knows the kind of impact in me. But when we talk and go back and think about that how we perform in the pandemic, what we learn in the pandemic, and how we can improve in the future any pandemic or outbreak response. So there are many lessons which we have learned. And what was the main driving force, how we did some of the study and we could derive. So we all know that how quickly China came up with the sequence when they declared the cases in the first week of January, and India was preparing itself with the help of VRDL for testing the initial cases. And as usual, we detected the initial cases from Kerala, the first 3 cases, in the last week of January. And being a virology institute, whenever we do any new disease detection, we always work on classical virology, do the virus isolation, look at the pathogenicity of the virus, prepare the reagents for testing, and these were the some initial phases. But there were more challenges due to pandemic, that evacuation of the people from Iran, Italy, and many other countries. And there was a huge number of team which was working in all operation with the government of India initiative, including army and other factors. But NIV was dealing with all those clinical research or the sample testing coming from these areas. So we talk about different variant of concern. In the beginning of the pandemic, nobody was knowing the virus was going to change so fast. Or there will be more challenges if we have a vaccine of the variant concerns. And if we talk about the different, and there was a new terms established in pandemic, the first wave, second wave, third wave, and these are different waves which make everybody's life actually shattered. So if we talk about these cases and how it impacted our thinking on traditional science, that's again a challenge of this virus. Whatever we thought in the beginning, it shattered, and it challenged to think novelly that how you can do the research. how you can prove that your vaccine is good or your thought is good on the treatment protocols. So these are the new challenges which I think Dr. Amita Jain chose this theme of program is like every microbes matters, wonderful theme. And that's what the COVID was all about, that it made that every variant is important and force everybody to do a genomic surveillance across the globe to understand how the virus was mutating. During the pandemic, with this, the first efforts was NIV to establish VRDLs and other testing laboratory, but out of that, there was other challenges. So if I remind you, in the beginning when the China reported that they isolated virus in the fibrosis cell, our lung embryo cells, you believe as a scientist and you try to do that. So we got this costly cell lines and we started doing isolation and nothing grow, no virus grow in those cell lines. You try to look at any study which was said animal model, and traditional model is mouse, but mouse was not a model for coronavirus. So initially we struggled for 2 months to understand this is published, how we replicate it, or how we use that published data, and we realized that we have to just forget it and left, you read the science, but you use your classical virology science to prove your own hypothesis, and we started from scratch. We started doing every experiment without believing that what is being published, and that's how we could go ahead and we could do the research. So some of the findings, for example, there was a need for serological kit. Indian government got a large number of kits from China and they failed here in Indian market because of the quality performance. So then we were given a challenge to make a kit in 1 month, which when, routinely we make a kit in 6 months, 8 months, with the luxury of time and taking everybody's consent. But that was a very, very harsh time. Everything was given on a very, very close deadline to prove and to finish it. And that with this, as you are solving one problem, there are multiple problems are coming. For example, there were mucormycosis cases and people started talking that mucormycosis is happening because of the variant of concern. Design a study for that. You have reinfection, breakthrough. You have, then you have booster doses and then further reinfection and breakthrough. So there was another challenge, and each time virus challenges, we are forced to do design a new study to prove what is happening to Indian population. Then we also work on different variant of concern and how it impacted the immunization. So this was the first kit which we created very quickly. The validation was done, and it was not only utilized for the serology, the fourth serology study which was done by NIV with the multiple center across the country, but also we use this kit for constantly in different research study and which help to understand about the virus. If we talk about the vaccine, there were many platforms prior to this pandemic which was used for vaccination. And as we were thinking about the same time, Serum Institute tied with Oxford, and they started working on Chedox vaccine. But government of India wanted to have an indigenous vaccine, and they wanted to have in a very, very short deadline time. And there was a challenge how we do it. So when we talk about the vaccine, so there were more than 300 candidate vaccine which was started during the pandemic, and only 11 were approved by WHO to be used for emergency. So you can imagine that having one of those vaccine from India is a really great matter of pride for all of us, that we may went to those kind of a standard which international body has set. If we talk about different phases of the vaccine, preclinical, clinical, and also then post-marketing survey of any vaccine, it takes long time. It always took 5 to 20 years. But during the pandemic, first time everybody shown that we can fast track. We can reduce the rate of debiism. We can reduce the challenges in the system for approval method. But when the pandemic started, there was a countrywide lockdown. There was no supplies available. And there were many challenges of the deployment of team, staff, numbers of the people, and passing through all the challenges and making your study to be international is really a challenge for any research team. And there was further, which I told you, that BOC, then many factors of the COVID pandemic, which you need to address while you are doing this study. So after 2 months of constant efforts, finally we could isolate the virus, which was shared by not only AIMS, by Dr. Lalithar's lab and Dr. same time from the KGMU and also from Jaipur lab, and we could get this virus. We could quickly develop all the assay which is required for a vaccine study, and then virus was transferred to Bharat Biotech. There was a constantly Zoom interactions on the study, how we go ahead, how we fast track, and once the vaccine candidate was prepared, quickly it was tested in an IV for antigenicity, stability, what will happen when you possess the virus, or what will happen, what kind of response you get with which adjuvant was studies in animal model. And we found that with LM and also with the IMDG, the IMDG was doing a better indigenicity and immunogenicity when we use a different vaccine model. The first challenge was to find a right animal model. So we used initially many animal model, but finally the hamster was proven to be the best model which was used for challenge study. So these 3 vaccine candidate and placebo was given to hamster. 3 doses of the vaccine were given. They were challenged, and then finally we could monitor not only the clinical but also after the challenge of the virus, what happened to these animals, and we could clearly see that vaccinated group was doing better, and they had a good immunogenicity, antigenicity, and also the protection from the virus. Later on, in the meantime, there was a new rule came that if you want to have approval for phase 3 study, you need to have a non-human primate challenge study data to be present in front of the committee. But in our country, we don't have pathogen-free non-human primate facility available in India. So there was a problem, and we called each research institute JUICE to get these animals. And if you have seen, then finally we have to go to forest after taking all approval, and then catch them, do a fitness testing for 2 months with the standard guideline, and finally we could challenge them. First we infected them, then challenge them, and we could monitor all as per the international regulations. We did all the studies, and we collected the data that include doing X-ray, everyday screening, their immunogenicity, also their clinical scoring, a neutralizing antibody response, a viral genome study, subgenomic study of the viral RNA. And then we monitor each and every 78 tissue of the monkeys were tested for different viral RNA and presence of these histopathology, immunochemistry. So all this data was gathered at a very great length, and finally we could conclude that 3 of the vaccine candidate which was used, the IMD was giving with the 3 microgram and 6 microgram the best result, and finally the candidate was chosen out of these study. Then phase 1, phase 2, phase 3 trials were done, and constantly NIB was engaged with Bharat Biotech, providing all kind of support, including preparing the site, creating the testing, doing the genomic studies when the breakthrough cases happen in either in the vaccinated group or in the control group, and finally we could get the data. And the 16th November, government declared that you can go ahead and vaccinate the individual. But very quickly we realized the first was given to healthcare worker, and a huge number of breakthrough presented. People started questioning if the vaccine is working. So you give any vaccine, all over the world people had the same observation that breakthrough was happening. The next challenge was to see that how the breakthrough, and how after the immunization, if breakthrough is happening, how it is impacting to individuals. So using the 17 sites of VRDL, we did a study among the placebo and also the vaccinated individual, and we did the complete genome sequencing, and we found not only that those people who had 2 doses of vaccinated, they didn't go to admission in the hospital, they didn't die, they didn't have the SPO2 compromised level. And that was a very important finding, and based on that we called as a disease modifying disease, which was a new term coined during the pandemic. And I'm talking about either it's a Covaxin or it's a Covishield, both the vaccinated individual. And if you see this map, in the northern area, we saw the presence of that time, the alpha variant of concern, and the green is all data variant, which was spreading quickly to the rest of the part. There was another claim happened when the Delta spread, that now there is a big wave coming for the pediatric population, and you have to ready for specific provisions for pediatric. So we did a multi-site study, and we found that the children has similar kind of viremia as adults, and they have similar kind of distribution of genome as it happened in the adults. And fortunately, it was true actually. We did not see any specific wave coming for a pediatric population and affecting them. So if I summarize quickly this study, we could develop with Bharat Biotech this vaccine in a very short time, and then we could get approval in 21 November by WHO. But prior to that, there was a lot more other studies done to give this data to WHO. That included constant testing variant of concern in the vaccinated individual. And for that, we have processed more than 10,000 samples just for virus isolation. And we have isolated around 400 virus, only COVID isolates, and many of them were the variant of concern to be used for different studies. We all know that recently in our country, XBB variant, and it evolved to XBB 1.15, and currently we have 1.16, which is going. So to follow these vaccinated individuals, we did a vaccine effectiveness study using the WHO protocol, and this study actually given again the practical efficacy and of the vaccine post-vaccination. There were multiple studies to monitor what is happening in the community after the vaccination and what kind of variations or mutations were happening, and we followed many cohorts in Maharashtra and rest of the country to understand more. We created a large number of regions in our facility, which was shared with BRDLs used in the study to make Atmanirbhar Bharat having own reagents, reduce the cost, and it also forced to reduce the cost. Prior to pandemic, if you are doing NGS sequencing, it used to cost at least 8,000 rupees, but now we can do sequencing it only 2,000 rupees. So that's the change which we brought. The testing, PCR testing, which used to cost thousands, now it can be done only in 50 rupees in India. So these are the sum of changes which happen because of the constant efforts and research. We not only work with the Bharat Biotech, but we also work with the Serum Institute on many products. Fortunately, unfortunately, they never work, but we have done a large number of studies with them for equine immunosera or other studies. We also did tested Zydus Cadila DNA vaccine in non-human primate, which is the first DNA vaccine was approved by Government of India. Though it was not very popular because they have 3 vaccine regime, and in the meantime, you get to properly immunize, you are losing the titer, and also there is a high probability of getting breakthrough infections or reinfection. So that doesn't become very popular, but definitely it has opened a new venue of different kind of vaccine production in India, and that has increased the appetite of Indian pharmaceuticals in making vaccine, which is relevant only to Indian population. So these are the sum of changes. We also work on many antivirals, monoclonal antibody, and with the Zydus Cadila, we have one very successful MABs which give a very good protection in hamster when we work on a preclinical study. A large number of studies only perform on the variant of concern and on the and knowing how the individual who are vaccinated performing. The latest study which we did after the China has a huge surge in January 23. So we can see in Indian population with the recent variants of concern, we tested and we found that we have now a very strong hybrid immunity. And that is why whenever there is an infection spread, people are recovering very fast, and we are not seeing any impact. But especially with the high-risk population, is still having the mask on or taking precautionary measures is still advisable, not only because of that, but also other respiratory infection has suddenly enhanced across the country, and we could see a spurge of many respiratory infections. So this is the recent data which is actually being coming in IGMR, and that shows how Indian immunity is presented and protected against different variant of concern. So there is so the bio threats will be always there, and they will keep coming. So the only thing which we can do, and everybody has agreed after a lot of study analysis, the response to a pandemic is a lot costlier than to invest in the preparedness. So if we whole system, and when I talk, it's not about ICMR, but as a whole system, as a country, we have to prepare, and we have to make all the infrastructure ready in a way that wherever any threat comes, we should identify the first, we do the containment, and we can reduce the disease burden, and we can stop the transmission of the disease. There are many other lessons which we learn that we should be ready, we should have better infrastructure, we should have a better collaboration among different stakeholders. And to make it happen, the government of India, the PSA office has started this One Health mission, which include all the stakeholders of the government, and that is in a process to bring all the BSL-3, all BSL-4, all the laboratory across the country, all the surveillance network, including from NCDC to everyone, to work as a team. It should not be, this is my research, my data, or my state. It should be, we have to think about whole country and how we are going to respond when such emergency happen. And there is a large number of studies is being planned under leadership of PSA office. With this, I just grab your attention to the book which was released and mostly going viral by Balla Bhargav, where we all wrote some of the chapter in that book. And there was a movie was released, and some of you must have seen the movie. In this movie, which if you are really interested to see how the laboratory people, you must watch the portion of the lab, which first time is presented in Indian context, that how we work in the laboratory, what are the challenges when you work in the animal model, and there is a huge team engaged in the pandemic. During the pandemic, we published around 180 research papers that focus only on the pandemic, and in these different, and these papers are published from Lancet to Nature and many high impact factor journals. So definitely we have enhanced the publication impact in India by publishing. So if we talk about the duration of pandemic, it means every week either we are writing a paper or we are revising or we are doing a probe. So entire team was engaged in a way, very systematic matter. There was team who are doing only testing. There were team who are doing only bioinformatic analysis. And I'm only talking, not only talking to BSL-4, but I'm also talking other department who engage with me that include headquarter, Bharat Biotech, and also many other department, Dr. Gajanan from DVG department, Dr. Varsha Poddar from National Influenza Center. But I also have a great and high regards for the army people who got engaged in our animal study and help in some of the very complex experiment. These three people in the uniform, one of is my husband, Colonel Sanjay, also who was involved in this study. And I have a great respect and thank to all NIV team which work tirelessly. The VRDL team across the country, they have done tremendous, excellent, fabulous job in the pandemic for testing, genomic surveillance, giving the data to each state, and taking the pressure and burden in each state. It was amazing work by everyone. With this, I also thank a different stakeholder present in different states who work along with us on different viral disease and provide the support. Thank you very much. And it's my very proud privilege to be here and present the work of NIV. Thank you very much.
Thank you. Thank you, Dr. Pragya. Very beautifully, I mean, a lot of struggle and very impressive work, and it's very good to know that now India is prepared. It's very comforting to know that we have a facility where we can, the so-called exotic viral diseases and hemorrhagic diseases which we used to study, we can work on that. And I think another important thing is preparation of indigenous kits and reagents, really empowered and cost-effective, and widely it could be made available to use in the country. So I must thank you. Thank you for a very good oration, very good presentation, and must congratulate you. Thank you.
Thank you.
So for the IIMM Endowment Lecture Award, I would now like to call upon Dr. Ranganathan N. Iyer, Senior Consultant and Head, Clinical Microbiology, Infections and Infection Control Hospitals, Rainbow Children Hospital Group. Sir's major interest of work revolved around clinical infectious management of ICU MDR infections, coccidian parasitic infections in immunocompetent patients, shigellosis, invasive pneumococcal infections, complicated UTI, and pediatric tuberculosis. He is an astute clinical microbiologist, a keen student of medicine and pathology with a focus on infection management, and is a mentor for those who wish to learn and has been an examiner for infectious disease exams widely in the country. He is a typical Aquarian and a friendly highliner with honest commitment to patient care, preserving his opinion on the topics of academic interest and great passion. Yes.
Good morning, everyone. This may be a talk with a small difference. Clinical microbiology, right from the time when we were postgraduates at the Armed Forces Medical College, I think Sir is there, General Raghunath Sir, and the first thing that he drilled into us is, if what you do doesn't make sense to a patient and his treatment, you have not made any sense at all, and you have not worked at all. This is something which I picked up right from day one at the Armed Forces Medical College when Sir talked to us. And it was reinforced again and again when I joined the Breach Candy Hospital and started working with some of the bigs, Dr. Farooq Udwadia, Dr. Temten Udwadia, and many of the others, Dr. Nargis Motasha, a gynecologist who is par excellence. Again, the same thing was reinforced again and again to us. Stop shaking your test tubes. What you do must make sense to my patient now, not even today, now. And if it doesn't make sense, I'm really sorry, what you're doing is absolutely useless to us. To patient care, and that is very important. The concentration and focus has always been on clinical outcomes of what we do and what we practice in clinical microbiology. I endeavor to share what we have come across as clinical outcomes in our treatment versus infection control protocols that we follow for managing CREs, carbapenem-resistant enterobacteriales, particularly Klebsiella pneumoniae, and more so in the pediatric population. Now, this is something which again is slightly different because everybody talks about managing infections in adults. But then very few papers and very few talks and very few focus is laid on managing neonates, infants, and children. Carbapenem-resistant organisms can target almost every single organ system in the body, so much so we are left perplexed and thinking, are we faced with the next pandemic? And that is of carbapenem-resistant organisms. Even as we are sitting and talking over here, I'm sure we are surrounded by a sea of carbapenem-resistant organisms, particularly enterobacteriales and Klebsiella pneumoniae in particular. And very soon, we would be fighting this pandemic in a useless manner. Are children small adults? If that answer is no, definitely children are not small adults. That is because the pharmacokinetics of various antibiotic distributions is different. Their body mass index is different, their weight is different. So almost everything and anything is different about neonates, infants, and children. And that's what makes them completely different from adults. So that is why children are not small adults. And if children are not small adults, neonates are not small children and adolescents, then the treatment of resistant and recalcitrant to treat infections is also going to be very different in this population of patients. Our first experience and brush with carbapenem resistance dates back to 2007 at the Global Hospital, where it's not that this was not the first patient with cholangiocarcinoma that we saw. We started seeing cholangiocarcinomas right from the day I joined the hospital. And this patient, almost all patients with these kind of tumors in the extrahepatic biliary tree would undergo an ERCP. And sooner or later, they would all go down with fever, chills, and rigor. A blood culture drawn at that time would flag off a Pseudomonas or a Klebsiella. But what was different about this Pseudomonas was that it turned out to be meropenem resistant. Soon we started thinking, in fact, there was a doubt on the quality control of the meropenem disc and the quality control of what we were doing as ABST testing. Once all that was sorted out, if you see right at the right-hand corner down, there is a metallobetalactamase E-test. Now, this is absolutely not used these days, but in 2007, it was used quite commonly because we wanted to know the possible type of carbapenem resistance that we were dealing with in that Pseudomonas isolate, whether it was an MBL producer. And sure enough, quite a few of our Pseudomonas isolates turned out to be MBL producers. Our first brush with carbapenem-resistant enterobacteriales and Klebsiella pneumoniae came in 2015. Until then, things were fairly hunky-dory. We were just moving around trying to treat all our neonatal sepsis. But this neonatal sepsis was slightly different, where we found a cluster of cases in our extreme preterm neonates, which were less than 25 weeks. We admit and care for 24-weekers and 25-weekers. And there, obviously, you have immature lungs and a hyaline membrane disease. And that necessitates a couple of doses of surfactant, most of the times. Almost 99% of these neonates would be administered with that. And sure enough, that would be followed by a Klebsiella pneumoniae bloodstream infection. This Klebsiella pneumoniae again proved to be very different because the meropenem MIC here was 4 micrograms per ml, which was completely resistant. This was followed by many other enterobacterials, which also proved to be carbapenem resistant. And the only thing that we were left doing were MICs for colistin and MICs for minocycline. And that was then later on triggered it changed to tigecycline, particularly for intra-abdominal cases. But then this had limited utility. Treatment successes and failures were completely unpredictable in pediatrics and more so in neonatology. We were also grappled with spread of the Klebsiella pneumoniae, and that proved a huge challenge to infection control in a neonatal unit and in a pediatric unit. Fortunately for us, the hospital shifted from road number 10, Banjara Hills, which was a very small unit with 140-bedded admission, to road number 2, Banjara Hills, very close to LV Prasad Eye Institute, and we started seeing more space, and therefore we could shift beds and keep 6 feet distance between 2 cribs or cots or whatever you call it in neonatal ICUs and pediatric ICUs. And that, along with surveillance for fecal cultures, we started sticking meropenem discs on fecal microbiology cultures, which came to us initially from high-risk units admissions of the PICU and the NICU, and then later on from BMT and oncology. Sometime in 2018-19, we said, no, I think we need to go stick this meropenem disc and look for surveillance of carbapenem-resistant enterobacterials in almost all our fecal microbiology cultures. And that's when we started a CRE screen on all fecal specimens. This is a 3-year data of 2021, 22, and 23. And if you see, there's been a steady increase. It's a small increase, but there is a steady increase from 2.34% to 4.21%. Hand hygiene initiatives were also put in place way back in 2016-17 itself, when we knew that we are dealing with a lot of CRE Klebsiellas, and they are going to remain with us for a long, long time, in the form of enhanced hand hygiene. All hand hygiene initiatives have 3 types of audits in our children's hospital. We do an initial availability audit to make sure that everything is available to people to use, then a direct observation audit, and simultaneously a consumption audit of hand hygiene to see how much is the use of solutions and rubs, particularly when they are there. And then all of this is presented in our infection control meetings. Environmental cleaning on its own, will it be a very good infection control measure? Debatable, controversial. But then you combine it with other things like hand hygiene, combine it with fecal microbiology surveillance and various other kinds of surveillance, and it proves to be a very good measure when you club 2-3 things together in infection control. And this was presented in one of the meetings, where, and then finally the department was invited to be a part of this implementation manual to prevent and control the spread of carbapenem-resistant organisms at the national and healthcare facility level by the WHO. 2017 saw the first reports from UCAST and various other publications which said, you're doing e-test for colistin, gradient diffusion and disk diffusion is no longer useful. So one needs to abandon this and try and do a broth microdilution. So we started doing broth microdilution for our polymyxin and colistin. And then came colistin resistance in 2017, the first case. Now here we had it both in neonates as well as in adults. Of course, we had our first colistin resistance because left, right, and center they were using meropenem plus colistin combination. Because, in an effort to treat the CRE Klebsiella infections in liver recipients. And sure enough, we saw it in a liver recipient in a live-related transplant with a Klebsiella pneumoniae with high MICs to colistin. Initially, it just increases, and I'll show you how our colistin MICs have been in pediatrics. The first case of pediatrics was again from a hemato-onco unit, AML M1, on chemotherapy, who was actually put on schedule for a bone marrow transplant. At this time, we also had our permission to go ahead and take DNB students. So I don't know, I don't think Rahul has come here, but Rahul Ranjan was our first DNB student. And promptly I pushed on colistin susceptibility and various methods of working on it as the first DNB dissertation thesis. And this was published this year, early this year, 2023, in February in the IJMM. We started looking for alternative agents because colistin resistance was on the high. It was close to 9 to 10% in our unit. So we looked at alternative agents. We found ceftazidime avibactam, ceftolozane tazobactam, cefiderocol, meropenem, veborbactam, imipenem, relibactam, astrionam, avibactam. All kinds of combinations are possible for beta-lactam lactamase inhibitors. However, what is available to you is what is useful to us, all right? And except for ceftazidime avibactam, till date, I don't think any of the others have hit the Indian shores. So obviously, we had only ceftazidime avibactam to combine with, and to use colistin combinations, combine it with tigecycline, combine it with rifampicin, though you have tuberculosis to grapple with, combine it with meropenem, combine it with ceftazidime avibactam. All sorts of funny combinations were being tried by our clinical colleagues in their effort to combat CRE infections. So we looked at, we just did a comparative thing between ceftazidime avibactam, ceftolozane tazobactam, and cefiderocol. Everything was very nice about ceftazidime avibactam, which was already available in India, except for the fact that it doesn't act on NDM1 isolates. And whether we like it or not, we are surrounded by a sea of Klebsiella pneumoniae, which are all NDM1 producers, or NDM1 alone, or NDM1 in combination with something else. But there is an NDM1, a New Delhi metallo-beta-lactamase, much to our utter chagrin. The adult dose is fairly well worked out, and the next challenge was, how are we going to use this in our pediatric population, number one? Two, what is the dose that we will use it, even if we get around to using ceftazidime avibactam, trying to do salvage therapy and survival benefit over colistin to our children, all right? And that's when we came across this particular International Journal of Antimicrobial Agents, a very short communication. Normally, you don't even look at those and try and only look at original articles. But then this proved to be a very nice thing sometime in 2019, 2020. We could do combination testing, something like a checkerboard assay or time-kill curves for combination agents that we could do. But then it was even more important to know the genes that you're dealing with, which confer carbapenem resistance before you even get around to doing synergy testing or combination testing. And the main ones that we need to look at are OXA48, NDM1, NDM1 plus OXA48, and KPC, IMP, and WIM. And then we could do a synergy testing using any of these, but we chose a gradient diffusion E-test superposition method out of these four, which were available at that time, and then work out a fractional inhibitory concentration index, which will tell you whether the combination is going to be synergistic additive, indifferent, or totally antagonistic. So this was the synergy testing protocol. We had to do individual MICs for ceftazidime, avibactam, and aztreonam and cross the e-test strips at their MICs. And that's when you get a zone of hope. And that is your zone of hope that you get. It's really a zone of hope when you are battling with carbapenem-resistant Klebsiella infections. Anybody who works in a private setup, a bread and butter microbiologist such as I am, and who is faced with phone calls day in and day out, sometimes even in the night and on Sundays and public holidays. We don't have holidays. We work on all days and at all times, 24/7. That's because people want answers now. It's not today. They want answers now. And for that, you need to do something which will make a huge difference, because neonates are fairly okay in the morning. They look dull in the evening, and the next day they're dead and gone. It's that fast that fulminant infections like Klebsiella pneumoniae, bacteremias can kill an infant or a neonate. This was the adult 24 cases that we managed to give out. Transplant recipients with biliary strictures and anastomosic leaks 8, COVID-19 with sepsis 8, urosepsis with a benign prostatic hypertrophy in 3, and then of course the list just goes on. And the combinations which were used even before people got around to believing us and saying, yes, they could probably use a combination of ceftazidime, avibactam plus aztreonam, were meropenem, colistin, tigecycline, all sorts of combinations were tried. And then finally, after they were all dusted, they got around to using ceftazidime, avibactam, and then look at the number of days after admission that this particular combination was put to use. But then what happened to our children? I was more concerned with what's happening to our children. So we divided them into two categories, the neonates and the infants and the children. The dose of ceftazidime avibactam is close to about 62.5 milligrams per kg in 3 doses. That means 3 times a day, along with aztreonam about 30 to 50 milligrams per kg, again in 3 doses to be given based on the severity of the infection. That's the kind of dose that we managed to work out based on a drug formulary and along with inputs from the clinical pharmacist at the Rainbow Children's Hospital. We had about 40 neonates with bloodstream infections, and there was an input because we had 2 VAPs, 1 necrotizing fasciitis who unfortunately died, and a purulent exudate from the abdomen in a necrotizing enterocolitis of the newborn. 36 of these 14 neonates were outborn neonates transferred from another facility to the hospital. We had inborn neonates only about 4, and 8 of them died, which means close to about 80% of the neonates could be survived this onslaught of Klebsiella pneumoniae, which was carbapenem resistant, and almost many of them even being colistin high MICs. A majority of them were preterm neonates, almost 50% of them. Some of them were late preterm, some of them were extreme preterm, less than 28 weekers, and term neonates were a few. There is a huge overlap on the risk factors and complication, and that's why the numbers will not add up to 40, but then there was a big overlap between those who were mechanically ventilated for respiratory distress, with surfactant, a couple of doses given to them. 5 of them had NEAT, necrotizing enterocolitis of the newborn. 14 of them had undergone surgery to correct either an anatomical anomaly or address a pathology. And 17 of them had deranged coagulation. Predictably, Klebsiella pneumoniae was our predominant organism, leaving E. coli far behind at 6. Now the carbapenemase gene detection. NDM or NDM plus OXA48 formed the largest carbapenemase gene group that we could find in 34 of these Klebsiella pneumoniae isolates. Synergy we demonstrated in 28 out of 34, which was fairly good. So we were encouraged to say that ceftazidime avibactam along with aztreonam could be used in these cases. 3 of these were OXA48, and ceftazidime avibactam alone was wonderfully susceptible in all 3 cases. The colistin susceptibility was fairly good 28 out of 34, and high MICs creeping up to 1 microgram per ml could be demonstrated in 4 of these cases, which means a combination of mero plus colistin cannot be used or will not be efficacious in these cases. Meropenem, the UCAST says that you do a meropenem MIC even if you find it to be carbapenem resistant. If it is 8 or less than 8, then you could probably use a combination of mero plus colistin, but then here all of them are more than 32 micrograms per ml. This was E. coli, which I'm not going to go into too many details because the number itself is pretty less, but synergy could be demonstrated in 4 out of 6 of these isolates. The clinical outcome, 8 neonates died of sepsis and septic shock despite the best efforts. The reasons for these again overlap. Extreme prematurity with necrotizing enterocolitis of the newborn, a polymicrobial bacteremia, sometimes with a co-infection with candidiasis, was found in a great majority of these 8 neonates who died. One neonate died because of the predominant condition with which this neonate was born, laryngomalacia with glossoptosis and subglottic stenosis. And another neonate was lost, unfortunately, to systemic sepsis following a necrotizing fasciitis, which was slightly avoidable. There were 58 infants and older children and adolescents. The admissions were for varied clinical conditions. I'll just project that. The nature of comorbidities, therefore, also varied. And the time from admission to bacteremia to diagnosis was all different in different cases. Clinical outcome was fairly good. 41 could be salvaged, and we could save all those infants and children from dying. We lost 17 infants and children, and some of them were avoidable. The attributable mortality is still being discerned in these cases in our pediatric intensive care unit, and that's why I've not been able to project that over here. The medical conditions for which these 58 infants and children were admitted to the PICU varied. Lymphoreticular malignancies and leukemias. Now here you will find the number of patients as a number. Within parentheses, you will find the percentage. There is a small hyphen, and then there is another numerical. That last numerical tells you in each group how many of these infants and children died. So out of 14 lymphoreticular malignancies and leukemias with AML, ALL, B-cell ALL, and non-Hodgkin's lymphoma dominating the list, 4 of them died. The important thing to note here is the number of deaths were few in each category, but then when you add all of that, it comes to a good 17 deaths. And some of these were avoidable. Like for instance, one couldn't avoid a DiGeorge syndrome with sepsis. One couldn't avoid miscellaneous conditions like scald burns. Many of them do die. But then some of them, like dengue shock syndrome with MODS and a DIC, could probably have been averted. Organisms causing, again here, Klebsiella pneumoniae had 81% of these isolates, topping the list with E. coli and Enterobacter species. A good majority of our Klebsiella isolates, I've just put all of them for comparison so as to save time. And a majority of them either elaborated NDM or an NDM plus OXO48. And 44 out of these 47 isolates we could demonstrate. That means close to about 93.61% of these children had synergy demonstrated, and they did benefit from this combination. Ceftazidime avibactam, predictably in all these cases where OXO48 alone was positive, was also susceptible. The colistin susceptibility, look at the high MIC values. They are pretty high in Klebsiella pneumoniae in 20 children with infants. That closes to about 42.5%, and in all these cases, a mero plus colistin MIC wouldn't have done the trick. Tigecycline MICs were done, and even there, the susceptibilities were not very well preserved in a great majority of these cases. The mortality in 17 of these patients, 41 patients, had a favorable outcome. Neutropenia with overwhelming sepsis dominated the list with respiratory failure secondary to a lung disease. However, HSCT-related complications, we had 3 HSCT patients, of which 2 died, and both of them died of a graft versus host disease. But which were the ones that which were the avoidable deaths in this list of 17 patients. The dengue shock syndrome was avoidable. The steroid-dependent nephrotic syndrome, definitely we could have probably salvaged these and added them to the number of patients who benefited from the therapy if our infection control and prevention precautions were even better. So therefore, all of this can happen only when we have some sort of a plan, a protocol or an algorithm in place, and this is what we have at the Rainbow Children's Hospital. As soon as the blood culture flags off, you do a grams, you know that it's a gram negative. We do something called as a RAST. I'm sure many, many labs in the country now do a RAST, but we started off in 2019, and now the UCAST has, I'm quite happy to share this. The UCAST has put our lab as a collaborative center for RAST in India and for many of our neighbors in Southeast Asia. You get results for gram negatives at the end of 4 hours, 6 hours, and 8 hours, and the soonest you can get, you do a final susceptibility, and if there is carbapenem resistance that can be made out on a RAST, one does a meropenem MIC, a BMD colistin, a tigecycline MIC, and we straight away elaborate a carbapenemase gene detection. So if the gene detected is only an OXO48, straight away do a ceftazidime avibactam MIC, a synergy testing may not be required. If the gene detection is an NDM1 or an NDM plus an OXO48, then of course one will have to follow that entire protocol for synergy testing, cross the agents and work on an FICI calculation and express the results. But if you have to do all of this, it has to be meaningful and it has to be and results must come out in a timely manner. Is it possible from the time the blood culture flags off to the time you give out your reports, 18 hours? Yes, it is possible to give it out within 18 to 24 hours, provided your lab functions 24/7, and you are available as a consultant. Consultant cover for managing infections and infection control must be 24/7. We cannot be leaving it to our technologists and saying they will do something and I'll come on Monday morning and try and do whatever I can or speak to whoever I can. For this, you need dedicated staff, you need timeliness, accuracy, precision. Therefore, treating MDR enterobacterials and preventing them, it's a very fine line. And this goes a very long way in preventive medicine, which is even more difficult because steps in prevention are always very difficult. Thank you so much. I must thank my staff, because unlike bigger institutes and medical colleges where you have an army of people to work on with postgraduates, with senior residents, with research pool officers, we are just 2 of us, and then followed by senior technologists. So I always would give my thanksgiving to my senior technologists because they are the ones who did come fairly well trained, but then they are ever willing to pick up whatever subtle points we do, and they are ever willing to put their small hands at work in the laboratory. Thank you so much, ladies and gentlemen, for your kind indulgence.
Thank you very much, Dr. Iyer. Very interesting talk, and I think something, like you said, could be this epidemic coming. So I think we all have to be careful. And very beautifully, I think you presented. Really enjoyed your talk, and congratulations.
Thank you.
Thank you.
Thank you, sir. I would now request our President, Dr. Arti, to please give a memento to our speaker, Dr. Iyer. Sir, just 2 minutes. This is from the organizer's side. A token of appreciation from the organizer's side, ma'am, I would request. And I would request Dr. Pragya also to come up. Banner aa jata hai piche. Dr. Pragya Yadav. Thank you. Thank you. So we break for tea here and we meet here back sharp at 12. The students who are presenting in the student symposium, please upload your PPTs at the backstage. The students may please upload their PPTs as soon as possible. Good afternoon. Settle down so that we can start the second part of pre-lunch session of IMM. That's the student hour. All the students who are standing outside, please come inside. All the delegates, please come forward. This is a PG symposium, a student symposium. Your presence is required by your peers. So I would now request our Honorable President, Dr. Aarti Kapil, to please come and chair the session. The IMM PG Student Symposium is held every year with 3 to 4 speakers and a moderator. This year, the moderator is Dr. Rahul Narang, and I invite him on this stage to please conduct the proceedings. The topic of the symposium is outbreak investigations for emerging zoonotic diseases.
Good noon, everybody. Honorable faculty members and PG students, SRs. Today we will conduct this symposium on theme of emerging outbreak investigations for emerging zoonotic infections. If you can send text messages to your friends outside, and if they can come inside, please. For this symposium, we have chosen 2 speakers who are second-year postgraduates and 2 who are third-year postgraduates. इसको आप कर सकते हो, कोई somebody can like put it on screen. It's not. It's done. Yeah. No, but then it's not there now. Okay. So we have first speaker, Dr. Aparna Singh, who will talk about outbreak response, introduction and basic terminology. Second is from CMC Vellore, Dr. Gigi Simla, who will talk about steps of outbreak investigations for zoonotic diseases and the role of rapid response team. So these 2 will talk about what is known and what is being followed. Next 2, Dr. Karthik from PGI Chandigarh, who is third year, will talk about which is being contemplated now by Government of India, and I am thankful to Dr. Pragya, who recently talked about One Health mission that is being steered by Principal Scientific Advisor, and role of various organizations that are working on zoonosis. DBT started first, then ICAR started. Indian Council of Medical Research and NCDC are partnering together with Government of India for zoonosis, and since our topic is on outbreak investigations for emerging zoonotic infections, so role of these organizations become more important. Dr. Karthik will talk about how there is a cross-bridging that is being contemplated between human, animal, and wildlife experts and different organizations. Fourth speaker is Dr. Harmanpreet Kaur from All India Institute of Medical Sciences, Raipur. She is third-year postgraduate, and she will talk about something that the role of medical colleges and institutes of national importance can play in controlling, preventing zoonosis. And she is talking about with a futuristic vision, because we have already seen that during COVID-19, microbiologists played a crucial role. So how medical microbiologists working in the medical colleges and institutions, and especially it becomes important for the postgraduates, how their role will be important in combating zoonotic diseases in India. So with these words, I invite my first speaker from Sanjay Gandhi PGI, Dr. Aparna Singh, to talk about outbreak response.
Thank you, sir. Good afternoon, everyone. Today I am presenting symposium on outbreak response introduction and basic terminology. Outline of the presentation is, in this presentation I will be talking about outbreak definition, important epidemiologic definitions, differentiating between outbreak and pseudo-outbreak, need for outbreak investigation, zoonosis definition, risk factors for zoonotic disease emergence, epidemiology and classification of zoonotic diseases and public health zoonotic diseases in India. Introduction, what is an outbreak? An outbreak is defined as the occurrence of disease at a rate greater than that expected within a specific geographical area over a defined period of time. What causes an outbreak? Outbreak is usually caused by an infection transmitted through person-to-person contact, animal-to-person contact, or from the environment or other media. Outbreak versus epidemic. Epidemic is the occurrence of more cases of a disease than expected in a given area or among a specific group of people over a particular period of time. Epidemic is usually applied to situations involving larger number of people over a wide geographical area, whereas outbreak is epidemic, which is limited to localized increase in the incidence of disease, such as village, town, or closed institution. Outbreak versus cluster. Cluster is an aggregation of cases grouped with respect to person, place, and time without regard to whether the number of cases is more than expected or not. Sometimes small outbreaks are referred to as clusters. Outbreak versus pseudo-outbreak. An outbreak is an increase in clinical disease or clinically relevant lab reports, whereas a pseudo-outbreak is an increase in reports or positive culture without evidence of disease. Pseudo-outbreak may be a surveillance or laboratory artifact due to new definitions, improved surveillance, new lab tests, poor sample collection, and laboratory error or contamination. So there is a need to identify a potential outbreak. An outbreak can be identified by regular reviews of surveillance and laboratory data. Clinicians reporting notifiable diseases can alert an unusual increase in disease rate. Now the question arises, why should an outbreak be investigated? Outbreaks should be investigated for the following purposes. First, to define the magnitude of the outbreak in terms of time, place, and person. Second, to identify the cause of the outbreak and its mode of transmission. It's not going. Next slide. Next slide is not.
You have to press this button, the sign.
Okay. Third, to control the outbreak. Fourth, to prevent similar outbreaks in the future. Fifth, to evaluate existing prevention and control strategies. Now I will be talking about zoonosis. Term zoonosis is derived from the Greek word zoon, which means animal, and nouzon means disease. Term zoonosis was coined by Rudolf Virchow in 1880. Rudolf Virchow stated that between animal and human medicines, there are no dividing lines, nor should there be. Health and disease in humans and animals differ only in detail and not in kind. WHO definition of zoonosis, as per 1959, zoonosis are those diseases which are naturally transmitted between vertebrate animals and man. A zoonosis is an infectious disease that has jumped from a non-human animal to humans. According to CDC, zoonotic diseases are caused by germs that spread between animal and people. Etiology of zoonotic diseases can be bacterial, viral, fungal, and parasitic. Modes of transmission of zoonotic diseases can be vector-borne, contact with a living organism that carries the disease pathogen, for example, being bitten by a tick or insect like mosquito or flea, direct contact, coming into contact with the bodily fluids of an infected animal, such as saliva, blood, urine, mucus, feces, etc. Indirect contact, coming into contact with the areas where animals live and roam, or surfaces that have been contaminated with pathogens, such as aquarium tank water, chicken coops, pet habitat, pet food and water dishes. Foodborne, consuming infected food such as unpasteurized milk, undercooked meat and eggs, or raw fruits and vegetables that are contaminated with the feces of an infected animal. Waterborne, drinking or coming into contact with the contaminated water that is contaminated with the feces of an infected animal. Now, what are the factors that led to the zoonotic disease emergence? Wildlife or forest encroachment. Ebola outbreak in West Africa was a result of forest loss, leading to closer contact between wildlife and human settlements. Increased travel, migration of humans has favored the spread of zoonotic diseases, for example, plague, yellow fever, and monkeypox. Intensive livestock farming practices and growing demand for protein-rich food led to the emergence of avian influenza. Poor sanitation and environmental changes leading to increase in vector population. For example, MERS-CoV virus viability was favored by higher temperature, high UV index, low relative humidity. Genetic mutation in pathogens can transform a common pathogen into its deadliest form, for example, COVID-19. Air pollution, cities with higher air pollution level are more affected with more COVID-19 cases and deaths. Increased population and urbanization. Urbanization affects the spread of pandemic, and high population density, poor sanitation, and poverty led to faster proliferation of the infectious disease. Urban expansion and land use change, forestry, mining, and agriculture has led to disease emergence, for example, Lyme's disease. Deforestation and animal trade. Emergence of bat-associated viruses is due to loss of bat habitat resulting from deforestation, example, Hendra and Nipah virus. Epidemiology of zoonotic diseases. 60% of existing human infectious diseases are zoonotic in origin. 75% of emerging infectious diseases of humans like Ebola, HIV, and influenza have an animal origin. 80% of agents with potential bioterrorist use are zoonotic pathogens. 5 new human diseases appear every year, 3 are of animal origin. Now the classification of zoonotic diseases. Zoonotic diseases are classified on 3 bases. First, according to the etiological agent, second, according to the reservoir host, and third, according to the mode of transmission. According to the etiological agent, zoonotic diseases can be bacterial zoonosis, rickettsial zoonosis, viral zoonosis, protozoal zoonosis, helminthic zoonosis, fungal zoonosis, acellular non-viral agent, that is prions, and ectoparasite. Examples are listed here. According to the reservoir host, zoonotic diseases can be anthropozoonosis. These are the infection transmitted to man from lower vertebrate animals. Examples are rabies, leptospirosis, plague, brucellosis, arboviral infections, and Q fever. Zooanthroponosis, infection transmitted from man to lower vertebrate animals. Examples are staphylococci, streptococci, diphtheria, enterobacteriaceae, human tuberculosis in cattle and parrots. Amphizoonosis are the infections that are maintained in both man and lower vertebrate animals and transmitted in either direction. Examples are salmonellosis and staphylococcosis. According to the mode of transmission, zoonotic diseases are direct zoonosis. These are the infections that are transmitted from an infected vertebrate host to a susceptible host by direct contact or through fomites. Examples are rabies, anthrax, brucellosis, leptospirosis, and toxoplasmosis. Cyclozoonosis are the infections that require more than one vertebrate host species, but no invertebrate host for the completion of the life cycle of the agent. Examples are echinococcosis and teniasis. Metazoonosis are the infections that are transmitted biologically by invertebrate vectors. Examples are plague, arboviral infections, schistosomiasis, and leishmaniasis. Saprozoonosis are the infections that require a vertebrate host and a non-animal developmental site like soil, plant material, pigeon dropping, etc. for the development of an infectious agent. Examples are aspergillosis, coccidioidomycosis, cryptococcosis, histoplasmosis, and zygomycosis. In India, the public health zoonotic diseases of importance are rabies, brucellosis, toxoplasmosis, cysticercosis, echinococcosis, leishmaniasis, Japanese encephalitis, plague, leptospirosis, Krupp typhus, Nipah, Cassandra forest disease, and Crimean Congo hemorrhagic fever. Now I will summarize the presentation. Continuous surveillance and research should be promoted to become well equipped for the future emergencies. Early detection and response to outbreaks should be promoted. More than half of all infections that people can get are zoonotic, and 3/4 of emerging infectious diseases are zoonotic in origin. According to an International Livestock Research Institute study, 13 zoonotic diseases are cause of 2.4 billion cases of human disease and 2.2 million deaths per year. Highest zoonotic diseases burden are in Ethiopia, Nigeria, Tanzania, and India. Zoonotic diseases is thus a global problem, and it has received priority attention of the WHO. So a proper outbreak investigation will help in identifying the proper epidemiological link of the infection and to prevent episodes of that outbreak in future. Thank you, everyone. The next speaker is Dr. Gigi Smilla, and she will be talking about steps of outbreak investigation and rapid response team.
We'll take up questions in the end of the symposium.
So good afternoon all. Today my topic of presentation is about the steps of outbreak investigation of a zoonotic disease and the role of the rapid response team. So basically there are 10 steps involved in an outbreak investigations, which I will detailing explaining in each slide from the following. So each step play a vital role in an outbreak investigations. So to make it more relatable and understandable, I'll be taking Nipah virus as an example throughout my presentation. So the very first step of an outbreak is verification of diagnosis. So for any outbreak, the diagnosis should be verified both clinically and laboratorily. So sometimes wrong reports or misinterpreted reports might give a false alarm of an outbreak. So any diagnosis should be verified clinically by evaluating the signs and symptoms, duration of exposure, and the onset of the disease. And the laboratory confirmation should be specific for that particular etiological agent. So in case of Nipah, the signs and symptoms will vary from fever with respiratory signs or fever with altered mental status. And the exposure and onset. Exposure would be due to bats, infected bats or infected palm fruit juice. And the laboratory confirmation will be done by RT-PCR. So to verify the diagnosis or to investigate an outbreak, there should be a deployment of rapid response team. So the rapid response team members must include chief medical health officer or a district health officer and an district animal husbandry officer and the district level wildlife representatives. So and likewise, state level zoonotic committee and the district level zoonotic committee should also be sensitized and apprised. So for any outbreak investigation to be effective, this rapid response team should be mobilized as soon as possible. So the deployment of Central Rapid Response Team can be mobilized by the relevant ministry, which is Ministry of Health and Family Welfare and the Ministry of Fishery, Animal Husbandry and the Dairies. In case of Nipah, the Rapid Response Team is situated in the Kozhikode Medical Hospital, which includes the Department of Human Health, Wildlife, Forest, Agriculture. So the next step will be the confirmation of the outbreak existence. So first step is verifying the diagnosis and the next should be the confirmation of outbreak. So any outbreak is a number of cases which is exceeding the what would be expected for a particular population over the particular period from comparing to the previous data. So this confirmation can be done by reviewing the surveillance data from clinicians, veterinarians as well as the laboratories. So since 2001 there has been 6 outbreaks in India. So the very first outbreak is in 2001 in West Bengal, Siliguri district. There has been 66 confirmed cases of Nipah, and among 66, 45 were died. And the next outbreak was in 2007, same West Bengal district, Nadia, where the mortality rate has been 100%. Among 5 confirmed cases, all 5 were died. And the next case outbreak was in first time in the South India in Kerala in 2018 in the Kozhikode district of Malappuram. There have been 18 confirmed cases and 17 were died among 18. So, and the next followed outbreak is in 2019 and 2021. In both years, there have been 1 confirmed cases, and in 2021, mortality rate was 100%. So the recent outbreak occurred in 2023 in the same Kerala, Kozhikode district. There 4 cases were confirmed, and among 4, 2 were died. So the third steps will be informing the key stakeholders about the investigations. So the key stakeholders would include local public, appropriate health facility staff, and the laboratory staff, and the local and national public health officials as appropriate. So next will be the constructing a case definition. So any case, for any outbreak, the standard case definition will ensure that every case is equivalent regardless of when or where it occurred or who identified it. So the elements of case definition must include a clinical criteria, signs and symptoms, and the person, place and the time criteria, as well as a laboratory confirmation. And the classification of case definition includes suspected case, probable case, and a confirmed case. In case of Nipah, the suspect case is a person from a community affected by a Nipah, where the disease outbreak has occurred, who has fever with new onset of altered mental status, or fever with seizure, or fever with headache. So the probable case will be a suspected case who has been residing in the same village where the confirmed case of Nipah were living, and who died before the complete diagnostic specimen could be found. And the confirmed case is laboratory confirmation of the case with an RT-PCR. So the fifth step is identifying and counting the number of cases and collecting the information of cases. So this case finding can be done by various aspects like active surveillance, passive surveillance, and the follow-up of the cases and the contact tracing. So active surveillance can be done by directly questioning the patient or the local public. And the passive surveillance means reviewing their medical records and finding out the cases. And follow-up can also be done in each cases. And contact tracing. Contact tracing is finding an individual who have been in the proximity with the confirmed case. So in case of Nipah, this contact tracing is done in 2021, where one index has been found presenting with fever in a private clinic on day 1. And then the day 2, the fever has been persisting. And the day 3, he was shifted to a hospital number 2. And the fever was persisting, and the patient has developed condition was deteriorating, and the patient has developed encephalitis as well as myocarditis. Then the patient was shifted to another hospital. So from this, the patient who have been in contact with the patient are all been quarantined and tested. So this is a contact tracing example. So among 4, active surveillance will serve as a prime important. So next is the describing the data by place. Before beginning any investigation, so detailed map of that particular area is advisable. A spot map will help in identifying the clustering of cases in a particular area. So in case of Nipah outbreak, 3 kilometers from the Kozhikode district will serve as an epicenter for Nipah outbreak. The 6th step would be the examining descriptive epidemiological features of cases. So descriptive epidemiological feature will describe the case by person, place and time. And it will also characterize the outbreak and identifies the population who are at risk and provides idea about the agent or the source or mode of transmission. And it also provides information to begin the control measures. So in case of Nipah, having close contact with infected animals such as pigs, bats or any humans infected with the virus, and also consuming raw date palm sap that is contaminated with the bat excretes. So in an outbreak, the epidemic curve will serve as a graphical representation of distribution of cases by time of onset of illness. In an epidemic curve, this x-axis will serve as a date or time of illness of onset, and the y-axis will tell us a number of cases of illness. So this is a graphical representation of a outbreak of febrile encephalitis in a Siliguri district in 2001. So where they have plotted a hospital associated and a non-hospital associated cases and deaths. So this epidemic curve should distinguish between a probable case and a confirmed case of, and the shape of the epidemic curve should suggest a single point source as well as ongoing transmission and any intermittent source of transmission. So this epidemic curve will depict the magnitude, time trend of the outbreak, and it also defines the incubation period and the pattern of spread and highlights the outliers. So outliers in an outbreak investigation must include earlier case as well as a later case, which have been lately diagnosed or have been misdiagnosed. So to take the precautionary measures, surveillance should be done in various aspects, like the pathogen should be reported, and the sero surveillance in both humans and the animals should be reported. And the syndrome surveillance, like in case of example, acute febrile illness, we need to track the earliest symptoms which is occurring, whether fever is followed by cough or cough is following the fever. And the risk surveillance should also be done, which is tracking the risk factors. And the seventh step would be the observation and review of patient care. So this standard observation tool will be useful in creating a reviewing the patient care. So this observation tools must include the medication details of the particular case, and any laboratory or any clinical investigation report should be reviewed. And after that, we need to generate a hypothesis. So this hypothesis will give an idea about the association between an exposure and the disease. So this hypothesis should be tested and confirmed. So it can be tested by using an analytical study. Example, case control study can be done, which will compare the exposure among cases and the controls. Like human samples, collecting and testing the animal and environmental samples will also be needed for any outbreak. So this will complement the epidemiological arms. So in case of Nipah, environmental samples like palm fruits can be tested and confirmed, and animal samples, bats or pigs can be tested and confirmed. So the final step would be the implementing control measures and the communication with the stakeholders about the investigations. So the control measure, the main goal is to stop the transmission and not necessarily to find the source of transmission. So the variety of control measures should be implemented, targeting in various aspects based on the initial observations. So and after control measures has been done, we need to follow up the patient, which will refine the case definition of an outbreak, and also it will modify the control measures or follow up the investigations. And it will also provide a better epidemiological evidence about the source and the mode of transmission. And it reviews the infection control measures if they need to be continued, changed or reduced. And the final report of the outbreak should be shared with all the stakeholders. That should describe the outbreak, interventions what we have made, and the effectiveness of the measures we have taken. So this to summarize, so according to WHO, outbreak investigation includes 10 steps approach, and the deployment of rapid response team is based on integrated disease surveillance program, and standard case definition is a prime need for the identification of an outbreak. So and this epidemic curve will depict the magnitude and time trend of an outbreak. So the primary goal is to stop the transmission through an integrated One Health approach. So the next speaker will be talking about the cross-bridging between human, animal and wildlife, which is presented by Dr. Karthik from PGI. Thank you.
Good afternoon to all. My topic of symposium is about cross-bridging between human, veterinary and wildlife. So I'm going to talk about One Health and their approaches, how they're helping us to prevent the zoonotic infections. So coming to the history of the One Health, after the outbreak of After the outbreak of H5N1, the avian influenza in 2006 and 7, it created an awareness regarding the health of humans and animals in the various organizations, which includes World Health Organization, Food and Agricultural Organization, World Organization of Animal Health. So they created the solution called One Health. They formed the strategic frameworks and the policies regarding the health of humans, animals, and the ecosystem interface for preventing the zoonotic infections. So these three organizations together called tripartite group. Later they collaborated with United Nations Environment Program. Now it is called a quadripartite group. So in 2015, they commit to eradicating the human deaths through the rabies by 2030. This One Health not only targeting the controlling of the zoonotic infection, it will also help us to prevent and control the antimicrobial resistance, which is a global threat nowadays. So these are the health challenges coming under the One Health. Foodborne disease due to the zoonotic infection, issues related to the climate change, food security, biodiversity, environmental health hazards like air, water pollution, and deforestation. So those quadripartite group, they signed a groundbreaking agreement regarding the One Health and their strengthen the collaboration between various sectors. And this is the definition of One Health, which is an integrated unifying approach that aims to sustainably balance and optimize the health of humans, animal, and ecosystem interface. It also recognizes that health of the humans, animals, and the environment, they are closely linked as well as the interdependent. So the successful implementation of the One Health requires 4 C's. Those are collaboration, communication, coordination, and capacity building in between the various sectors. So why do we need One Health nowadays? So the various microbes, they are infecting the humans and animals, they are sharing common environment. So effort in either only one sector, it cannot prevent or eliminate the disease. Example, in case of influenza A, the person-to-person transmission of the infection, which will be higher during the climate changes like absolute humidity in both summer and winter season. So the environment plays an important role in transmission of the various zoonotic infections. So what is the reason behind the new emerging zoonotic infections? One of the reasons are spillover events, which refers to cross-species transmission of the pathogen from the natural reservoir host to the new host species, which includes animals and the humans. So there are 3 stages. In stage 1, which is called pre-emergent state, in which the naturally occurring pathogen, it will be transmitted in between the animal reservoirs. From the wild animals to the domestic animals during some ecological factors like urbanization, increased population growth, or deforestation. In stage 2, which is called local emergent state, in which the transmission, in which the spillover even already occurred in the humans. So the person-to-person transmission only at the localized level. Whereas in stage 3, the person-to-person transmission indefinitely present at the global level, there will be an emergence of pandemic or epidemics. So these 2 pictures shows that spillover event and the stages and the control of the spillover events. In first picture, the light pink areas represents the stage 1 spillover from the wild animals to the domestic animals. The light green area represents the disease outbreak which occurring in the domestic animals. The red area represents the disease amplification and transmission among the humans, which is stage 2 and stage 3 spillover. So the second picture, when we are taking early action or forecasting readiness in the wild animals, or we are controlling this infection in the domestic animals, which will lead us to control the infection in the animals and humans, it will be a benefits to both humans and animals when we are comparing to the continuous spillover events. So the early control of the zoonotic disease in both cost effective and prevents the human disease. These waves are represents the spillover events. So when we are taking the early action, by taking care of the animals and humans on the early stage of the disease outbreak, there will be less cost for the controlling the outbreak. Otherwise, it will be huge. Example, in case of COVID, we face that situation and we control this infection during the stage 3. So what is the reason behind the SARS-CoV-2 outbreak and the new variants? So this paper published in Journal of Clinical Microbiology in April 2023, they given the role of spillover and spillback in the transmission and the importance of the One Health. So this picture shows the various transmission pathways of the COVID-19 from the natural reservoir host to the humans and the non-human animals. So in spillover, the natural reservoir host here is a bats. So from the bats, it will be transmitted directly or from the intermediate host. There are 2 important spillover here is spillback and the secondary spillover. In spillback, the pathogen transmitted from the humans to the non-human animals, they are cats, dogs, and gorilla, etc. In secondary spillover, which is importance to the public health concern and also for the One Health, because which is transmitted in between the humans and animals in the both ways, from the animals to human and the humans to animal. So in this paper, they mentioned the reason behind the current and future pandemics due to the virus readily transmitted in between these animal host and humans. So it will lead us to accommodate the mutation in that new host, and there will be a formation of new genetic variant and increased transmission capabilities. So continuous surveillance on these animals will give us a clue about the new genetic variant of the COVID-19. So cross notification, which is important one, because early information sharing between the two health sector will give us a clue about the source of infection. So example, in Lyme disease, the two forest guards reported with the history of high-grade fever, headache and muscle ache with the history of tick bite. So the same day, human health sector people, they inform this notification to the animal health sector. So the animal health sector, human sector and the forest sector people reach the forest area and visit the premises, and they will take the samples from the ticks and they will send it to the laboratory and diagnose of the infection, and the further action will be taken. So cross-reporting of the zoonotic disease, which is essential one, because early action will be taken by the concerned sector and collaborative action to mitigate the early event. So this picture shows the cross-reporting between two health sector, human health sector and animal health sector at various sector levels, district, international, national and state level. So coming to important part of One Health is PPR approach. P means prevention, P means preparedness, and R means response. So prevention here is primary prevention, because we are preventing the spillover from the animals to the humans. Preparedness, response are secondary prevention, because spillover event already occurred in the humans. We are preventing the spreading of the disease among the humans. So what is the aim of prevention? So we are going to prevent the zoonotic disease by studying the dynamics of the infection and the risk factors by the One Health coordination mechanism. These are the strategies for the prevention. There are 4 strategies. Important one are understanding the infection dynamics in the natural host and the environment. There are many plans under this strategy, which includes state can provide the lab facility to detect the antibodies which is present in the animals and humans for detecting the prevalence of the infection. And also the state can establish a robust surveillance system during the ecological factors and post-disaster conditions like earthquake and flood. The research institute can focus on continuous monitoring on the natural reservoir host which harbors the pathogen. In case of Japanese encephalitis, pigs are the amplifying and natural host. So we can continuous monitoring on these animals will give us a clue about the evidence of the virus and the risk population. And next strategy is addressing the anthropogenic drivers of the disease emergence. So state can analyze the disease emergence based on the route of transmission and the continuous surveillance on the wildlife trading in the black market, and surveillance in between states if there any outbreak in the nearby states. So next strategy is risk reduction activities. So this is not only the single approach, this is the individual, community and government approach. So state can determine the risk posed by the zoonotic disease based on the prevalence, severity and the population vulnerability, because in example, respiratory tract infection common in the children and the elder age group. So pet owners, they should register their pets, and the full vaccination of the rabies should be done. And the state can establish the effective vaccination strategy and the collaborative mechanism and chemoprophylaxis to the vulnerable people. They are coming under the risk reduction activities. Integrated surveillance in between the animal, human and the environmental health sector also give us a clue about the early outbreak of the infection. So in the preparedness part, early warning signal is the important one strategy. So early warning signals can come from the various sources like human health sector, veterinary health sector, media and community. So continuous surveillance of these sectors will give us evidence about the warning signals about the emerging zoonotic infections. So sentinel surveillance is important one, because of the unexpected death of the animals also give us a clue about the disease emergence. In case of Casnur forest disease, dead monkeys will give us evidence established disease in the particular area. So what are the challenges for managing the zoonosis? Poor level of awareness among policy decision makers about the serious nature of the disease, and the inadequate resources and skilled manpower for the control of the zoonotic infections, weakness or absence of the collaboration or cooperation between the public health, veterinary, agriculture, and wildlife sectors, weak disease surveillance system, and inadequate diagnostic capacity to detect the zoonotic infections. So insufficient capacities of the countries to plan, mobilize, implement appropriate control measures. So what are the strategies for controlling the zoonotic infection? By building the effective collaboration between the animal, human, and environmental health sectors, and strengthening the laboratory diagnosis for the novel pathogens, improving the case management and the infection control, integrating vector control measures, developing the epidemic preparedness response capacities for the emerging zoonotic infections, improving the surveillance for the early detection of the disease threats in the humans. So these are the existing surveillance system which present in India currently. For human disease, integrated disease surveillance program. For animal disease, national animal disease reporting system and national animal disease referral expert system. So coming to the summary of my topic is, with the help of the One Health, the world better able to predict, detect, and respond to the particular human threat, and also improve the health of the humans, animals, and environment by continuous sustainable development at the various sector level. So thank you. I'm calling next speaker Harman Preet Kaur for role of microbiologist.
Good afternoon, everyone. I'll be speaking on role of microbiologists and regional coordinator for One Health. So Government of India, along with National Center for Disease Control, they have taken up many projects for controlling zoonotic diseases in India. And the first project is National One Health Program for Prevention and Control of Zoonosis. And the second one is National Program on Containment of Antimicrobial Resistance. In fact, India has hosted and presided G20 Summit of 2023, and the theme was Vasudeva Kutumbakam, which means world is one family. And zoonotic influenza tops the list amongst top 10 prioritized zoonotic diseases in India, followed by anthrax, Japanese encephalitis, brucellosis, dengue, rabies, scrub typhus, plague, and the last is Crimean Congo hemorrhagic fever. And NCDC, along with various national institutes like Indian Council of Medical Research, Indian Council of Agricultural Research, Department of Animal Husbandry and Dairying, along with Wildlife Institute of India, with their respective ministries, they have collaborated at the national level to formulate and design policies for One Health, and there will be 15 regional coordinators who will be operating those policies and plans in their respective regions, and there will be 32 sentinel surveillance sites to study the burden of zoonosis and epidemiological risk factors associated with the diseases. And this is the institutional mechanism of One Health. This is the schematic diagram of institutional mechanism of One Health, where NCDC, ICAR, ICMR, along with Wildlife Institute of India and DAHD, they are collaborating at the national level to design the policies for One Health program. And then there are 15 regional coordinators who will be guiding different state level zoonotic committees and district level zoonotic committees. They'll do active disease surveillance in the 32 surveillance sites, where they'll do active surveillance for disease risk mapping. And then where do these regional coordinators stand in this National One Health program for control of National One Health program? First will be to strengthen the intersectoral coordination on zoonotic diseases. Second is to organize joint training for human health, veterinary and wildlife departments. Third will be laboratory strengthening for diagnosis of zoonotic diseases. Fourth will be mapping of zoonotic diseases. We can actively do risk mapping of, we can do active risk mapping of the zoonotic diseases in the endemic areas. And the fifth will be advocacy in preparation of state action plan for prevention and control of zoonotic diseases. And the last will be IEC activities, that is information, education and communication activities. And what will be the role of VRDLs? VRDLs have the objective to create infrastructure for timely identification of viruses and other agents and to develop capacity for identification of novel and unknown viruses, emerging and re-emerging viral strains, and to develop diagnostic kits. And we all know that VRDLs have performed really well to control the COVID pandemic in India. This is the structure of VRDL, this is the network of VRDL labs in India. We have grade 1 regional laboratories, grade 2 state laboratories and grade 3 medical college laboratories, where NIV Pune and NCDC Delhi are the apex laboratories, and National Institute of Epidemiology Chennai manage the data generated by the network of VRDLs, and they also do timely analysis. So what will be our role as microbiologists in this One Health program? Our role will be to strengthen laboratory diagnosis for early detection and diagnosis of zoonotic diseases, which is very critical for preventing outbreak and controlling transmissions. It is also important to develop, standardize and validate new diagnostic tests for zoonotic diseases that are emerging or that are difficult to diagnose. So to strengthen the laboratory diagnosis, under ICMR, under PM Abhim program, is working to expand its network of BSL-3 and BSL-4 laboratories to strengthen diagnostic infrastructure. And VRDL along with ICMR, along with NIV and NIE, and along with state VRDL labs, they have worked hard to contain the Nipah outbreak of 2023 in Calicut district of Kerala. They all worked together and they did active contact tracing of 900 people to contain the Nipah outbreak in the district. And then we as microbiologists will guide how to take sample, when to take sample, and from where to take sample. And then these samples will be sent to the respective laboratories where they will be processed with standardized SOPs in BSL national network. And the data generated from each laboratory, that is human laboratory or animal laboratory, will be uploaded in the data cloud, and the data from different labs will be linked by common identifiers to develop a comprehensive on disease outbreak. So we as microbiologists have role from sample collection to data processing, and then to sample processing to data collection, so that we can collaborate at each and every sector for the operation of National One Health program. And we all know that the existing BSL-3 laboratories are fixed. So sample transportation and collection is a cumbersome project. For that, ICMR has joined hands with CleanseAids to design and build India's first mobile BSL-3 enhanced laboratory. And we have ICMR has deployed this mobile BSL-3 for Nipah outbreak in Kerala. The second role of microbiologist will be to establish sentinel surveillance sites. This is used to monitor the occurrence of zoonotic diseases in both humans and animals, and this information can be used to detect outbreaks early and to track the spread of the diseases. The third aim will be in research and development to develop point-of-care diagnostic kits, which will be available at the periphery. And fourth will be molecular epidemiology of zoonotic diseases to trace and track the spread of the pathogens. And fifth will be intersectoral linkage for detection of antimicrobial resistance. And the final will be provide quality control and quality assurance facilities to the other laboratories. What will be the role of different medical colleges in this National One Health program. The first will be to provide referral services for management of zoonotic diseases in the center, and the other will be laboratory services for diagnosis of various zoonotic diseases. So I'll explain here the model of AIIMS Bibinagar, which they have established at their regional center. They have collaborated with various state colleges to collect samples from humans and companion animals, and the state labs will process those humans and companion animal samples. And they have also collaborated with Center for DNA Fingerprinting and Diagnostics, where they'll be taking samples from the domestic animals, and they'll be working on zoonotic or MDR TB. They have also collaborated with National Research Center for Meat, where they will be taking samples from animals and samples from animal handlers from the slaughterhouses. And they have also collaborated with Center for Cellular and Molecular Biology for Animal BSL-3 services. They have collaborated with Amrita School of Biotechnology, Kolam, for epigenetic services. And then the most important thing is they have established One Health demographic surveillance system around AIIMS Bibinagar, where they are actively collecting demographic data from the humans, and they are also taking data about animals, housing conditions, and from the livestock also. So they are working at every level, at every sector. They are taking samples from the rivers if they are flowing through that village, and they are also taking water samples from the sewage waters. And then we have AIIMS Raipur model also. We have collaborated with Kamadhenu Veterinary Institute, and we have also collaborated with Indira Gandhi Krishi Vidyalaya for agricultural, which will provide which will be a stakeholder for agriculture, which will be an agriculture institute, and will provide and which will help us in collecting samples from the environment. And the aim will be to operationalize the center devoted to achieve One Health concept and to establish One Health laboratory. So where do we microbiologists stand in this One Health program? Our role is to move from conventional diagnostic microbiology to clinical microbiology, and to a great extent we have achieved that. And now our role will be further to inculcate that clinical microbiology into the field work in real time at the ground level, so that we can achieve public health, we can practice public health microbiology and achieve One Health. So our goal will be to identify emerging and re-emerging pathogens, and to trace the spread of pathogens, and to develop policy and plans to control the zoonotic diseases and to reduce antimicrobial resistance. Thank you.
Thank you, Dr. Aparna, Gigi, Karthik, and Harman. I think all of you can come now to the stage, and we will take questions on the presentation. Yes.
Just one comment before we start the questions. First of all, I must congratulate you and congratulate Dr. Narang to have taken such an important topic for symposium, because one experience I tell you now from being on the other side of the table, one question very commonly asked in interviews as well as viva, as well as theory, is the outbreak investigations. And you know what happens in microbiology? The first thing everybody says is collect samples from the environment, the swabs, which is not the thing, because we have to get that out of our mind. That way, I think this was very broad and very wide coverage. Some of the aspects probably even we haven't really read. So that way, very beautifully done. I must congratulate you all. Thank you. And now I'll give it back to Dr. Narang.
Thank you. Thank you so much, ma'am. So we'll take up questions now.
Do you think this is important? How important is public health microbiology? Anybody, please? Instead of a question, a comment. Yes, sir. Mics. Sir, please, I'm giving you the mic.
The question which I would like to ask is that the models that the four speakers have given us the very beautiful plan, and I like the presentation. The question I would like to raise here is that whether the plan would differ depending on the type of microbial agent that is causing the outbreak. For example, virus. Another example could be any bacteria which is causing outbreak or epidemic, or say parasitic infections like malaria-like infections. Just I would like to know.
So anybody wants to take up this question? Third year postgraduates, yeah. Udhar aa gaya.
For now, we have one common plan to address all the infectious diseases. We don't have any different plans for.
Think and tell. Will be different for respiratory viruses, will be different for malaria.
Yeah, for diseases which are vector-borne, we have national vector-borne disease control programs. And for in case we have any outbreak of respiratory viruses, we have different plans.
The purpose of asking this question is relevant in the sense that there are many clinical syndromes where the etiological agent cannot be pinpointedly suspected. It could be in some time the viral etiology, it could be some parasitic etiology, it could be rickettsial disease, or it could be something else. So the question remains that what should be plan of the nature of investigation that you organize?
Sir, I think ICMR is well aware of this, and now they have started syndromic approach. And recently there has been a meeting, and they are proposing a study, task force study, where they will utilize services of VRDL to work on different kind of agents in addition to viral agents using One Health approach. And similar question was also raised while optimizing BSL 3-4 laboratories across India. Dr. Pragya was part of that meeting, and we know that right now the BSL 3 laboratories that are working in India are either working on viruses or bacteria. So we have to optimize them also, but then syndromic approach with initially as conceived by ICMR appears to be a better approach, sir.
Thank you. Thank you.
Yes, Dr. Muneen, I was going to ask you actually.
I'm Dr. Monil, and I'm Joint Director in Center for Viral and Zoonotic Diseases in National Center for Disease Control, and one of the stakeholders in drafting these guidelines for regional coordinators and the sentinels. So I just wanted to ask, as you are upcoming new faculty, when you say about that we are moving from the hardcore microbiology to the clinical microbiology, do you think that diagnostic stewardship is going to play an important role in this? And what do you mean by diagnostic stewardship when you are saying that we are going to diagnose the outbreaks? So any idea about it? I simplify my question. Suppose you get a patient of pyrexia of unknown origin, and he is landing up at 14th day of illness. Now, what are the kind of tests you want to do? And there has been lots of deaths in the same area having similar kind of presentations from a particular village in a block. So what kind of test do you think that you are going to do? and they might be children. You can't take a sample more than 5 ml or maybe more than a 2 ml. So what kind of test you are going to first?
Chalo, I think you apply your mind and then during lunch discuss with madam. Yes, this is an important. One last comment, sir. Yes, please. Then we'll break for you.
I have been listening to this, and I saw the theme was a brilliant theme, One Health program, and I must applaud the ICMR's initiative in this. The VRDL, which started during Dr. V.M. Katoch's time, because I hold his hand and took it to Banaras. Earlier in our days, clinicians or clinicians were not caring for the microbiologist. And microbiologists were always considered as their wives, whatever they order, we have to do it. Or they don't even care for the report and they start their own treatment. That's how we have got the MDRs and all, which Dr. Aarti talked a very nice talk there about Salmonella typhi and other paratyphi and etc. So my intention to come here was to congratulate the organizer and the Government of India for holding such, and this type of symposium should be held in different institutions as well to educate them.
Thank you, sir. Thank you. Thank you, everyone. Thank you, Dr. Narang.
Thank you.
And the students.
Thank you, ma'am. I would like to request now the chairperson, Dr. Aarti Kapil, to please give a memento to all our speakers. Dr. Gigi. Dr. Aparna ka ready hai? Dr. Aparna? No. Dr. Aparna? Dr. Karthik? And Dr. Harmanpreet. Dr. Harmanpreet. Please give a big round of applause for these young budding microbiologists who've presented such great presentations in front of you, and I think enriching all of us with their academic inputs. I'll now request Dr. Aarti, our chairperson, to please give a token of appreciation to the moderator, Dr. Rahul. Thank you so much, sir. So that brings us to the end of the IMM session on day 1 of this 46th National Conference, and we'll meet at 2 p.m. after the lunch break. Lunch is downstairs? Lunch is down in the lawns. For lunch, please move downstairs to the lawns.
all those who are interested in the IM quiz, please send your name, email ID, and phone number to these numbers, which are displayed on the slide. Please do it as the earliest.
इधर सीधा में
horizontal. Good afternoon, everyone. I welcome you all once again for the post-lunch session. Now we'll be holding a symposium by US Centers for Disease Control and Prevention on role of microbiologists in disease surveillance and outbreak investigations. For this, I would like to invite our chairpersons, Dr. B.L. Cherwal, sir, Dr. B.L. Cherwal, sir, is Director and Professor Microbiology at Lady Hardinge Medical College, New Delhi. Sir has an honorable resume, along with which he has received a distinguished award of Double Helical National Award, Bhujwala Award, DMA Distinguished Service Award, and Silver Scotch Award. I welcome you, sir. I request Dr. Sherwal, sir, to kindly introduce and invite our co-chair for the day and also our speakers for the symposium. Thank you, sir.
Thank you for the opportunity. May I request to come on dais Dr. Dan Vendredi, who is the lead AMR, to chair the session. I understand the post-lunch session is really very tedious, session where people are very difficult to find, and if they are there physically, they are not there. But I am sure our speaker will make sure that you are not fell asleep. I request on behalf of the organizers to the audience and the delegates, can you please move little forward, because this hall is little big. My personal request, please try to come forward. Maybe the front 10 rows or 15 rows. Again, I request, please try to come forward, whosoever is sitting in this hall, to make the presence in such a way that the speakers are really heard well. This session is known as basically CDC own session, and we are going to listen to two wonderful speakers. One is Dr. Mala Chabra, who will be speaking on the role of microbiologist, and another is Dr. Mayank Dwivedi. As we all know that there are two important areas for microbiologists to act and to practice. One is, as we all know, that we are working in the hospitals. We try to be known as clinical microbiologists, and we are working hard for diagnosing the disease and trying to be part of the treatment of the patient being treated in the hospital. The second area, which is nowadays important, is public health microbiologists, who are responsible for surveillance and for preventing of the infections in the society at large. So during this session, we will try to define the role of individual, that is public health, and of the clinical microbiologist, and see how combined together they can make an impact on the healthcare services in India. I call upon the first speaker, Dr. Mala Chabra. She is a consultant microbiologist in Atal Bihari Vajpayee Institute of Medical Sciences and Dr. RML Hospital. She was formerly working as Additional Director, HOD Zoonosis Division, NCDC. She is co-PI of VRDL at ABVIMS and Dr. RML Hospital. She has been coordinating the national programs for rabies, leptospirosis, and One Health. She is member joint monitoring group under DGHS for emerging and re-emerging infections. She has worked in outbreak field and lab investigation up to molecular characterization in Siliguri, Nipah, and Chicken 2006 to 16. While she is being posted in RML, I remember where I was working as the medical superintendent and director. I know that how much hard work she has been doing. Whenever we used to call any help from microbiology point of view, we cannot call upon anybody else than Dr. Mala Chabra. She has basically her credit to 17 manuals and 6 chapters. I remember she was the person who was instrumental in diagnosing Zika, the first case of Zika in Delhi. And as a friend, as a family, she has been associated since very long. We share the alma mater from Rohtak. I really look forward that today's talk by Dr. Mala will be definitely eye-opening for you and will be of use for the younger people, that what is the role of microbiologists in the outbreak investigation. Particularly, we have all seen that what havoc an outbreak can do from epidemic to pandemic. Dr. Mala Chabra. Thank you.
Good afternoon, everyone. And at the outset, I would like to thank the organizers for giving me this opportunity, the subject which is very close to my heart, and special mention and thanks to Dr. Sherwal, the chairperson of this session. As I know him personally and professionally, and he's been the medical superintendent of Dr. RML Hospital and Atal Bihari Vajpayee Institute of Medical Sciences, and has been source of inspiration for many aspects and subjects that I've dealt with in my career. I also thank the co-chair who we consult at times when we are in difficulty, and he guides us in various IPC practices, specifically when I have sought help from him. So thank you. And role of microbiologist in outbreak investigations and disease surveillance. I'll mainly focus on outbreak investigations. And I always wondered when these kind of things were shown to me, they used to come on my table. I worked at, as Sir said, NCDC for 18 years in the zoonosis division, headed the zoonosis division. Nipah virus hits India again. Health worker succumbs to Nipah. Is it something to do with me? Am I trained in this? Was always the question that came to me. Scrub typhus made headlines in all the newspapers in Delhi and other places. This was from media scanning at IDSP all over the country. Should I be concerned? Should I be doing something about it? Do I have any role to play in this? Heading the zoonosis division, this was it. One morning, I got this picture in my email box in 2011 with this picture asking me, ki, Madam, kya kare? Because I was in zoonosis division, I had, responsibility of both animal and human sector. And, I didn't know what to do because it now deals in coordination. So it really tickled my thought. Another one, that what is the role of microbiologist? And should we now, is it time that we get out of the lab and go to the field to prove ourselves? Our training, by and large, is that of laboratory. But our perspective is much wider. We can play a huge role in public health, and let me tell you, clinical or medical microbiology or clinical microbiology and public health microbiology are not segregated. What we are doing is, when we put in the data somewhere and help in policy making and decision making, it is public health microbiology. When we go out to investigate a case, an outbreak of infectious disease, it is public health microbiology. So there is no dividing line to that, right? So, after a lot of thought, what is the role that a microbiologist can play in clinic, including everything? So public health is a larger or broader umbrella which covers the lab as well as the field. So diagnostic laboratory investigations, we all know, we are trained for that. But specifically, when you go in field, then sample collection, storage and transportation is your responsibility. What lab tests would be done is your responsibility. Epilab investigations in surveillance and outbreak investigation needs whole input in microbiology of a microbiologist. I will just highlight each of these points as we go down in my presentation. Biorisk management, biosafety and biosecurity. I am talking about both human and animal health sector. So, human health sector, do you understand what biosafety and biosecurity is? So, if I have to put it very simply, biosafety is, protecting yourself from dangerous organisms. Right? You're protecting personnel from dangerous organisms. And what is biosecurity? You're protecting organisms from dangerous people so that they are not misused. That is also the responsibility of the microbiologist. Quality management, all of us understand that. I will elaborate on that as I go along. Applied microbiology research. Training and teaching, like what we are doing now, and management and communication. I will touch upon all these in my subsequent slides. So lab investigation in public health is, what we have to do is, we know our principles, but what is going to be more applicable while we are investigating an outbreak becomes very important. Let me give you an example of JE. Japanese encephalitis. 2006, we were doing, we used to get the kits from National Institute of Virology, Pune, Japanese encephalitis kits, and we received samples from all over the country. Suddenly the kits would, at the end of the expiry, we would collect samples from the hospitals of undifferentiated fever and put the test. That time it was not an organized system, 2006 I'm talking about. And we used to just feel that, okay, let the kit not go waste, let us put up the tests. And suddenly we got a JE positive. So, do you think we can release the report from the laboratory? Positive JE in Delhi. I'm sure you would understand that you cannot just release the report because you have to find an epidemiological link to it. So I was at a privileged position at NCDC, so we activated our entomology division and epidemiology division to investigate that. They went into the field and they found the mosquito, they found the virus in the mosquito and found the reservoir host, and we could release the report. Similarly, scrub typhus. Suddenly, the early warning signal was generated from the lab. We were testing samples not to waste the kit, so we wanted to, do justice to the kit at that time, that we are not wasting the kit. We tested pyrexia of unknown origin, and we found scrub typhus in one of the samples, and we traced the sample to one of the private hospitals, where we realized that this patient is very sick on ventilator. And when we told them that this is what it is, you should start doxycycline, they did. But we did not release the report till we did the EpiLab investigation complete. So we did entomology investigations, we did epidemiology investigations, and we found that that was the time when yoga was being popularized. And this is a very prominent place in South Delhi, where they made huge parks, and they cleared the scrub bushes from there. So there were a lot of mite eye lines, and people had that notion of walking in the parks barefoot. कि नंगे पैर चलेंगे तो आंखें ठीक रहेंगी, सेहत के लिए अच्छा है. And in turn, they got bitten by the mites and developed scrub typhus. And that is how we realized that when we started testing, when we, scrub typhus, high degree of clinical suspicion, appropriate and timely lab diagnosis, and appropriate institution of treatment immediately removes patient from ventilator and decreases mortality and morbidity. So laboratory also gives signals that we have to pick up, and that is part of the public health. Assess risk to respond to potential health threats. That is biosafety levels. Who would do it? Which organism belongs to biosafety level 2, 3, or 4? Now, this again, an organism here in our country may be of much lower biosafety level than that in US. So you cannot take it for granted that all the organisms that US lists as biosafety 4 or biosafety 3 are biosafety 3 for us also, because it depends on endemicity of the disease. So it is the role of microbiologists to do that. Recognize the specific issues with use of laboratory and epidemiological methods in investigation of rare and emerging disease. Precisely this. Now, Nipah virus, Crimean Congo hemorrhagic virus, Ebola virus, we've tested all, but we have to be careful about where we can test these organisms, where we can test anthrax. Now, US anthrax is on high biosafety level, whereas we can do anthrax. Anthrax is endemic in our country. So, but for opening the culture, other things can easily be done in any BSL-2 laboratory. Can somebody help to move the slides, please? Okay. The development of microbiological case definition. So when you go for an outbreak investigation, you must have seen the IDSP data. When you are putting the data in the records, you have 3 kinds of definition. Probable definition, which is more of symptom-based, it's more of a clinical definition. But suspect and confirmed definition definitely needs inputs from microbiologists. When do we say a definition is suspect? We link the clinical symptoms to a screening test. It is not a confirmatory test. And so we have to decide which is the screening test in what disease. And if we see confirmatory definition, it is based on the confirmatory test. So microbiologists give an input in that. Then sampling strategies. What should be the sampling strategy in any outbreak? What is the adequate number of samples to be tested, and what is the safe way of collecting those samples? I think COVID-19 has taught us this, and I need not elaborate on this, but I want to mention that here we have to broaden our perspective in not only collecting human samples, but also animal samples and environmental samples. I will talk about One Health approach in investigating outbreaks of animal origin or vector-borne diseases that needs that One Health approach. So we have to broaden our perspective and not close our laboratories to other samples testing. Then incident response team coordination. Always when you investigate an outbreak, there will be an incident response team. There we have to collaborate with them. Now coming to outbreak investigations, all this will be well understood if we understand how to undertake an outbreak. Now, when I was talking in one of the sessions with the IAMM organization, I asked them that why don't we teach outbreak investigation in our course? Because that is the main point, that people do not know how to investigate the outbreak. I was recently in Rajasthan when we were speaking. Now, Rajasthan, Dr. Bharti Malhotra is here. She is a pro One Health person, and she gives a lot of training to people. Despite that, there were other people, when they were asked to investigate the outbreak of scrub typhus, they immediately asked, हमें करना क्या है? आप बोल तो रहे हो माइक्रोबायोलॉजी को, हमें करना क्या है? So, करना क्या है is very important that we have to learn. So, outbreak investigation is a very complex subject which involves multi-stage process with the multidisciplinary team, which includes public health officials, epidemiologists, microbiologists, and healthcare professionals. And idea is to coordinate with one another and come to a good investigations. Let's understand the steps of outbreak investigations to understand the role of microbiology in all these steps. So, identification and confirmation. So, how is outbreak confirmed? Usually the outbreak is confirmed by number of cases of specific disease or syndrome when there is unusual rise in number. Or there is new finding in lab, as I've just said for JE and scrub typhus. But always remember, I forgot to put that slogan, that all outbreaks will start, we call it an outbreak, the starting point is the laboratory. And when we finish the outbreak, the end point is also the laboratory. Laboratory will tell you whether the outbreak has ended or not. So that is the role of microbiology. So first is case identification, then you formulate a case definition. So what is the case definition? It is, what constitutes a case. So you have to, and this will help you to do consistency in reporting linked to a confirmed case or to a geographical area. I'll give you an example. Recently, I went for an outbreak investigation of Nipah virus disease in Kerala. And there, when we formulated a case definition said, now this time in Nipah virus in Kerala, there were a lot of respiratory symptoms. So we formulated a case definition as fever with respiratory symptoms. Now, you see, everybody, there was an outbreak of influenza going on there. So everybody was rushing to hospital. So we realized that we have to quickly link it to the contact with the confirmed case and geographical area where the patient resided to get a substantial and a number that is really required that need to be tested. Unnecessary testing of other cases, we will miss out the other tests. COVID-19, for example, initial case definition in March 2020 was people coming from abroad with fever and upper respiratory symptoms. Later, the case definitions was modified when the first case was detected in January of 2020. In March of 2020, that fever with respiratory symptoms coming from those areas plus contact with this case. When the disease starts spreading, we attached the case definition to containment zones. And subsequently, when it became widespread, we left those containment zones and we started testing all the fever cases with upper respiratory symptoms. So this is how the case definition involves, and at every step, the input from microbiologists was required. Then we derive information from area, whatever information is available from the outbreak area, and start the surveillance. Surveillance can be both active surveillance, which we usually start during the outbreak. And what is active surveillance? That you actively ask people to come and get themselves tested based on your case definition. That is contact tracing and testing. Again, what test needs to be done, how it needs to be done, needs microbiological inputs. Then data collection of all the affected individuals, including demographics, symptoms, and potential source of exposure, becomes important part of the surveillance, including here the reports that are generated, because now your reports and contact tracing reports have to be matched to see what is the effect and how the outbreak is spreading. Then you generate a hypothesis, and hypothesis is regarding the source of the outbreak and how the disease is spreading. Here again, microbiology input will be very important. Now coming to investigation, after we interview people, there again, while interviewing people, microbiologists are required to go to the field. Why? Because when you get a full perspective of how the disease transmission is occur, you will be able to decide what is the test required and how do you interpret the results. And this also holds good for animal and environmental investigation. Then sample collection in the field becomes very important for biosafety concerns. So we are collecting biological samples, how to store the samples, what temperature are you going to keep those samples, for how long, what is the sample size that you are looking at. So in an outbreak, when we do outbreak investigation, it is not necessary, for example, dengue outbreak, that you test all the samples. So representative samples would be sufficient. Then triple packaging and where to send the samples, whether they are state labs or you have to send it to the central lab. Now this also becomes very important because it is such a challenge to identify labs in the field. So you have to have your contacts right as to know where the samples are going to go depending on what you are suspecting in an outbreak. This becomes more difficult when your outbreak is from the unknown etiology. I'll give you an example here. 2006 again, chikungunya outbreak. No, when I was sent to Malegaon, there we just saw people coming with fever and bending and high-grade fever, joint pains. We just were collecting samples. We didn't know where we are going to take the samples. But we collected the samples as acute samples, marked the date of onset, and brought it to Delhi. Then we heard from Madagascar that it is something called chikungunya and that is spreading. Could it be chikungunya? We looked at our library in NCDC, 1964, the entire outbreak was investigated at NCDC and they had facilities even for isolating the virus that time in mosquitoes. That hint gave us that we should start isolating the virus in C6, 36 cell lines which were there with us, and we were successful in that. Subsequently, National Institute of Virology, Pune, and we got the kits with the help of WHO and CDC at that time come in, and we tested those samples and they turned out to be positive. So that kind of linkage and thought process should go in while you are investigating the outbreak. Then, how do you transport? Now, the biggest challenge, it is easier to say here, but when you have to transport the samples from the field to any other lab, you will not find. Do you know any couriers who will take biological samples as per the IATA guidelines? So it is very important as part of the preparedness that we have a courier service in place so that we can transport the samples properly to the lab which is going to test, to the appropriate lab. And this has to be done after informing that lab so that they are ready to receive the samples and they don't spoil your samples on the way when a person does not know what to do with the samples. So with prior information, you can
Mala, request to
expedite. Okay, sir. So those were sample collection. Then lab testing, of course, I need not say. Identification of organisms is primary responsibility of microbiology, and pathogen characterization, be it by serotyping, biotyping, AST, or molecular methods. And what example do I give? Then the super work done by INSACOG, the consortium of 64 labs, which identified various strains and related it to the clinical outcome of the disease. Then after confirmation of source of the outbreak, that you can only do when you go in field. Like this time, Nipah virus outbreak, we went and saw the bat colonies roosting there. And we exactly knew what fruit samples have to be collected and what other epidemiological samples would be significant. So no point collecting water samples there or any other samples except for eaten fruit of the bat. Similarly, control measures, COVID pandemic has taught us that even while isolation and quarantine, microbiological inputs become very, very important, and we participated in treatment management guidelines of COVID-19. Then vaccination strategy. Microbiology becomes very, very important when you decide vaccination strategy. I will give you an example of meningococcal meningitis. Now, Delhi experiences meningococcal. We've just analyzed the data every 20 years. Now, it is about 20 years. Should we give vaccination to all concerned? Why are we not giving meningococcal vaccine to everybody, all the children that under UPI? So such decisions have to be taken because this vaccine is very expensive. What type of vaccine is it? How long would it induce the immunity? Those kind of considerations will come from the microbiology to help develop the policy. Then communication. Once you are in a field, communicating with the public, with the officials and media becomes important, and subsequent steps are monitoring and evaluation. How do you declare the end of the outbreak is at least 2 weeks after the last case has been reported from that area. Document the report and then also highlight what are the lessons learned. Because once you highlight the lessons learned, only then you will be able to investigate future outbreaks in a better way. Always there will be shortcomings once you are investigating the outbreak and you improve, but you tend to get into another outbreaks before you get outbreak of a similar organism. So lessons learned. So we participated when Ebola came in, though the outbreak never occurred, but our team, the preparedness was always there, and we were in pre-epidemic phase for Ebola, whether it was making guidelines for the healthcare workers, or the airline officials, or screening at the airports, or scanning at the airports and collecting samples from the people who are coming from the affected states, and follow-up of the Ebola patient. This I want to mention that we detected one positive case in semen who was a recovered case of Ebola. And then we continuously followed it up, collected his semen samples every month till it became negative because he was a young individual, we could not let him go. Now that time, WHO said that if a person is positive, he should abstain from sexual behavior for up to 3 months. But we detected Ebola virus antigen in semen, not in blood, in semen for about 5 months, and he was quarantined for 5 months. And then finally the international guidelines were also changed. So in pandemic, role of microbiologist is well known in diagnostic testing. What about so many 3,000 odd laboratories participating, kit validation, quality assurance and standardization, development of diagnostic kits by ICMR and ICR, and mind you, these kits worked for both animals and human pathogens. Pathogen identification and characterization, I have already spoken of INSAGOG, policy development, capacity building, public health awareness campaigns, all of us participated in that, and most important, infection prevention control practices, be it PPE, disinfection, or any other. And of course, operational research, pathogenicity, epi-lab research, etc. And it is very important to collaborate with clinicians and public health officials. Timeliness of reports and timeliness of communication makes all the difference. One Health and microbiology, I want to say that microbes are same, sectors are divided. Same infections are caused in animals and humans, and when the interface decreases, the disease gets transmitted to humans. So this is best understood by microbiologists. Effective coordination with mutual respect with epidemiologists, veterinary experts, wildlife experts, ecologists and epidemiologists will give us that approach to prevent and control zoonotic disease infections in our country. IPC, huge role, whether we make technical specification, when there was shortage of kits, PPE kits, we designed alternate methodologies to use to prevent infection transmission. Various disinfections, I will highlight one particular thing about the disinfection tunnels that came up. You know, everybody was so wanted to put a disinfection tunnel in the hospital. And we microbiologists stood up to it that there is no point spraying human beings with disinfection. Everybody else, even clinical colleagues, wanted that disinfection tunnels to come up because they did not properly understand what disinfection is used for. So role of microbiology is very important. And last, I think, slide, management and communication. Now, when you are going in field for outbreak investigation, it is very important. You are in a now Nipah virus, I was the team lead. When the team was sent to the Italy, I was the team lead to fetch these students of Corona. So you have a multidisciplinary team and you have to manage it very well. You have to manage with state administrations, that includes, you know, ministers, bureaucrats, state officials, your own teammates because they are from multiple disciplines, other teams that are working in the field, field workers, and general community. And on the other hand, whoever has sent you from central government, you have to manage with them also. Central administration, central officials, central incident response team, and lab team. And of course, media management becomes very important. So it is really an art, and you have to communicate at right time, at right level to come to it. So with that, I thank you. And it is time to get out of the lab and come to the field for giving your full potential and inputs for outbreak investigation and make a dent in the society for a betterment that we do in microbiology. Thank you so much for your patience listening. Thank you, sir.
Thank you, Dr. Mala. May I request you to join the dais? We will take question together. Now I call upon the next speaker, Dr. Mayank Dwivedi, who is working as a public health specialist with CDC.
Sir, in the interest of time, let's skip introduction.
I understand, but I just wanted to tell people that he is medically born in the same institute, KGMU. He is a product of this institute. His medical training has been in KGMU and in SPGI. So that is very, very important. A graduate and postgraduate student from KGMU and SPGI Lucknow is with us and going to talk.
Thank you, everyone. Thank you, sir. So I am here to tell you stories from outbreak investigations, and it's fine? Okay, thank you. So first of all, just wanted to tell you, as Dr. Mala already mentioned, there is difference between public health and clinical medicine, and then difference between public health microbiology and clinical microbiology. And this stems from difference between public health and clinical medicine. In clinical medicine, we are concerned about the care of individual patient, while in public health, we are looking we are trying to improve the health of community, the health of populations. I'll give you two examples just to make this distinction clear. Though these are interrelated, but these are very distinct disciplines. First is accidents happening at Agra-Mathura highway. So thing is, so some of these examples may go outside the confines of laboratory. We are very much comfortable and we are very much familiar with the confines and certainties of our laboratories. Yet, when an outbreak unfolds, our role expands beyond these boundaries. And we find ourselves navigating complex situations, mostly without any support. And there we have to use our skills to uncover vital information that can save lives. So I'm sharing stories from that. First is Agra-Mathura highway. This has nothing to do with lab, but we investigated. Our team investigated it. What is the approach of clinical medicine for these accidents? They will try to improve facilities of hospitals, try to provide care to more individuals, care to injured persons, improvise their ICU facilities, treatment facilities, rehabilitation facilities. But public health has a broader perspective. In public health, we try to prevent the occurrence of certain diseases, outbreaks, these situations like this. So we investigated it. There were two issues. We found there were two issues. One was there was no rate limit, speed limit. And then the road was, and the major cause of death was bursting of tires. In Agra, Mathura highway, the major cause of accident was bursting of tire. And this resulted from two things. First was high speed. Second was road. Road was made of cement rather than the coal tar or bitumen. So that in the cemented road, its friction is very high. And because of friction, tire become very hot and old tires burst. And when you are driving at the speed of 140, 160, you cannot control the speed and direction if the tire bursts. So that was a major cause of accident. And what did we do? We provided two recommendations. First, there should be a speed limit. Now you can see there is a speed limit. And the second was road should be made of softer material. So other roads are being now developed with that material. The second example is Salmonella outbreak. We detected an outbreak, a major outbreak of Salmonella in a district. I'm not going to name the district, but we detected an outbreak. And when we reached there, it was about 7 to 10 days. 7 to 10 days happened, passed, and we could not find much. The only important finding was that they had a common water supply. We investigated, we tested water rigorously, we couldn't find anything there. So we went back, we reviewed the water treatment facility. We reviewed their log, their practices. We interviewed staff there, and we could found that there was a breach. About 2 weeks ago, there was a breach in the water treatment facility. And for 3 to 4 days, they were supplying the untreated water to the community, and that was enough. That bolus of Salmonella typhi was enough to cause an outbreak. So we prepared the report and shared with district magistrate and tried to ensure that this kind of things never happen again. So this was second example. Just wanted to tell you that because of you guys, epidemiology is changing. This is one example, KFD, Kyasanar forest disease. When I was a student in KGMU, I used to study this, and this was limited to only one district. We were told that this disease was confined to only one district of Karnataka, that was Shimoga district. And all the epidemiology was around that developed, all the hypothesis was developed around that. Now, with the increase in testing capacity, we know that this disease happens all across the Western Ghats. Why is this something we should? Is it something strange? No, the amplifier host is monkey. You cannot control the movement of monkey. These monkeys move all and roam all across the jungles of Western Ghat, and they carry diseases. So you are responsible for changes. This change in epidemiology. Leptospirosis. Again, that time we knew that it was a disease of coastal region. Now we know that it's not a disease confined to coastal region. It's all across India. Scrub typhus. Now we know that this is not restricted to certain terrain. It's widely prevalent across India, and we know that this is an important cause of acute encephalitis syndrome. I'm not going into details of outbreaks. I wanted to discuss this thing, how to differentiate outbreak from pseudo-outbreaks. Not every increase in number of cases is outbreak. There are various causes of increase in number of cases. Outbreak is just one of those causes. I want to discuss this. This just important example. Sometimes you get you get a report of pseudo outbreak, or sometimes when you analyze data, what they did in Spain, they analyzed the data of pneumococcal meningitis for along 20 years, and they found that after 2000, there was significant increase in number of cases. If you can see the size of bars. Size of bars indicate the number of pneumococcal meningitis, and you can see there was significant increase, but that was not because of outbreak. That was because they just introduced one rapid diagnostic test kit. They they started testing for pneumococcal antigen in urine, and that was non-invasive test. And that increased the number of cases. So that's example of pseudo outbreak. Labs. So you know what we find in labs. What the positive cases are just tip of iceberg. Only few patients reach to the lab, and among those, only few are tested positive. So every positive case may be a tip of iceberg, and this is our responsibility. That our long-term goal is to set up laboratory-based surveillance where labs identify outbreak. They give early warning signal. There is likelihood of outbreak. Just go and see. If you guys start calling district surveillance officer and tell that, sir, there may be an outbreak, we have made our line list. This got froze. Sorry, this is line list. This is what is line list? In line list, one row represent one case. Just simple demographic findings like name, age, gender, address. And if you see there are 4 positive salmonella cases from a particular region or area, you can just call district surveillance officer, sir, there is likely an outbreak. That's our goal. There is likely an outbreak of cholera or salmonella, typhoid. I'm not going into detail of this. Not this. This is my favorite, but I'll skip. So I just wanted to tell you that environmental testing is not usually very helpful. We don't encourage. We want labs to test samples of patients, not environment. Whether it is routine culture, routine surveillance culture, water or food, we don't want labs to test these unless there are specific situations, because mostly we don't get etiological agent here. We get etiological agent in actual clinical specimen. Suppose if you find salmonella or E. coli in water, what will it indicate? Nothing. It will only indicate that water is contaminated. But what agent caused infection, this water or food won't provide that information. I'll give you, I'm just showing you a report of a state reference laboratory, food laboratory, and what they reported. They tested sabji and some of the few items, and they reported certain harmful bacteria grown in those samples. Hence, these samples are unfit for consumption. I don't know what harmful bacteria are, but we discourage this kind of reporting. Another example. This is an outbreak of acute. This was I think Anup is here. Dr. Anup Velayudhan, he's with ICMR, he's scientist D. He investigated this outbreak. Actually, this outbreak happened twice. One of them was investigated by Dr. Anup when he was pursuing the Epidemic Intelligence Service with CDC and CDC. It's a joint course that we run in India. So around 20% in a district of Tamil Nadu, that's Kadalur district, reported with complaints of vomiting and dizziness. Dizziness is kind of faint feeling after eating a common meal. I'm not going into detail because of lack of time, but we could, based on his work, we could identify that this was a shellfish poisoning. And what is shellfish? We read about shellfish. Shellfish is a bivalve mollusk. They are filter feeders. Oysters, clams, mussels, cockles, they are they are shellfish, and they are filter feeders. What they filter? They filter dinoflagellates, dinoflagellate algae, blue-green algae, and these algae contain heat-resistance toxins. These toxins get concentrated in shellfish, and these are heat-resistant toxins. They don't get destroyed even if you boil the shellfish for 2.5 hours, and they can cause different types of poisoning: diuretic, neurotoxic, paralytic, amnestic, that is loss of memory. And there is not much you can do, but there are signs. We developed alert system. There are signs based on which you can identify, you can tell when algal bloom is going to happen. What is algal bloom? When number of dinoflagellate algae grow out of proportion. That is algal bloom. And that time, these shellfish, these bivalve mollusks have very high concentration of toxins, and there are signs. What are those signs? Discoloration of water. These are colored, these blue-green algae are colored, so water get colored, maybe blue, green, red, brown. So discoloration of water is one indication. Fish kill is another indication. They consume oxygen, they consume oxygen dissolved in water, and now fishes don't get oxygen. They start dying. And when fishes die, their protein, their meat, their flesh putrefies, and that further consumes oxygen, and then more fishes die. So this is a downward spiral known as fish kill. Then there is respiratory irritation syndrome. The people living in coastal area, they develop irritation because these dinoflagellate algae have smaller walls, very thin walls, and they release toxins in air. You inhale, and you develop pharyngitis, and you because of contact of those toxins, you can develop conjunctivitis also. So what we did, we are microbiologists, we are lab scientists, we need to do something. So we developed a method by which you can give early warning signals of algal bloom. So cell counting, by counting cells present in seawater, you can tell that algal bloom has happened or is about to happen. So if there are more than 5,000 dinoflagellate algae or more than 5,000 cells per liter of seawater, it's indication of algal bloom. Another example, we started 12 minutes late, maybe 15 minutes late. I'll take 5 more minutes. Another example is outbreak, food poisoning outbreak I won't say food poisoning, an outbreak of diarrhea and vomiting in political rally participants in Cuddalore. How many of you have attended political rally, and do you know they give money? So I don't know what is the current rate. We used to get 50 rupees. So they attended on 23rd July, 55 individuals attended. They also arranged buses and food. So 55 individuals attended the rally, and on 23rd and 24th and 25th, the later half of 24th and 25th, we got 42 individuals in hospitals with complaints of diarrhea and vomiting. These are the findings. Median incubation period, little more than 10 hours. Most common presenting symptom was diarrhea followed by vomiting. No sign of invasiveness, no fever, no dysentery. In fact, there were no blood cells, no WBCs in stool. Recovered, and these all recovered very fast without giving antibiotics. Epidemiological investigation done by our AIS officers suggested role of lemon rice. We suspected Bacillus cereus. But the district lab isolated and identified 4 strains of Shigella flexneri, which were confirmed by the state reference lab. So these are the findings of epidemiological investigation. I'm not going into detail of that, but I just wanted to show you the risk, relative risk. When they consume, they prepared food and they ate the same food again and again. In the morning, on 23rd July afternoon when they ate that, you can see the last column. Relative risk more than 1 is the indication that that particular food was responsible for food poisoning. There was nothing, you can see, so there is nothing more than 1. In the evening, when they consumed the same food in the evening, risk ratio, see the risk ratio of lemon rice, it was 2.1. Means it was likely cause of food poisoning. The same rice was consumed by family members of one of the Gram Pradhan next day, and see what was the relative risk among those. So definitely this was lemon rice was was responsible for this food poisoning. It was a food poisoning outbreak, but what to do? Lab isolated Shigella flexneri from 4 samples, and they serotyped it. So points in favor of Shigella flexneri, none. Incubation period too short. The Shigella flexneri or any invasive bacteria needs to establish itself in the intestine, then it will produce symptom. It was too short. Sorry, it's not moving. It was non-invasive. No sign of invasiveness. We couldn't find anything in the stool. It's working now. No WBC, no RBC, no sign of dysentery, fever, anything, and no sign of invasiveness. And stool findings were non-suggestive. I mentioned they recovered fast without administration of antibiotics. That doesn't happen with Shigella usually. And we did environmental investigation. This rice was cooked, and there is a process where they dry it on a cloth on the ground. So that may be likely source of contamination. We know Bacillus cereus is heavily present in the soil. Their spores are present in soil. So I went there and we reviewed the lab results. These 4 isolates were retested. On repeat testing, 2 were indole positive and gas producer. You know there are exceptions with Shigella flexneri, but these 2 exceptions are not found together. So they never happened together, so they were not Shigella. They were E. coli. 3rd was motile on hanging drop examination. 4th was negative on serotyping also. So it was clear that it was E. coli, and I requested the state reference lab to perform the same biochemical typing, to do the same biochemical test that we did in the district lab, and they confirmed that it was E. coli. And basically those isolates were atypical strains of E. coli. So take home message, never perform serotyping without confirming with biochemical reaction. Why? Because they are genetically very similar. If you directly go for serotyping, you may miss out. You may report E. coli as Shigella flexneri or some other Shigella. Thank you very much.
Thank you, Dr. Mayank. Because of the shortage of time, actually, I'll request the audience to interact with the speakers during the tea time if there's any query and question. Because we have already crossed 15 minutes of the session. So with this, I'm really thankful to both of our speakers, Dr. Mala Chhabra and Dr. Mayank Dwivedi. Thank you.
I just wanted to say I'm not doing anything alone. My team is very big, and my fellow Dr. Indranil Roy is responsible for more than 50% of work we do, and rest is done by the team. Richa is here, Anshuman is here. So I think I do only 5% of the work. Rest 95 these guys do, and I take credit. Thank you.
Thank you, sir. We would request our chairpersons to please felicitate our speakers with the mementos. Thank you. Dr. Mala Chhabra, ma'am. Dr. Mayank Trivedi, sir. Thank you, sir. Before starting on the next session, on the screen we have displayed the numbers. Those who are interested in the quiz tomorrow, please contact on these numbers, give your email ID and phone number. Thank you. Our next session, superbugs and antibacterial agents beyond the looking glass. We request our chairpersons to please come up on the stage. Our first chairperson, Professor Kashinath Prasad. He is the former professor and head of department, LGPGI Lucknow, senior consultant and head, microbiology, Apollo Medics Super Speciality Hospital, Lucknow. He has received many distinguished awards like S.K. Naik Award, S.C. Agarwal Oration Award, U.C. Chaturvedi Oration Award, and Endowment Award, Oration Award in 2021. He has more than 240 publications and book chapters more than 10. So please. Our next chairperson, Professor Maitri Bandyopadhyay. Ma'am is professor and head of department at R.G. Kar Medical College, Kolkata. She has received many awards, including Subodh Mitra Medal and A.N. Chakravarti Gold Medal Award. She has nearly 42 publications in various national and international journals. Our next chairperson, Professor Lahari Sakia, who is professor of microbiology at Guwahati Medical College, Guwahati. She has more than 62 publications and 7 completed projects. I would now request our chairpersons to kindly introduce our speakers. Thank you.
So, thanks to the organizing committee for giving us the opportunity to chair this important session on antimicrobials and the surveillance. I think first speaker is Dr. Balaji, and I think Balaji need not any introduction to the microbiology fraternity because we know him very well. He has been involved in many, you can say that many projects, as well as he has been involved in many works, and his main area of interest is sepsis, vaccine preventable invasive bacterial diseases, antimicrobial resistant mechanism surveillance of molecular epidemiology, then vaccine preventable invasive bacterial diseases. He has received so many awards, and he has got more than 400 publications in peer-reviewed journal. So I think Dr. Balaji is an asset to Microbiology Society in India, and he is a good speaker as well, and he will be speaking on new antimicrobials and their utility. I think Dr. Balaji, please.
Good afternoon, everyone. For the next 20 minutes, I would like to talk about the newer antimicrobials laboratory experience. So it's only the in vitro results which I am going to present. And first and foremost is, I am not going to discuss anything, means there are many number of new antimicrobials are available, but what is important and applicable, useful for Indian settings, I would like to present only those antibiotics. So I will be covering therapeutic scope in Indian context, and I'm not talking about the carbapenem sparing agents, not for ESBL treatments. I'm going to talk about treatments for carbapenem resistant organisms. What are all the alternatives available? And what is the resistant mechanism as of now? Currently it's prevailing in India. And what are all colistin sparing choices are available? So among this, recently FDA approved drugs that includes cefiderocol and cefepime taniborbactam, and in phase 3 trial, which will be available in the next 2 to 3 years. It will be approved by FDA, and thereafter it may take few years to reach India. Those drugs include Aztreonam avibactam, cefepime taniborbactam, and cefepime zidibactam. Then in phase 1, etrapenam zidibactam is there. These are all the colistin sparing alternatives which we have. And early phase drugs for Indian utility is, which are all preclinical. It's yet to start with phase 1, but it's more promising. We need to follow up this. One is Aztreonam nacubactam, meropenem zerborbactam. Then apart from IV therapies, there is a desperate need for oral antibiotics, because other than amoxiclav, nothing is oral FDA approved or UCAST or CLSI approved oral antibiotic. Beta-lactam is inhibitor are available. In that place, tebipenem, gipotidan, ceftibutrin plus avibactam. Avibactam now as an IV is getting converted into oral. Then ceftibutrin plus ledaborbactam, ceftibutrin plus zeroborbactams are available. Then apart from this gram-negative, quickly I would like to go through the difficult to treat Staph aureus and streptococcal pneumonia organisms. So for Staph aureus, the sister of linezolid, that is contezolid. Then among the newer generation quinolins, dilafloxacin and levonorfloxacin, and among for streptococcus pneumoniae, omadacycline and nafithromycin, I would like to say. I will briefly go through these slides. It's not moving. Which one? Right, huh? This one. Yes, thanks. So, unique challenging AMR scenario in India and warrants unique solution. That is, whatever the resistant mechanism for the bugs described in North America, Europe, or even in the Asian continent, our neighboring countries may not applicable for India. So each country has got their own bugs and their own resistant mechanism. Think global, act local, they say. So it's very important for you to understand your own prevailing resistant mechanism and tailor the therapy according to that. So that's very important. So given that, I would like to mention here what are all the carbapenem resistant mechanism we see among the most common gram-negative organisms. That is E. coli, Klebsiella, Pseudomonas, Acinetobacter baumannii. In E. coli, you can see NDM is the most predominant resistant mechanism for carbapenem, and it's almost 97%. And very uniquely in India, we do have all the NDM positive, PB3 positive, penicillin binding protein 3 positive. So that limits the newer antibiotic efficiency. That's why it's important to understand about PB3. Moving to Klebsiella pneumoniae, it's a dual enzyme, is the most common, NDM plus OXA48, almost 65%. The remaining are OXA48, 1 or 2 only NDM is there. In addition to the enzymes, porin channel, outer membrane protein 35, 36 and AMP-C are common. And among the Pseudomonas aeruginosa, it's very unique. Please note that unlike in US or in Europe, in Pseudomonas aeruginosa in India, we have resistant mechanism problem with efflux porin by default. In addition to that, third one is Pseudomonas derived cephalosporinase and NDM 95%. So it's a difficult to treat organism. Carbapenem resistant means it's not due to efflux or porin or Pseudomonas derived cephalosporinase. It's mainly due to NDM combined with all three. So it's going very challenging to treat. And last is Acinetobacter baumannii. If it is carbapenem resistant 100%, it's assumed that it's due to OXA23. In addition, it's worldwide. In India, in addition to OXA23, we do see NDM. So both together, dual carbapenemase resistant is due to 60%. So this is another slide which shows you that in different countries, different ESBL rates and different carbapenem resistant rates are there. So it's not only the resistant mechanism, the prevalence is also different, the treatment is also different. Please note that one. So here I would like to take 2 minutes to talk about the penicillin binding protein 3. This is the receptor for carbapenems. So when there is an PB3 insertion, genetic event occurring, then what happens is that meropenem, imipenem, etrapenem, biapenem, they cannot go and bind penicillin binding protein, so it cannot do its function. So this is the problem with the penicillin binding protein 3 insertion, and in India almost all the NDM positive E. coli, that is carbapenemase producing E. coli has got PB3. So because of that, there is a huge limitation with all the novel therapies, which I will be informing you in the subsequent slides. So what is the impact of PB3? It reduces the activity of novel beta-lactam, beta-lactamase inhibitor, and which are all the drugs affected? Loss of activity to ceftazidime avibactam and to some amount aztreonam avibactam, cefiderocol, cefepime taniborbactam. And very important, as I mentioned to you earlier, the NDM, almost all the NDM positive E. coli are positive for PB3. So it's a little difficult to treat. The insertion sequence are 4 different types. The most common is YRIK and YRIN. And to identify this, usually you need to do the PCR, then amplify by Sanger sequencing, and you need to identify. Since it's a little difficult every place to do the Sanger sequencing, we have converted this detection system to MAMA PCR, where you can identify this single nucleotide polymorphism. So this in India, the problem is majorly now with the E. coli, carbapenem resistant E. coli, which is due to NDM, PB3, and this is fine, but if AMPC is there, even more challenging it will be to treat. So now, as of now, carbapenem resistant are either treated with ceftazidime avibactam plus aztreonam or colistin. It's better to use ceftazidime avibactam plus aztreonam, much better than using the colistin. Colistin does a lot of damage, and more important, few points I would like to tell here. There are plenty of problems. First is colistin. It's very impossible to attain 2 microgram per ml in plasma with the normal kidney. In the damaged kidney, it's easy to do, but in normal kidney, it's very difficult. Why it's very difficult to achieve 2 microgram per ml? That is the susceptible rate is, the colistin comes as a prodrug. It has to get converted into active drug. So the time taken to get converted into active drug is slower compared to the active drug getting deactivated or thrown out from the system. So degradation is much faster, but conversion is slower. Due to this imbalance, maintaining 2 microgram of colistin in the blood is very challenging. It's next to impossible in the normal kidney. That's why they have not given susceptible breakpoints. And nephrotoxicity is the other major problem. 30% of the people will have nephrotoxicity by 3rd day, 50% by 5th day, 90% by 9th day in the ICU admitted patients. And it has absolutely very minimal activity with the lung infection, poor penetration, and of course polymyxin doesn't have a role in the urinary concentration, and no susceptible CLSI guideline is there, and IDSI doesn't recommend this drug to be given as a monotherapy. So now, since I said 2 major problems, try to avoid colistin and the problem with the PB3, now I will go for the newer antibiotics. So colistin sparing, carbapenemase resistant, you want to give colistin, but you want to avoid the colistin. What are all the drugs available? Cefiderocol and sulbactam, darolobactam. cefiderocol we have tested in our lab for E. coli. You can see the susceptibility rate is 39.4% according to CLSI guideline and 40% by the FDA breakpoints. So this is as similar to ceftazidime avibactam susceptibility rate. So this doesn't give any advantage. Cefiderocol in Indian settings is not going to give any advantage for E. coli. Whereas when it comes to Klebsiella pneumoniae, its cefiderocol susceptibility is 82%. So cefiderocol works very well for Klebsiella, but not for E. coli, that is due to PB3. And if you look at the MIC distribution, you can see for E. coli, it's close to the breakpoints. Very quickly it will develop resistant. Whereas for Klebsiella, it's spread across and it's not very early, it's going to develop a resistant. So it's better to use for Klebsiella than for E. coli. And this is supported by from various country literatures. You can see here, this is from US, this is from Europe and from Saudi Arabia. Everyone, they had the similar experience like ours. Next slide, please. Okay. Now, what will be the effect of cefiderocol on Pseudomonas aeruginosa? Again, not that great. It's about 60% with the CLSI breakpoint and only 7% with the FDA breakpoint and only 26% with the UCAST. So it's going to be very challenged which one we are going to follow, how we are going to report. So initially, we were very disappointed. Initially, we thought that cefiderocol is going to be a life-saving drug because they said it takes care of all the enzymes. For various reasons, now we can see other than Klebsiella, it's not promising for an Indian setting. The next FDA-approved drug is sulbactam, darolobactam. Darolobactam is essentially an inhibitor of OXA23 and all other beta-lactamases. Usually, the antibiotic here is sulbactam. This is the choice for carbapenem-resistant Acinetobacter baumannii. Darolobactam is a beta-lactamase inhibitor. It inhibits class A, class C and OXA carbapenemases, but no metallo-beta-lactamase. And in the ATTACK trial, they have used this drug along with imipenem, not separately. So it cannot be used as an empirical monotherapy. It should be used only as a combined therapy with colistin or carbapenem. So definitely, other interesting thing is in India, similar to Pseudomonas, here also apart from OXA23, you have NDM in Acinetobacter baumannii. It's almost 60% of the Acinetobacter baumannii in India has got NDM. Sulbactam, darolobactam is specific for only OXA23, not for NDM. So again, it's going to be challenging. I'm really doubtful whether this drug is going to be useful as a monotherapy, as a combined therapy for Acinetobacter, it will have value. But again, it's not as it was promising earlier. It was little disappointing. Now you can treat Acinetobacter baumannii carbapenem resistant with sulbactam, darolobactam plus minocycline. Now I move on to the polymyxin sparing options where you would like to again avoid colistins. 3 drugs are very promising for Indian setting. One is Aztreonam avibactam, Cefepime taniborbactam and Cefepime zidibactam. So look at this Aztreonam avibactam, that's what we are giving. We are giving Aztreonam avibactam, it's not available. You are giving as ceftazidime avibactam plus Aztreonam. It works well. So PB3 limits its activity. In addition to PB3, if AMP-C is there, then it's really difficult to treat. Virtually it will be resistant. So with Aztreonam avibactam, you can see 73% susceptibility in E. coli, whereas in Klebsiella pneumonia, almost 100% it is susceptible. So in E. coli, this is the problem is due to PB3. PB3 is not present in Klebsiella. It's present only in E. coli. So that's the story about the Aztreonam avibactam. Very promising, very good. The only problem is the Aztreonam has to be given simultaneously with the ceftazidime avibactam. Now I move on to Cefepime taniborbactam. Lot of promising, and we were excited to see the result because this is the first metallo-beta-lactamase inhibitor. Because in India, all the 4 escape pathogens has got NDM in it. So this is the desperate inhibitor which we wanted. So, but when we tested at CMC with the Cefepime taniborbactam with the carbapenem-resistant organism, we were quite shocked to see only 12% were susceptible. And for Klebsiella pneumonia, it's good, up to 74% it was susceptible. Again, here the problem for E. coli is penicillin binding protein 3, and the Taniborbactam being a large molecule, it was not working. And not only our studies from elsewhere, from Europe, from China, and from Saudi Arabia also gives the same result. So it's very clear for metallobeta-lactamase producing organism with the PB3, cefiderocol is not working, and cefepime taniborbactam for E. coli, again it's not working. Both are working very well for Klebsiella. Now I move on to the third important drug, cefepime zidibactam. This is not a beta-lactamase inhibitor, it is beta-lactam enhancer. It's a beta-lactam enhancer. So this is supposed to be the game changer or blockbuster molecule or antibiotic. Why we say that is because it's stable to all the beta-lactams. This includes all the ESBLs, carbapenemases, AMPC, everything. Because that's the reason why this seems to be good. This zidibactam binds to PB2, cefepime binds to PB3. So combined binding of PB2, PB3, it's very fast, binds and kills the organism even before the enzyme gets released. So even because it evades the enzyme and gets knocked the organism. So that's how it brings E. coli susceptible, even though NDM PB3 is there. It makes Klebsiella susceptible, NDM oxa K loss is there. And it works against NDM, efflux, sporin and PDC for Pseudomonas aeruginosa. And it also susceptible to ceftazidime, avibactam, ceftolozane, tazobactam, imipenem, relibactam, resistant Pseudomonas aeruginosa. And for Acinetobacter baumannii also it works well. However, this is a laboratory information, it's not a clinical information. The clinical trial is in phase 3 in few countries in the Eastern Europe. So once that comes, then maybe this is one of the life-saving drugs for India and wherever the NDM producing escape pathogens are there. So this is our experience. You can see 100% against NDM and NDM OXA48 dual enzyme producers. Again for Klebsiella, for NDM OXA48 and OXA48 alone. So finally, to sum up, for gram-negative, cefepime zidibactam seems to be a very good drug for E. coli and Klebsiella. Aztreonam avibactam, there is slight limitation with E. coli. If there is no AMPC, definitely it will work well. Cefiderocol, not great for E. coli, better for Klebsiella. Cefepime taniborbactam, it's total disaster. Now I move on to Pseudomonas aeruginosa. Here first I would like to give information on the resistance is redefined. So what is the redefined definition is, the old definition for MDR Pseudomonas aeruginosa was defined in 2008. Out of this 8 class of antibiotics, if 3 class 1 or 2 drugs are resistant, then it is called as MDR. Out of this 8, 3 classes needs to be resistant. Whereas in 2012, what they have said is, MDR is, they have taken off these 3 drugs. Monobactam, that is Aztreonam, fosfomycin and polymyxin. And they said this 6 drugs class, any 3 is resistant, then it is MDR. This is what's said in 2012. In 2022, what they say is difficult to treat. Means if it is resistant to beta-lactams, beta-lactamase inhibitor, carbapenem and fluoroquinolone, then that is called as difficult to treat. So now the meaning Pseudomonas, it's best it is defined as difficult to treat rather than MDR. And what is XDR? Here resistant to colistin sparing agents, not colistin. If colistin is resistant, it's almost like a pan-drug resistant. So XDR means ceftazidime avibactam, ceftolozane tazobactam, imipenem relibactam and fosfomycin. If all these are resistant, then it's called as XDR. And you see in Indian settings, it's quite complex. NDM is in 65% in DTR and OPRD loss, porin loss is up to 60%, hyperreflux up to 35%, and Pseudomonas derived cephalosporinase almost 30%. And you can see the relationship, the dual mechanism through 3 mechanism, four mechanisms contributions here. It's quite complex. If it is DTR, it's not that easy to treat Pseudomonas aeruginosa. And this is where cefepime zidibactam we have tested and we found 100% susceptible. Again, there are many compassionate uses were there, and almost all the compassionate use with the cefepime zidibactam, the patient were recovered. So that's why this is promising for us. Next is polymyxin sparing options. Two important drugs which should be looked for. It's in the preclinical stage. One is Osteonam nacubactam, another one is Meropenem zerborbactam. Osteonam nacubactam, it's only for enterobacterials, not for Pseudomonas and Acinetobacter. We tried with 1:1 and fixed concentration 4. With the fixed concentration 4, we achieved high level of susceptibility as well as the MICs are very low. Whereas with 1:1 concentration, it was around the break point. It's not a good news. For Klebsiella pneumoniae, same story. A fixed dose of 4 nacubactam seems to be much better than the 1:1 ratio. Next is Etrapenum zidibactam. Because etrapenum is a single dose therapy, that zidibactam 2 gram each can knock off all the enzymes, ESBLs, MBLs, OXA48, PB3, everything. So it's an important one, but it won't be useful for Pseudomonas and Acinetobacter pneumoniae. This will be best for outpatient antibiotic therapy, OPAT therapy. And the Indian drug getting evaluated in US. Next slide. Okay, Meropenem zerborbactam. This is my favorite drug. Meropenem plus zerborbactam, cholesterol sparing. Why I said it's most favorite is it inhibits ESBL, it inhibits AMPC, it inhibits KPC, inhibits OXA48, inhibits OXA23 and all other beta-lactamases, metallobeta-lactamases. So whatever the plasmid mediated enzyme we know, it knocks off everything. And also takes care of porin loss and efflux. What is the end result? You can see susceptibility to carbapenem-resistant enterobacterials, carbapenem-resistant KPC, metallobeta-lactamase, and highly susceptible to meropenem-resistant to Acinetobacter baumannii. So there are oral antibiotics which is very important we need to have. One is, sure sir, tebipenem, geopotidine for sexually transmitted disease, ceftibutine avibactam for most of the ESBLs, then ceftibutine levodorbactam and zerborbactam. So with that, I would like to end my talk. Thank you. And I would like to thank Dr. Kamini Walia for ICMR for providing funds to test all these novel antibiotics with Indian resistant isolates. And my team at Vellore, who have been doing all the phenotypic characterization and the genotypic characterization. Thank you.
Okay, thank you very much, Dr. Balaji, for your excellent presentation.
Our next speaker is Dr. Itisha Gupta. She is MD FRC Path, Consultant Medical Microbiologist, UK Health Security Agency and University Hospital, Birmingham. She is also infection prevention and control, cardiac and renal infection. So, introducing Dr. Itisha.
Thank you very much. Thank you very much, chairpersons. And it's a really homecoming for me because I have been a KGMC graduate and I was a postgraduate here. So I really thank the organizing committee for inviting me to speak here and give a talk on this. And then Dr. Prasad, I've worked with him as a senior resident in STPGI, so before I went to UK, so it's really good to see familiar faces. So moving on from gram-negatives and difficult gram-negatives to gram-positive infections. And the way I've structured my talk is to talk about how the MRSA has evolved, and it's always good to know the history when we start to look at the future and managing the future. So the history of MRSA, it is about 70 years old. Thank you. About 70 years old. MRSA was first detected as soon as we had methicillin antibiotic. MRSA was soon detected within a year. And then its prevalence started to increase, and it became 5% prevalence within a decade, and it assumed epidemic proportions in the 80s and the 90s. And there were different clones of MRSA being detected. And you could see how the
the molecular epidemiology evolved for MRSA. Also, you would be able to see that it actually increased, that it's been a very dynamic process. And then the two clones emerged. So as you see, the survival of the fittest, really. The eMRSA 15 and the eMRSA 16 were the commonest types of molecular MRSAs that actually accounted for about 85% of MRSA's bloodstream infections in the late 2007. And then you could have this country-specific MRSA lineages. So like in USA, they had the USA 300 clone. But why are we talking about MRSA? We're all worried about the gram-negatives. Actually, the MRSA is still there. It still causes difficult-to-treat infection. The burden of infection is still quite high, especially in our ICUs. About 25 to sometimes 50% of Staph aureus are MRSAs, and the antibiotics to treat them are few and far between. The MRSA bacteremia, but there is some good news. There has been quite a lot of actually emphasis on managing MRSAs in terms of diagnosis, prevention, and controlling, and that has led to a significant decline in MRSA bloodstream infections, both in US and UK. In fact, in England, the MRSA bacteremia rates fell to about 85% in a decade. So there is a light at the end of the tunnel. And I think, yes, gram-negatives are really causing havoc at this time, but if we work sustained and consistently, we will win that battle also. So why are we then concerned about MRSA? There is a lot of it. It causes poor outcomes. But there is something quite fundamental difference between the MRSA and the gram-negatives. With the MRSA, when we actually pick up, we can pick up the colonization. Colonization is a significant risk factor for subsequent infections, and there are studies which have showed that 29% of people who actually get MRSAs develop MRSA infections. There can be a huge number of MRSA reservoir in the hospitals. There are colonized patients, there are infected patients, and there are also healthcare workers, and that I will talk to later in my slides. There could also be patient environment. So just around the patient, you can have a lot of MRSA. If you actually sample the environment, the bed sheets, the trolley, the drawers, you will see MRSAs there. Therefore, the hand-to-hand hygiene is so important. The transmission of MRSA happens. So how can you actually prevent MRSA infections? Firstly, you have to prevent MRSA acquisitions. And when I say acquisitions, that doesn't mean that it is actually infection. It could just be that someone has become colonized with MRSA. You need to have infection control policies. You have to have MRSA-specific policies because every bug demands a different style of transmission route. So you have to have tailor-made policies for each bug. You also have to have environmental control measures like good cleaning, good decontamination. What also is needed is a good surveillance. And what we need is actually to provide that data to the stakeholder, because there's no point in we collecting the data, producing fancy graphs and charts, and then keeping it. You have to make the data powerful. Give it to the clinicians, give it to the managers of the hospital, and present them with the problems and say, look, how are we going to solve this problem? And then you need to educate. And when we get educated in our medicine is a lifelong education. All bugs keep coming, all new things keep coming. You need to keep yourself educating, and we need to educate our clinical colleagues. So moving on to surveillance, and MRSA actually in gram-positives really offered us a way of finding surveillance and how we can use that surveillance to our benefit and make sure that you actually get past and get some benefit out of it. So surveillance, in a nutshell, just means keeping a watchful eye. You are gathering information for action, but what you need to make sure that the surveillance is targeted. There's a systematic way of doing a surveillance. Why do we want to do surveillance? What action are you going to take when you're doing surveillance? And what aim do you actually feel that you want to achieve when you're doing the surveillance? So for MRSA in perspective, you have to know, you have to do your own risk assessment for developing a surveillance system. You need to know what are your internal infection rates for MRSA. If it is endemic, if you're only getting 1% of your patients coming in who are MRSA, or if there are 50% of patients in your hospitals who are colonized with MRSA. So that kind of surveillance is quite important to develop the future action plan of how you're going to do it. So you need to know your internal infection rates. You also need to know what is your laboratory strength. What is your laboratory detection strategies? How is your environment? What is your infrastructure of your hospital? So some of the hospitals in UK still have Nightingale wards, although very few and far between, but they are still. So those are kind of areas where the transmission can easily happen. How many toilets are actually there in the wards? We don't actually see those things. We neglect them. They are left to the cleaning people. But actually, your best efforts can actually match the cleaning standards of the hospital. So all of those things are really important in preventing the outbreaks. MRSA was important, and it's even more important for the gram-negatives. Can you please move the slide, please? So active surveillance. So in terms of MRSA, one thing is a good thing in that you can develop an active screening program, which either could be a universal screening program or it could be high risk, depending on what is your prevalence rate within the hospital. From universal MRSA screening program, what I mean is every patient that is coming into hospital is being screened for MRSA. So you want to know what is the burden of infection of MRSA that is coming into hospital. And then you can develop your own policy to prevent those patients. What can you do for those patients? And there is something that you can do for MRSA. Then also what you can do is, you can do either a high-risk screening program. So if you don't want to do a universal screening program, suppose you say that the prevalence of MRSA is really low. This is a very high tertiary care center. The prevalence is really low. I don't want to set up a universal screening program. You can then set up a high-risk program. You can say, well, my ICU population is clearly very high-risk population. My dialysis patients are really high-risk. The dialysis patient, I work very closely with the renal infection team, and the dialysis population are very high-risk for Staph aureus infections and MRSA infections. So you can have a screening program for dialysis patients, burns patient, neonatal patients, transplant patients. So you can actually tailor-made according to the specialist that is served within the hospital. Then when you detect that the patient is MRSA positive, so when they mean MRSA positive, they could be colonized with MRSA. So what you want is to decolonize these patients so that they don't go and develop MRSA infections. And that is the aim, and that is where the prevention comes into place. So for decolonization strategies also, similar approach. You can have universal decolonizations, or you can have targeted, or you can combine the two together. So for universal decolonizations, there was a time in UK when in sort of 2010, when the MRSA was really, really rampant, and some of the hospitals approached a universal policy where everyone coming into the hospital was screened, then given decolonization therapy, and then if it was negative, was stopped. So you can then do empirical choices, and you can then stop if it is. In terms of targeted, like still on ICU settings, every patient that comes into ICU settings gets screened, and you can decolonize that patient, and if it becomes negative, you can stop it. So you actually try and define a surveillance strategy, a decolonization and a screening strategy for your own hospital based on your risk assessment. Or you can do a combination of both. So the decolonization then reduces MRSA carriage, and then once the patient's it is not going to eradicate the MRSA. And I know in many years ago, people used to call it eradication therapy. But I think we have to really move out of that word called eradication, because you're not getting rid of MRSA. You are just reducing the bioburden of that MRSA to an undetectable level. And there are times when we actually see these patients who are MRSA positive, then come back into hospital maybe 1 year later, 2 years later. And because we have got electronic systems, we can see these patients have come back. And you test them again, they are negative, and then they become positive. So there is somewhere that actually it is there, but it's just you reduce the burden. And therefore, you have to have an MRSA monitoring program. And the laboratory systems really help into it. You can develop a laboratory alert system. So whatever reports are then in your lab, whatever results are being generated on a daily basis, you can choose. We keep, every 2 years, we keep updating, we keep revising our laboratory alert systems to see which ones of the organisms at this moment are really important. Like last year, we saw a huge wave of group A strep infections in children in November, December, and we really updated our laboratory alert, and daily list that came out, we did a group A alert search from the whole lab. So you can tailor it. This is what the microbiologist load is, because you want to know the results as they are happening before you want to act on the hospital patients. So in MRSA, your main aim is to prevent MRSA, which leads to colonization and infection. Then this is all clinical infection prevention. Coupled with it, what is very important is molecular surveillance, because all these bacteria, it's the genes that are actually causing the problems. You need to know where is the bacterial genotyping, how is the movement. The COVID has proved that so much, how much mutations. Now, some bugs move very quick in terms of genomics. Some bugs are quite static. However, there is still some shift and drift, and you can actually see that how it's moved. For that, you really need good molecular methods. Previously, we used to do pulse fields, MLST typing, SPA typings, used to identify epidemic clones, and whenever there was an outbreak, we would see which outbreak it is causing, how it is spreading, and you could look at the evolutionary dynamics. In recent years, whole genome sequence has come up, where you can define the transmission genomics and of a single clone. So a lot of these previous genotypes and typing had become limited resolutions. They were, discriminatory potential was low, so the whole genome sequence was able to define within them how, whether there was any linkages or not, and they actually can tell us more about the outbreak management and provide good epidata. So in terms of the surveillance for healthcare workers, the healthcare workers interact with patients all the time. So they themselves become colonized, or they can become infected. Sometimes they are the victims of their profession. And we need to know whether they are colonized, whether they are infection. What is actually the evidence? Are they really colonized, or is it that this is just a transient colonization that we are picking them? And there have been several reports of healthcare workers linked to the source of outbreaks, and that they have been implicated. So this is nearly 20 years old. Very few first reports that it started to emerge, where people started to look at healthcare workers screening. And 20 years ago, they just looked at a few 320 healthcare workers and found that about 5.3% of their healthcare workers were actually colonized with MRSA. And some of them actually just transient colonized, of 6 of them. And when they tested them again, they were negative. And 11 of them, they just decolonized and they became negative. And just a couple of them, they had to give systemic antibiotics for them to become negative. But what they didn't do was they didn't link any of this data to any infection outcome. So it was just a snapshot that, yes, healthcare workers can also be MRSA positive. Then there have been 1 or 2 other papers that have been published where people have started to look at a bigger area, about 4-year surveillance, where they found that MRSA colonization rate within the healthcare workers, and then they tested them again, and it fell. But it was done targeted on orthopedic units. So something, because I've been involved for 16 years in infection control, this MRSA is kind of a bread and butter thing that we actually do day in and day out in the hospital. I was involved in, and which I did an oral presentation in America about how we use whole genome sequence to identify the linkage of a healthcare worker in an MRSA outbreak. So as I said, we do MRSA universal screening. Every patient gets screened. Then we also introduced a long-stay repeat MRSA screening. So every 28 days screen, the entire hospital would get screened. So we pick up if there's been any colonizations, any new acquisitions in the hospital. Therefore, we can target them so that they don't develop infections. Coupled with them, we also had an in-house where we could do good serotypings. And we could actually then develop a molecular profile for each MRSA. So we developed a program where we would look up for any acquisitions. So any patients that come up positive or was negative initially became positive, and then we actually did the serotyping. And if we had more than 2 cases on the ward, we would type them to see whether they were linked. And this is how we were able to develop a program of MRSA outbreak, infection, period of increased incidence management in the hospital. So we detected an outbreak where we actually found that there were 3 cases of MRSA on a single stroke ward. And when we typed them together, they were all the same serotype. Then we actually developed another. So we put in place lots of actions, hand hygiene, audits, cleaning, decontamination, and then we had another case, which was also the same serotype. So what we had to do is we then decided that we should now screen the staff. So staff screening is not something you would start to do from initial, but we then started the staff screening. Can you just move the slide, please? Yeah, thank you. Yes, I'm using that. Yeah, thank you. So we then did environmental sampling. They were all negative. My experience with environmental sampling is not great. We did a lot of hand hygiene and MRSA audits. We did the staff screening. We found 5 healthcare workers which were positive for MRSA. 3 of them were the same serotype that was actually in the outbreak serotype. 1 was the serotype that was different, and another one was a different serotype. We decolonized the staff members, and 4 of them became negative very quickly because we have to test them again, and only if they are negative, we stop testing. But if they are just positive in their nose, we will let them work on the ward. But if they are positive on any of the other lesions, so suppose if they've got hand cuts or anything, they have to be negative, 3 negatives before we can allow them on the ward. At that time, 1 healthcare worker, which was positive for a completely different outbreak strain, completely different strain unrelated to the outbreak, had a skin condition. So we restricted the activities of that healthcare worker for about 4 months, and we repeatedly rescreened that. And after 3 months, this healthcare worker became negative, and we allowed this healthcare worker back on the ward. After about a year, we had another MRSA outbreak. We actually detected 7 cases in the first phase of the second outbreak. And this time, the serotyping was exactly the same as the healthcare worker that we had 1 year ago. And we took the healthcare worker out of the clinical role completely. And then we had 2 further cases. Now we were in a dilemma that now the healthcare worker is also not there on the ward. Why are we getting more cases? And is there any environmental reservoir? Where do we need to look at? What else do we need to do? So we worked very closely with the estates department. So we cleared all the filters of the ward and everything. We checked everything. We cleaned all the filter panels and everything on the ward to actually make sure that we've done everything. But we just didn't know where these 2 had come from. What should we do next? So we decided to do whole genome sequencing on that. And that was a really helpful exercise for us. It's a really helpful exercise for us. And what we found was the 2 later cases were actually not related. And there was just published a whole genome sequencing where they had used a neonatal outbreak to investigate a neonatal outbreak and do a whole genome sequence. And what we found was that actually the eye if you could look at, that had over 100 SNP difference to the outbreak strain and with the healthcare worker. So we knew, although the serotyping was same, but the healthcare worker and the new cases were completely different. So we were able to open the ward because there were times when we had closed the ward for screening, for cleaning. For 1 month, we just didn't open the ward. And it was really very high pressure for the patients, stroke patients. Where do they go? We put them in a different ward. We try and do them. We have to speak to the managers, chief exec, board meetings. It's can become quite tricky. So, but we wanted to do that what was the best and safe thing for the patients. We just couldn't get the healthcare worker in. So healthcare worker management can be very challenging. We tried to do a lot of decolonization attempts for these healthcare workers. We tried 3 courses. We kept her off the work completely, but still she was becoming positive.
Sorry, Dr. Ritisha, kindly wind up.
Yeah. And then we actually did the OCI health management, HR, and we did antibiotic treatment, and she was still positive. And we took her off completely, and we put her on a different ward, and she moved house. And after 6 months, she became negative, and we then let her back. So there was a story with a happy ending. There has been more reports with the healthcare workers where people have used whole genome sequence, and that has lately been published in June 23, where they found a PVL positive healthcare worker. So, and I think main important point that I would like to stress for the healthcare workers would be to check for any skin conditions on the ward, and therefore OCI Health is quite important to do that, where you can find MRSA harbors. So whether we should routinely screen, there is no published control data to actually say that we should routinely screen MRSAs, healthcare workers, because there's been no linkage to any infection prevention in routine screening. And also, you sometimes find that the healthcare workers are just the victims. They are also stigmatized, and their careers can be at stake. So both the 2 guidelines from UK and US actually say that you only screen at the time of just outbreak scenarios and when you can do active surveillance. And there are downside of routine screening. It's costly, it's low value, it's time consuming, and there's quite a lot of emotional things attached to it. You can only do targeted surveillance when you need to do, and it is good economical value. If you actually have an outbreak, then you can go and do this. But what is really important is that you need to be able to do genotyping for healthcare workers. I'll stop here because I know. Yeah, thanks.
It's our earnest request to all the participants, kindly stick to your time, please. And thank you, Dr. Itisha, for your elaborative study over MRSA and all the healthcare settings. It's a very conditional approach to the gram-positive organism, and now we are going to the next speaker.
Our next speaker is Dr. Tarinas. She is professor in microbiology and head of the INSERM research team regist and member of the French National Reference Laboratory. And her field of interest is the carbapenem resistance enterobacteriaceae, where she has done many works, including the characterization of that. Oh, he is a male. Sorry, characterization of many clinically relevant carbapenemase according to the several aspects like structure, function, biochemistry, epidemiology, and genetics. He has more than 385 publications and 4 patents. So, and these are published in very high index journal. So now I request Dr. Nas to continue his lecture.
Thank you very much for this very kind introduction. I'm very delighted to be here to talk about an issue that is taking a lot of time in our laboratory, as probably in many laboratories here also in India, which is diagnostics and rapid diagnostics and how it can be useful to tackle AMR. So here are my conflicts of interest. This is probably a slide that you have seen many times, and I don't want to go back on the numbers. You can discuss the numbers. Maybe they're wrong, maybe they're exaggerated. But nevertheless, this study had the merit to really nail down the problem, and since everyone is speaking and showing this kind of slides. What people may know less is that they also proposed some solutions. And among the solutions they have proposed is rapid diagnostics. And this rapid diagnostic is useful for many other points, for increased awareness of the public, for epidemiological, because you need to know what's going on in your hospital, But also, as we heard before, we have now many new drugs coming on the market. Some are already not useful, some will be useful, and some will only be useful on certain bacteria and in certain mechanisms. So these rapid diagnostics are also useful for this. And in France, we use them now for a strategy called search and isolate. We look for patients that are colonized, and whenever they are colonized, we isolate them from the others. So hygiene, infection control measure, it's very important. For therapy also, we know that MBLs are not responding to certain new molecules, so this is also useful for proposed empiric therapy. But not only, we know also, we have known many studies in France and in Europe, in the United States, where they show that rapid diagnostic, even though buying the test may be more expensive than doing traditional identification, yet when you compare it with an outbreak and the management of an outbreak, rapid diagnostic is not more expensive. On the contrary, it's cost saving. And then, of course, the many other factors. In France, whenever you come back from another country, you put on isolation. And this, if you do it with classical microbiology, it takes 48 hours. When you do it with rapid diagnostic, it takes a few hours, and then you can release the patients from the isolation, and releasing the patient from isolations will also allow him to have better care. Here is just to mention also when you deal with blood cultures, and there's a recent study in Italy where they showed that when you do rapid diagnostics, it's useful to implement the right antibiotic therapy, and this has a direct consequence on patient survival. If you do it within 6 hours, you see you have still a high death rate because those bugs are highly drug resistant. But if you wait for 48 hours, you have almost 70% of death rate. So according, it's better like this? Okay. So if you have rapid diagnostic, you get also better answer. Okay. Excuse me. Okay.
The right click.
Okay, the right click. So the best example, as we have heard, is E. coli. For me, E. coli is really a model system because E. coli is our best friend and it's our worst enemy. And as you know, not so long ago, E. coli was multi-susceptible, and now we have bugs that are nearly impossible to treat. And this is a big problem. And of course, carbapenems still are a main tool for therapy. And when we look at this mechanism, we have heard it before, it's now a global problem. It's not the problem of one country. Every country is interconnected, and what happens in one country will happen in another country. So it's really, we have to be together. And the important point is to know the epidemiology in each country. And in India, such as in France, we know what all the delinquents are, and they're always the 5 main carbapenemases: Vim, IMP, KPC, NDM, and OXA48. So those are really the ones that we need to look for importantly. And in India, basically those 5 bugs are also present in Enterobacterales, Pseudomonas, and Acinetobacter. Carbapenem resistance is something that we know for a long time. As soon as carbapenems were introduced into clinics, carbapenem resistance emerged, and this was mainly due to ESBL producing or cephalosporinase producing together with impermeability. This is a problem, of course, for the patients because the bug becomes almost resistant to every antibiotic, and it's becoming very difficult to treat. Nevertheless, these bugs have a high fitness cost, and they don't spread, and you don't see many descriptions of epidemic situations with these bugs. With carbapenemase producers, the situation is different because they come with highly infeodated, highly epidemic clones. They come on plasmids. Plasmid can spread. And often they're very difficult to detect because sometimes the MICs are below the breakpoints, and yet there will be a failure due to carbapenem treatment. So we need tools that will be able to make the distinction between carbapenemase producer and non-carbapenemase producer because the consequence is not the same. We have phenotypic tools, we have molecular tools, we have biochemical tools, and we have lateral flow. We have many different tools. None is perfect. Only what you have to do is to know which one you have to use and which one is best in your setting. And when you compare all these different tools, you see that you have specificity, sensitivity, which are sometimes very good for phenotypic tests. Some are very good, such as the SIM test. But they have all in common one negative aspect. It takes at least 24 to 48 hours to perform. And also, you need cold chain, you need different incubators, which is also sometimes a problem. You have molecular, you have biochemical tests. They are not perfect either. They have one disadvantage, that you need a cold chain. You need -20s to store the antibiotics or the reagents for molecular biology. And they have also drawbacks because they are not specific. And then you have lateral flow immunoassay, which basically summarizes all what we expect. It's rapid, less than 15 minutes in most of the cases. You don't need any cold chain. You don't need any trained personnel. And it's highly specific, highly sensitive, given you work with colonies, of course. And usually it's much cheaper than molecular assays. And the other advantage of lateral flow immunoassay is that it fulfills the criteria that are set up by WHO, which are the assured criteria, which means they are affordable. The price is not too outrageous. It's sensitive, it's specific, it's user-friendly. You don't need any trained personnel to do the testing. The testing, it's very, very easy. And you basically take one colony, you put it in your buffer, and then you put 2 or 3 drops of the buffer on the strip. And if it's positive, in 2 minutes, you have the results. You have to wait 15 minutes to be able to tell it's negative. And then, so it's rapid. You don't need any equipment. Basically, everything is in the kit. And it's deliverable, which means you can keep it at room temperature on your shelf for almost 24 months. So there is no need of cold chain and no need of electricity. So this is what WHO has set up. The principle is very easy. You have 2 antibodies, one which is on the strip, one which is not fixed on the strip, but which is labeled with colloidal gold. And if an antigen is present, you will have a sandwich that is built on the strip upon migration. And then you basically will just analyze the band that appears on the strip. And as I said, if it's positive, usually in 2 minutes you get a signal. If it's negative, you have to wait for 15 minutes. But it's very, 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 very rapid. And of course, for those who want or are keen on quality, you can also do some automatic reading, and you can take pictures of the reading. We have evaluated, especially the CARBA5 at the National Reference Center, and we basically took the isolates representing the French epidemiology. It was more than 3,000 isolates that we have compared, and we looked what we would have identified. And as you see, sensitivity, specificity are nearly 100%. Of course, you will miss some just because the assay is not detecting them. Such we have a few Proteus mirabilis that produce OXA23. We won't see them because we just don't detect them with the assay. And we had one OXA48 producer that was not a carbapenemase producer anymore. It was more an ESBL. And this one, we detected it, but nevertheless, it was an OXA48. So very good data. And also lateral flow immunoassay, it's an assay that can easily be improved, can be easily modified, and we can add other targets. And there are different assays that are currently being developed, and there should be soon, at least as research use only, available, which are for VAN-A, VAN-B detection, for oxacarbapenemases in Acinetobacter, for other ESBLs, for other plasmid-encoded MCs, and so on and so forth. There are many different assays that can be developed. So we wanted to know whether this assay is also feasible, suitable for Pseudomonas and Acinetobacter, and we did a first study. And in this study, it turned out that for Pseudomonas, we missed about 15% of the carbapenemase producers. And among these 15, we had some IMPs that were missed. So this led us to develop an assay, a novel assay, which is now the one that is currently available here in India as well, which incorporated all the IMPs that can be detected. And this is a main advantage, especially over PCR, such as the ones from Cepheid, the GenExpert, which doesn't detect IMP2 variants. And IMP2 variants are quite frequent in Southeast Asia, and I've seen also here in India. So now we have all the IMPs, but yet we didn't have 100% in Pseudomonas because we missed the guess. So we developed another assay that detects guess carbapenemases. And by using guess carbapenemase together with the CARBA5, we have now nearly 100% detection of Pseudomonas. And with Acinetobacter, unfortunately, we had only 12% of detection because basically we detected only NDM. So this led us also to develop another assay which detects at this time all the carbapenemases that are encountered in Acinetobacter, OXA23, OXA40, OXA58, and we also added NDMs. And by doing this, we have 100% detection of carbapenemases in Acinetobacter in France, and which is probably the case in many, many countries. And this lateral flow immunoassay are now included in the French recommendation of detection of carbapenemesis, which is whenever you have a reduced susceptibility to carbapenems on an antibiogram, you do rapid testing. If it's positive, you have a carbapenem. If it's negative, but the antibiogram is still compatible, you do biochemical testing. And the two together, you have now nearly 100% of detection. So can we do more? Can we use those lateral flow immunoassays directly on clinical samples? Because this is very important to know. So basically, the samples we have tested is urine, blood, and rectal swabs. Of course, when you spread the urine samples on plates, you get colonies. So you can do it on colonies, you can do it on the antibiogram of the blood culture, and you can do it also on rectal swabs that are plate and chromogenic media. Basically, it takes still 24 hours. Can we do more? Can we use these assays directly on urine samples, directly on positive blood culture, and direct on rectal swabs? So the first thing we tried is on blood cultures, because we know on blood cultures you have at least 10 to the 8 bacteria per ml, so you have a lot of bacteria, so the assay should work. The first step we did, we used different steps of centrifugation, washes, centrifugation, and then lysis, and then put it on the strip, and it worked quite well, be it with Carba5 or CTXM. Then we thought, okay, we are probably not the only one. And then we looked in the literature, and in the literature, indeed, many different places evaluated directly on blood cultures. And in every case, it worked. And usually they compared it with molecular biology. And in most of the cases, the Carba5 was superior to molecular tests just because Carba5 detects all the IMP variants, whereas molecular testing, especially the studies that were done in Southeast Asia, failed to detect IMP2 variants, which gave them much lower results. And the assay was also compared with biochemical assays, and again, with biochemical assays, the results were quite comparable, which is very good. And then well, I need help. Oh, now it appears. You have to come so that the slides appear. Okay, well then there's a recent study in Singapore where they compared this time Carba5 with whole genome sequencing, molecular testing such as GenExpert. And again, Carba5 had nearly 97% good results compared to whole genome sequencing. And the main difference was that in some isolates they detected a gene, but the gene was not expressed. With Carba5, you detect a product, you detect an expressed gene, which is different from molecular testing. And of course, the results with GenExpert was lower because IMP2 variants were not detected. And in Italy, where they use this assay now in routine on blood cultures, they have really decided that this is a very valuable assay to infer very rapidly the presence of carbapenemase in blood culture, and this result is useful for implement rapid treatment of the patients. What about rectal screening? Classical rectal screening, you take a rectal swab, perianal or anal swampling, you spread it on chromogenic plates, you wait 24 hours, you see the colonies, and then you can test your colonies. Or sometimes you can do enrichment culture when you have doubt, maybe it's a recent colonization, there's not enough bacteria in the digestive tract. And then you can do your favorite immunochromatographic assay or whichever assay you want to do. But still, it will take 24 to 40 hours. Can we do faster? Yes, we can, of course, we can use molecular tools, and that's where really molecular biology is very strong, because it can detect basically one bacteria in your sample. But our Greek colleagues, as they have a lot of carbapenemase in their hospital, they wanted to know, can they use this device also on patients that are in ICUs in Greece? And that they did it. And they found that in 80% of the cases, just by doing your rectal swab samples, the transport media, put it on the strip, you have 80% of positivity as compared to molecular. And if you do a 3 hours incubation time, they reached 100%, which means with 3 hours of incubation of your rectal swab samples for ICU patients, you had 100% of agreement with molecular biology. But of course, the price is very different. So then we thought, okay, doing these washes, the centrifugation washes, it's a lot of hands-on time. And hands-on time, this is very valuable and very costly in Europe. So together with NJ Biotech and the CEA, we have developed a little assay which is called the BL Detect tool. I don't know what the name will be at the very end, but it's the prototypical BL Detect tool. And we have evaluated it directly on urine samples, blood cultures, and rectal swabs. And the principle is very easy. There is a filtration device. you take the liquid by a syringe, you filtrate, you take the filter cup, put the filter cup in the tank, put your extraction buffer, and then you screw the cap on the tank, and by screwing it, you will put the liquid through the membrane, and then you take the device, put it on the strip, press a little bit, and the liquid will run on the strip. And this together takes like less than 5 minutes, and within less than 5 minutes, plus the migration time, you get the results directly of these samples. And when we evaluated it on urine samples, on blood cultures, it worked perfectly well. We had above 95% of specificity, sensitivity, positive and negative predictive value. On rectal swabs, it was a little bit lower because we also included patients that were coming from other wards than ICUs, which probably had a lower amount of bacteria in their digestive tract. But this device can help you within 20 minutes from a blood culture to let you know whether there is a carbapenemase present or not in the patient. So AMR is a big problem. It represents really a public health threat all over the world. And we have this projection by Jim O'Neill, which says that more people will die by the year of 2050 than cancer if nothing is done. And today, we still don't know how we can cope with this problem. And everywhere, AMR increases, continues to increase. So we're still a little bit behind of what we should have. Probably we should have no decrease, but the decrease is not coming. And we knew that new molecules will arrive, but as we heard before, most of these molecules, even though they are new, we know that already a resistance exists, such as E. coli that has this insertion in PBP3, which will be resistant already to astronamavibactam, to ceftazidimavibactam, to cefiderocol, and probably other molecules will also fall into this category. So the only thing we can do is prevent the bug to spread until we get really new molecules with new targets and things like this. And the only way we can do this is use rapid diagnostic. Rapid diagnostic is really the key point, which will allow you to use the right antibiotic to treat the patients, but also to maybe isolate the patients, or at least to have a knowledge that the patient is a carrier, which may prevent also transmission to other patients. And we have now many assays available. Most of them have been validated for the global carbapenemases, the 5 main carbapenemases. None is perfect. This is something inherent to biology. You will always have situations where you will miss something, but it's your job to know the limitations, and then you can use them very efficiently. And lateral flow immunoassay, at least for us, we have now completely moved out PCR from our lab, and we do everything now with lateral flow immunoassay, because it's just much faster and it's more specific and sensitive than molecular, on colonies at least. And the advantage also of lateral flow immunoassay is that you can very rapidly adapt it. We have seen that we missed some IMPs. In less than 6 months, the new assay, the one that is now commercially available, includes all the IMP variants. So it's something that can be adapted very rapidly, which is almost not always the case with molecular assays. And whatever you use, you always have to position the assay based on your epidemiology and based on your risk. You have to know where the danger is coming from, and you have to target these patients. It's impossible that you target everyone, so you have to have a risk-based assessment, and if you use them in a risk-based assessment, it helps a lot to reduce the spread of this bacteria. And just to finish, why rapid testing for AMR? Because of hygiene, because of treatment, and because of the reduced cost as compared to an epidemic outbreak. And just to give you some hope, and this is a paper that I like to show because it really illustrates the beginning of carbapenemase-producing isolates in Israel, where they had an increase, incredible increase. They were everywhere in every hospital. And at one point, they decided to do rapid diagnostic. In this case, they developed an in-house PCR, and they used the in-house PCR to sort the patients, positive, negative. They cohorted the patients, and within 1 year, they managed to go back to the basal level of the carbapenemase producers. So even though you think everything is lost, there's no chance, there is still a chance if you do the right things, at least for the hospitals. Of course, what happens in the community, we cannot control it, but at least in the hospitals, hospitals need to stay safe, and the way to keep them safe is to prevent these bugs from spreading. And here's just to acknowledge some of the people in my lab that participated in these works and that shared some slides with me. Thank you very much for your attention.
Thank you, Dr. Nas. It's really promising diagnostic test for management of the multi-drug resistance gram-negative bacterial infection, because it is rapid as well as it
Yes.
that is more advantageous, particularly in ICU cases, stream infection, because we can do directly from the blood. Yeah. In urine also, particularly upper urinary infection, we can directly do from there also.
For urine, you have to be careful that you have a positive gram stain. You get at least 2 to the 5 bacteria per ml. The urine cases you have to, it's no problem. But with E. coli, the breakpoint is 3. So these you may not see them, but with gram stain you won't see them either. So for us, the criteria is positive gram stain, we can do the test. If the gram stain is negative, then we don't do it.
Okay.
For gram-negative
If we take
bacteria.
the urine,
Okay, thank you very much, Dr. Nash, for your nice presentation. I think next speaker is Dr. Dhruv Chaudhary. Dr. Dhruv Chaudhary. So he is professor and chair of pulmonary medicine in PGI Rohtak, and he has been the president of Indian Society of Critical Care Medicine. And he has got a lot of projects he has conducted, around 16 projects submitted by various agencies, and he has been chair and member of various technical committees, especially of ICMR. So Dr. Dhruv Chaudhary, please.
Thank you, Professor Prasad. And right in the beginning, thanks to my gratitude to the organizers and also Dr. Sherwal, Dr. Sonal, and the local one to giving me a chance to address the microbiologist. And right in the beginning, I say, as I was telling to Dr. Sherwal, there are 5 for clinical medicine and microbiology. And we are learning some of it by looking at WIM, etc. For a clinician like me, when we started learning it, it was more like a Farsi. So we are learning now, and we will say how we are utilizing it in clinical practice. And more so, being an intensivist and pulmonologist, we are facing this problem day to day. Excuse me. I think all of it is not moving here. Thank you so much. I think let's look at it. When I said in the beginning that one of the important issues for the clinicians, for us to understand the language and the jargon of the microbiology together. In this particular next 15 to 16 minutes, I shall be discussing about the need of new antimicrobial agents. Some of you already has been talked by my previous speakers. What is the application of the repurposed antimicrobials in clinical practice? And what is the efficacy of these antimicrobial therapy combination in clinical medicine? And what are the case studies? So we will be learning it by the case studies. And all of us know the ICMR, but in clinical practice, we all know in our intensive care units, our hospitals, that we are staring at a very high degree of resistance. We did one of the study with the Department of Microbiology on the people who were coming to us day 1 itself in intensive care unit from the community, and the rectal swabs were positive for, out of the 90, nearly 40 were resistant for meropenem on day 1 itself, both on culture as well as molecular diagnostic. So it's a huge challenge. Another challenge which we are seeing in intensive care unit is not only the bacteria, but the fungi, and especially Candida auris has also started coming to us. Salmonella overall this year and last year, seeing susceptibility has come back, and we are using those as and ceftriaxone. Finding on a there we need to have a solid and intensive programs of hospital control. Why we need antimicrobials? Most of us have discussed the crisis which is there in the are not working together. You look at the antibiotics, chances of focus on genes like cholesterol, the microbial peptides, look at nanoparticles. And we think broad materials, so you're looking in terms of low response, it can the ability resistance working classical tubercular therapy, which we are using it up to reduce the incidence of evolution of resistance and the broad spectrum right in the beginning, so that the sickest of the sick can be taken advantage. And we all know in the critical care, delay in starting of right antibiotics can actually increase mortality. So appropriate choice at an appropriate time becomes important for us, and the multiple targets. So the chances and probability of development of resistance becomes much less. Here, so clinical case. Now this is a gentleman who has been 82 years old, having a history of hypertension and recent onset diabetes of 4 months, presented to us with a high grade, producing minimal whitish to yellowish expectorations, and was also complaining of increase in breathlessness over last 3 to 4 days. He was conscious-oriented, he was having tachypneic, he was normotensive, he was having a SpO2 of 94, respiratory marked tachycardia was there, and breathing was rapid and shallow. Clinically, we could not detect any cyanosis. There was no clubbing, no lymphadenopathy or pedal edema. JVP was not raised. Respiratory system has all the features suggestive of consolidation on the right side. The rest of the examination was normal. We did an X-ray test in this patient. You can see a very large consolidation in the right lower lobe, which is there, and then also on the side we are there. We are now commonly using bedside ultrasonography for the detection, and the diagnosis was at that point of time. Over a period of time, this is at the day 1 at time 0, but it is over a period of next 48 to 72 hours when this patient presented to us on the Friday evening. His procalcitonin level was just borderline 0.42, but CRP was significantly elevated. And when looked at the blood sugar, you can see was markedly elevated, and so was the HbA1c. On the blood gases, he had a mild hypoxemia, and along with it, lactates were also just the borderline. This gentleman had received ceftriaxone and dexamethasone outside for a day. Then it was followed by a meropenem over a day and was subsequently referred to PGMS Roth with no response to the treatment. That's what happens in the clinical practice every day. So this patient came to us. What he looked, he was having a minimal expectoration. So we took up the microbiological evaluation. The first we get a report, fire available, found out Klebsiella pneumoniae 10 to the power 5 copies per ml, and it was having resistance to CTXM, IMP, NDM, MOXA 48 like that is we got it in this patient. Urinary antigens reports came later on. It was negative both for pneumococcal and Legionella, and blood cultures also of the, and the sputum both had shown to us Klebsiella, which was MDRO sensitive to polymyxin and tigecycline. That was the report which was given, but that came up to us. On this is what we got. So what? Obviously, it was a community acquired pneumonia with extended drug resistant strain, which of the Klebsiella, and then as we started him, initially he was put on when he came up spot on piperacillin, tazobactam with the glycoside. Looked at it, the pattern diabetic and also probability of his having a high drug resistance. But when the report came up, it was shifted over to ceftazidime, avibactam. B was also added. We did not use cholesterol because the lung penetration PK/PD of polymyxin B is better than that of polymyxin. If it would have been kidney, we would have used polymyxin E. So is there a classification of beta-lactamases? That's how we are clinically also. We are getting into your domain, and we are also discussing every day with our clinical microbiology friends. And this is the classical Ambler classifications which we are looking it up. It helps us in the choice of the antibiotics which are there. Obviously, this patient is multiple one, and you can see this as an NDM positive, and the data already have been shown. Means practically meropenem or imipenem would not have been effective. Therefore, we were left with no other choice to accept to use ceftazidime, avibactam, along with the polymyxin, a combination because person is a diabetic, elderly, and a high risk for complications in this particular case. So when you look at it, the patient started doing better over 7 days. Now, when you start looking at avibactam, vebrobactam, and rilabactam, which we are all talking much more, except avibactam, nothing is available in a combination as of today with us as far as the country is concerned. You will realize that to select avibactam, the combination we used it in terms of it, which is an AMP, that is a polymer B, is to ensure the right at the beginning. It does. I must confess, it took us 24 hours before we had next day when the patient came just over to the appropriate antibiotics. Now, these are the classifications. I will not go into it, but what is very important is that if somebody is having a KPCs, again, ceftazidime, maybe bactam is important. Somebody has a impervim, again, ceftazidime, maybe bactam there. But again, because the data which has been shown, people tend to use it with azithromycin. Our institution, azithromycin sometimes is not available, so we are using much more with the polymyxin, or even when we are finding out sensitivity with the aminoglycoside is better, so we are using a combination of ceftazidime, avibactam with aminoglycosides. Unfortunately, outside I am seeing that practically the use of aminoglycoside has gone away, and that is surprising because probably it is the corporate, the way they are moving, people are not using, and amikacin is still a big savior for us. These are the KPC Klebsiella pneumonias. I will not go into detail about it. Already too much has been discussed. What is important is we need to see that ceftazidime, avibactam, cefiderocol is available now. And also we are using sometimes tigecycline. But tigecycline we are preferably using in patients who have intra-abdominal infections because here the dose requirement is much higher because of the specific pharmacokinetic properties. And when I'm writing any antibiotic, I need to be very, very clear that what is the, I need to be aware about the PK/PD of it. Like no beta-lactam in my intensive care unit or my unit can be given in a bolus dose all the time because they work with an MIC, so has to be given in an infusion form. Yes, these are the approved for the severe cap. That is what IDSA has come up. But I think it is still not applicable to our country because we need to prove it up. It's a good MCQ exercise, but from the practical purposes, it's more of a theory than a practical thing, so I will not be discussing beyond this. Let's look at the case number 2. 22-year unmarried male college student came to us with the organophosphorus poisoning. And this patient obviously had a frank features of OP poisoning, was intubated, ventilated, and over a period of time, what we found that by 7th day started showing signs of worsening hypoxemia, fever, and purulent ones. At that time, procalcitonin was high, and then we evaluated it up. Obviously, this patient has a second onset of a fever, so VAP incidence tends to be quite high because they can't cough, they have a lot of secretions and other problems. So this gentleman had a clinical diagnosis of VAP, and that's what we look at. The X-ray which we did day 1 was normal. When we did it on the X-ray on the second day, you will see multiple lesions. You can start seeing on the left side, air bronchogram is there, and this patient had a classical VAP in this setting. So what we did at that point of time, again we went for the microbial confirmation. We know that Acinetobacter baumannii is not uncommon. What was very surprising was that we had a combination of H. influenzae and Acinetobacter baumannii coming up into it. Now, we used to divide VAP, early VAP, late VAP. Early VAP were the one which were common bacteriological agents which were there. And the second thing which we need to differentiate in intensive care is this Acinetobacter baumannii is a colonizer or is actually causing the disease? Now, we have 2 organisms. And another thing which we are very commonly finding with these molecular tests is the presence of the virus and the bacteria. So probably we need to redefine our etiology in our context, and it looks that viruses probably are predisposing very significantly to secondary bacterial infection and pneumonia in our cases. So this patient again, because of the fact that has an NDM and VIM, and for the VIM is the type 1, so we used piperacillin tazobactam, and NDM was also there, so we used in this patient polymyxin. So we need to be careful when we are interpreting, in particular, the biofire testing and multiplex RT-PCR. In the biofire, we are looking at it 10 to the power 7 and above bin when they are coming up to be a part and parcel clinically with clinical correlation. That is also fundamental. It has to be correlated clinically, when it is there, we are using it up. Again, we use it and patient did well. Now, the question is, I know it's a very crowded slide, and I'm sorry, we took it from the up-to-date 9th November. But what is very important is that the crab or the Acinetobacter baumannii infection is a pain in the neck for the intensivist anywhere in the country. And the question is how we look at it. I'm sorry, it is too small actually in it. What is very important, we need to look at it is the person is sickness of the patient. If it is moderate to severe illness, then we start looking at it, the first line, second line, and third line drug. The first line option of the drugs are the beta-lactam, which practically, if you are having a WIM or other hydrolases of a group A, are not effective because they are ESBL. So we look at the second line agents in these cases, it is polymyxin, and along with it, the polymyxin is the tigecycline, and also sometimes the cefiderocol, which is becoming now available, it is there. But by and large, because of now the availability of polymyxins B and E, both we tend to use it much more, but we always use Acinetobacter with a combination therapy, never with a single drug. Both Pseudomonas as well as Acinetobacter in intensive care unit. And again, you will see our savior continues still be dominantly polymyxin. And along with it, when we have to use Acinetobacter in these things, we are either using tigecycline along with it, that is the one which is commonly, or minocycline, or alternatively, now we are using your ceftazidime avibactam plus azithromycin. That is a combination which is there, so it becomes 3 drugs. That is what is being used as of now. Case 3 again is something we need to look at it. Newly diagnosed with diabetes, persistent fever of a 1-month duration. He had smoker and also as a farmer. Investigations we found, you look at it, how high HbA1c was, 13. And in this patient, we found out, again has a pneumonia, we found out Klebsiella pneumoniae, same pattern, Vim, NDA, Boxa 48, IMP, KPC. But we were uncomfortable because for practically he has been on antibiotics for last 4 to 6 weeks. What is happening? So we did a BAL in these cases to have the access to the lower respiratory tract. And here we found KOH stains for fungus were positive, and there was a bit of a dispute in the lab between aspergillosis or mucor, but the culture turning out to be rhizopus. That's what it is there. This was the first, you can see the classical bird nest appearance and a necrotizing pneumonia right up and both left and right side. So this patient initially, because he had received everything, so he was again put on combination of ceftazidime, avibactam plus polymyxin. He was not improving as much. Once the report of KOH came to us in 48 hours, 70 hours, we put him in additional to liposomal amphotericin B. And this patient was suspected mucor. This is what you can see, deterioration had taken place over a period of 11 days. So we continued the antibiotics plus antifungal. Later on, this patient started showing improvement, and he was given roughly 4.8 grams of amphotericin, which is liposomal. Please also look at it, which liposomal. There are multiple types of 3 types of varieties are there, having a different pharmacokinetic property. So right choice is the important thing in these antibiotics. And when we looked at it, after that on the discharge, we put this patient on posaconazole, and he is receiving it. He continues to be on treatment, and you can see with this treatment, he has started showing an improvement. So my dear friends, it's not only bacteria which are looking, we are also looking in terms of management, in terms of fungi which are coming to us, which are playing an important role. And last 3 months, why we put this up, in my intensive care unit, we have 18 patients of aspergillosis and mucor of the lung admitted with us, and all being diabetic uncontrolled. So this is a very important risk factor, all receiving all sort of antibiotics. So they had 2 or 3 hospital admissions and then coming to us. And antibiotics, when you're using extensively itself, become a drug pattern for a fungus which was there. So that's again an important risk factor which we need to see. So the repurposing of antibiotics, this is a beautiful article which is written in the Frontiers of Pharmacology. Repurposed antibiotics are good, but they have put a selective pressure on bacteria. Without prescription, over-the-counter risks have been extensive, and introducing co-resistance to other antibiotics. So it's not only that repurpose are a panacea for us, they may help us for some point of time. People are also using today the resistance breakers, like the combination which has come, ceftriaxone and piperacillin tazobactam, coming with EDTA and sulbactam, which is being used, which is being tested now. Trials are going on. Anecdotal report, we have also used it. It does make a difference, but we need to have a large-scale study in our context to see whether it can make any difference or not. Now, these are the mechanisms which are there. I'm not going to get into bore you at the end of the day about it, but what is very important is we need to realize, and I tell all the time, that the God has given this universe not only to us, but to the bacteria also. So we need to be sensible about it, how to go ahead, what we choose. Probably we thought that the word is only for us. I think that is very, very important for us. So when you are doing the repurpose in our activities, our actions also need to be there. Besides choosing the right, no doubt about it, we need to have the right drugs, right timing, right doses, knowing right pharmacokinetics and dynamics. Only then, whatever the drug you may use, will use it. And there will be a time limit now, like an expiry date for all antibiotics, which are coming to us the way we are using it up. I think that's very important. I will not go into it. To summarize, my dear friends, MDR is rampant in community as well as in hospitals. So right choice of antibiotics, the first right choice is the key. But it does not mean we overuse it. And once you have done the right choice, you will see results are better. But at the same time, if the culture comes up, if I have a drug-resistant genotype, I have a phenotype. If the phenotype tells it is sensitive, it is responding, I will go by phenotype, then by the genotype, because that is telling me the actions if that drug has been tested, and I will, from the broad spectrum, come to the narrow spectrum. Molecular diagnostic tests are going to play a key role with their limitation, and I think now it's a time, and I will request Dr. Sherwal, Dr. Pallabre, and Dr. Prasad, all of you are sitting here, to tell us where do we put them into evaluation of these cases, because the lab cultures take time. They are extremely sensitive, that is their thing. Probably for the diagnostic, yes, but not for the follow-up. We need to now put them in the algorithms of the management of community-acquired pneumonias, and multidisciplinary approach is the need of the hour. The clinician must speak with microbiologists, vice versa, and also pharmacologists and pharmacists. We have to change our culture, change our attitude, change our practices. I think that is a message I would like to give with it. And obviously, need of the MDT is the need of the hour to manage complicated infections, and the famous Bollywood lines, which they say, "Yehi hai right choice, baby," but that only possible if we understand and we realize that our every action will retard and will have an action from the microbes. Therefore, thank you very much for the patient listening. We need to here to protect and preserve antibiotics and humankind. Thank you very much.
Sir, may I ask you something?
Please, I will be more than happy.
Sir, regarding your long time experience in the CCU, so last 2-3 years we are seeing much more emergence of some newer agents like Chrysobacterium indulgens, Flavobacterium meningosepticum, Elizabeth kingia, like that. So what is experience about you?
Ma'am, Streptococcus, etc. is coming, especially since we have started using automatic ID systems. They have started coming much more. And most of the time, our impression is they're never the first time. It's only when the people in the hospitals or ICUs for a period of roughly 2 to 3 weeks, it's that point of time we start coming up, A. Secondly, we look at the pathophysiology of sepsis. This is a phase of immune senescence, where itself because of sepsis you have immunosuppression. That is the time it is coming up. We often on in 2-3 months we see one of these type of coming up. But by and large prognosis is poor, irrespective of the fact whatever the antibiotics which have been written and which we use against them, by and large.
Sir, can I ask another query from
Please.
the first case? And you have cultured Klebsiella pneumoniae both from sputum as well as from blood.
Yeah.
But the PCT was almost, it is normal 0.48, I think.
Yes.
So how you...
Ma'am, procalcitonin, if you look at the ROC curves, they are around 0.81. So that is why we go for the diagnostics for it. For us, initially it is more of ruling out, and later on for practicing reduction or stopping the antibiotics. Therefore, this discordance can come up at the lab at what point of time you have done it. Yeah. So that makes the difference. But we had gone in this case by the CRP. You see, the poor man's inflammatory marker is CRP, especially once we know it. It's so sensitive it gives us. But you are right, procalcitonin, not necessarily that it will be high all the time. That's what I say. In roughly 80% sensitivity specificity, that is why nobody has put it to make a diagnosis of a bacterial infection. Ruling out, it may be. It's not for the ruling in.
But you have radiological evidence as well.
That is there, sir. So it's a comprehensive clinical lab plus response to the treatment. That's how we decide about it.
So, but it is surprising that you got an XDR, a patient coming from the community with an XDR Klebsiella.
So that's fortunately
that
our
is
savior,
unfortunate.
honestly speaking.
The patient might have earlier history of admission. So that
Absolutely not, sir. He was even denying diabetes. Till we found out that he has a metformin, we found it out. And probably the major risk factor had been. Sir, the problem what is happening is most of the people are very fond of now chicken and all. And when you look at the poultry as a growth promoters, we are all talking about writing it. Poultry and the veterinary is putting a lot of pressure on it. So somewhere, this was one of the very important persons within the state. And they have been interacting with the people. He was an 82-year-old retired army personnel. Even we were surprised. We thought it's a pneumococcal, the way it was. But when it turned out to be Klebsiella, it was 10 to the power 5, still not 10 to the power 7, but clinical picture was there. And later on, we were proven right that we did the right thing. But it took us around about 2 weeks of therapy before he settled down. That's surprising, but that's where the molecular tests are coming to our rescue. And that's why I'm saying, gradually from skepticism, I'm going it that it should be part of it. But how to, because the cost is so high. We are in the government sector providing free of cost, but otherwise it is 10 to 15,000 rupees in the practice.
So, yeah.
Thank you.
Support from the microbiology department.
I think your answer, the question is important. Your question is very important. Ma'am, only thing I can say is your answer to your question is only three alphabets. Y-E-S, yes. And in fact, the last slide they've changed up. I had thanked and acknowledgement by Department of Microbiology, Dr. Aparna Parmar. Nidhi is here. I have been after them. I breathe down their neck. In fact, I tell them to send their postgraduates for the rounds. But my request is, answer I said is yes. Let the microbiologists get out of that thing. This is the basic department. It's a hardcore clinical department. We need your support. With your support only we can move it up and start putting your postgraduates for rotation in internal medicine, pediatrics. They will also learn. They will also improve. And the third place where I say is the pharmacology. They tell us a lot. It's a combination of it. That's why you will see in the past, Professor of Medicine used to be Professor of Clinical Pharmacology, but never of a clinical microbiology. So your support is required. Dr. Pallabhre sir is sitting here. PJ, I think, never it used to go away with it in the morning sessions, discussing it up. They were part and parcel, integral part of the team. I think that gives your answer. And thank you so much. ICMR, we are part of it. PJ Rohtak is one of the part of surveillance of it. And I will request you also to tell our department to more actively participate in clinical thing. Rest, they are doing a wonderful job. Thank you so much.
Thank you very much for your excellent presentation and
discussions. Thank you, sir.
I think this sort of discussion is more required than, I think the case-based discussions are more important, and that will also inspire the young microbiologists to collaborate with the...
Absolutely, sir. That's what I was telling Dr. Sherwal also. Time to have a joint fellowships, joint programs, where we also learn. We have learning in a hard way because as an intensive care, we can't get out of the MDROs. Whereas in the general medicine wards, probably that is not the thing. And we are having a daily discussion, debate with our colleagues from cardiothoracic, from surgicals, because they just had a pen of six antibiotics going on. They're scared something may not go to the implant. I think we need to stop that practice. That will happen only when we sit together, work as a MDT. Thanks, sir. Thank you very much.
Thank you very much, sir. So it is the time to call upon the dais Dr. Kamini Walia. She is PhD MP, Scientist G of ICMR. And she has done her PhD from PGI and Masters in Public Health from John Hopkins.
I think, no, this session is over, I think.
Sorry, ma'am, this is for the
but
next
there are a
session.
lot of papers here which is creating confusion. So from next time, I think that you should put the program here. There is no program and the CV of the speakers, and you can remove the old ones because there is a lot of papers here. All the papers have not been removed.
Sorry for the inconvenience, sir.
Okay, thank you very much for your nice audience.
We would now request our chairpersons to kindly felicitate our speakers. Professor Ken Prasad, sir. Please felicitate Dr. Itisha Gupta, ma'am. Dr. Theri Nas. Dr. Dhruv Chaudhary, sir, please come. Thank you, ma'am. Thank you, sir. Before our last session, last call for the IMM quiz tomorrow. All those candidates who are interested, please report in the hall at 8 a.m. because the quiz will start by 8:30 a.m. We have again displayed the number of persons whom you need to contact for the quiz. Kindly send your name, your email ID and phone number on any of the two numbers displayed. Our next session is Strategies and Synergies Combating Antimicrobial Resistance. We would like to welcome our chairpersons. First, Dr. Pratibha Narang, ma'am, who is ex-director, professor, dean, Mahatma Gandhi Institute of Medical Sciences. She has published nearly 129 papers, guest lectures 61, orations 10. She has been awarded with Lupin Thai Gold Medal Oration Award, Dr. Walker's Lifetime Achievement Award, and IM Endowment Award. Ma'am, please come on stage. Our next chairperson is Professor Bashir Ahmed Formde, Professor and Head of Department Microbiology, Sher-e Kashmir Institute of Medical Sciences. He has been honored with Lifetime Achievement Award in recognition of outstanding contribution during COVID-19 pandemic. He had nearly 110 publications and more than 18 projects, with ongoing 8 projects presently. So